All right. I'd like to start by thanking everyone for taking their time to come here today. I guess some of you know me in my name, but other people just know a bunch of emails you got from me. So my name's Tom Joseph, and I'll apologize in advance for all the emails, but it's okay. We got you here today. We got a very aggressive agenda today. We're going to be covering a lot of material, but we want everyone to be comfortable. To that end, please feel free to get up and find your way to the restroom, which is out that door and to your right if you have the need, or to replenish your refreshment, coffee, juice, whatever. All right? Because we want to keep this as casual but as informative as possible throughout the day. We've actually tried to build in some 5-minute breaks in the agenda, and as long as we can keep on track for each segment, those should work. Now, part of the process that we had set up was to ask everyone to take a survey prior to the meeting, and I thank those that have participated in that survey. We have that information, and we will share it later on in the meeting, because once we get through some of the material, we're going to ask everyone to take that survey again. That will give us some judgment of whether or not we've moved a needle and moved anybody's minds as a result of the meeting information. Actually, today is the result of 11 months of planning, collaborating, listening, and taking action on the part of the project team that's been involved. And based largely on the suggestion of Dr. Dale Robertson, who is our USGS team leader, who you'll meet soon, we put together this event. And this event has not been a singular effort, but one of a collaborative effort of the 3 lake associations. The planning, the preparation of the work packet, and so much more. So to that end, I'd like to offer a special thanks to the steering committee who have worked so hard to make this happen today. Jeannie Williams and Rick Dornbos from 3 Lakes, would you please kind of just let everybody know who you are? Rick, Jeannie, thank you very much. Thanks for putting up with me for all these months. Peg Comfort, say hello to everybody. And Trish Narwood, okay, they have been just absolutely terrific. And if you want to really express your thanks for all the technical information in the packet, Trish is probably the key person, although everyone contributed. And she had a couple late nights, I think, along the way to make it happen. And then we've got the 3 people, myself included, from the Torch Lake Protection Alliance, Dan Shazowski and Bob Milleron. Is Bob? There's Bob. Okay. So that's our team, and that's the ones that brought together. So one thing to keep in mind is that this event has not been the destination, but rather this is the start point. Going forward, our vision is that this will become an annual State of the Lake event, and hopefully we are off to a good start today. We've got 2 key objectives of this meeting, and we try to publish it enough so people had it in their minds. We put it on the invitation and we've broadcast it several times. But the first of those objectives, to evaluate the hypotheses of the golden brown algae formation and try and identify what we believe would be the top 3 and develop a plan to pursue that. And the second, and equally important, is to work on the foundation of a long-term monitoring and assessment program for Torch Lake going forward. So with that in mind, I'd like my co-conspirators, if you will, Peg Comfort from the Torch Conservation Center and Fred Siddle from the 3 Lakes Association to come up and join me so that on the behalf of the 3 organizations, we want to welcome you to this prestigious event together. Fred, you want to come up? Peg, you want to come up? I'd like you to introduce yourselves, and then what we're going to do is we're going to go around the table of this group, who are basically our work group, and ask them to just do a quick introduction of themselves. Okay? Say hello, everybody.
Hello, everybody. And just a little housekeeping. Help yourself to coffee, tea, juice, water. If there's anything you need in terms of refreshments with muffins, etc., just let me know.
Okay. One other item. Mike Strange, who is our video person that's taking this all on YouTube, and we have a link to that if you want that for future reference. He has hearing impaired devices if anyone needs them if they're having trouble talking. And to that end, the speakers, as we go through the other presentations and question and answer period times, make sure you're close to the microphone, not so much for the people in the room, but so that it gets on the video correctly. All right, and I don't know if you can tell if what we're doing is working. All right. And then I've got one last procedural thing. We have time at the end of the agenda for Q&A. We have papers and pens for people to write down your questions. We won't be able to maintain our agenda if we constantly stop for questions. And a lot of times people have a question that will be answered in some other presentation. But we don't want to leave without having questions answered. So please write down your questions during the meeting, and we'll have time at the end to address them.
Thanks, Tom. I'm Fred Siddle. I'm on the board of Three Lakes Association, and I just want to steal a couple of minutes of your show just to give a little introduction to people on where we've been over the last 10 years. In 2014, the end of 2014, and in 2015, Three Lakes Association, started raising the public awareness of golden brown algae on Torch Lake. And by the end of 2015, we had over 100 benthic groundwater samples analyzed. By the end of 2021, that was many, many hundreds of samples that included comparisons of nearshore water to chemistry to deep basin water chemistry to groundwater chemistry, even measuring the available phosphorus to the algae right in the few centimeters of benthic sediment and directly above it. And this was all a very highly structured sampling plan. This was not monitoring. These were with replicates, sometimes repeated from one year to the next, with the objective of answering specific questions. And all that was done under the guidance of TLA's longtime water quality chair, Becky Norris. Becky wanted to be here today, but for reasons beyond her control could not join us, but I'm sure she'll be following this proceedings online. I don't think Becky has received enough acknowledgement of that 7-year effort because she not only headed up water quality, but did most of the field work along with help from some very dedicated volunteers, Trish Narwhal, Dean Branson, Rick Dornbos. There was a lot of field work involved in all this, and this created the data set that becomes the starting point, as Tom mentioned, for today's proceedings and for our experts to get a jump start, on thoughts of what we can do about golden brown algae. So I wanted to make sure that we give a good shout out to Becky for all that work. And the board of Three Lakes is very much looking forward to this unprecedented assembly of experts. I think it's really great to have so many partner organizations now stepping up, and I'm looking forward to learning a lot today.
Okay, well, thank you. Thank you very much, both of you. So we have the agenda put together. We've made it public. I don't have to go through it, all right? And I hope that we are successful in accomplishing a good cross-examination of dialogue. Now, when I talked about the questions, that does not include this panel. We expect this panel to question, challenge, whatever, the discussions that they have amongst themselves. That's how we're going to learn. So without any further ado, and right on schedule just about, we're going to go to the next item, which actually, Peg, you're the first one up. Come on back. Peg's going to give us a brief historical information on the studies at Torch Lake.
Well, when I asked Tom I probably don't need this. I'm not going to stand in front of that.
It's okay.
I didn't know where to start. So I thought I'd start at the beginning. Our family, I told him I'd cry at this meeting. So get ready for it. Our family goes back to 1907 here. And so we have a deep deep roots, as many other people do in this Torch Lake community. And our son was born here, so he's actually a local. We've lived here 40 years, but we're not considered local yet. And never will be, by the way. So there's a distinction there. But we've been involved with a lot of activities.
Okay, would you be able to use the microphone so that our remote
Sure.
users will hear you?
Is that better?
If you move it more in the center, they'll be able to see you as well.
There's a
Yeah.
bright
It's-
light.
It's off.
I got rid of the
It's
bright light.
off. Yeah.
Okay, are we on now? Are we good?
That's
Okay.
good.
Thank you, Peg.
Okay.
Sorry for the interruption.
That's all right. At any rate, our son was born here in 1984. We built a house on the lake in 1992. And when he graduated from Michigan State in 2006, he never imagined that 20 years later, when he brought his children here, they'd have to wear water shoes because of the zebra mussels would cut their feet. He never imagined that they wouldn't be able to look for Petoskey stones because they're all covered with algae. He never imagined that they would be unable to swim in the lake because of swimmer's itch. So in his short lifetime here, he's seen these dramatic changes. So we feel a real strong sense of urgency about this matter. And we're really grateful to have all of you here to help us figure out not just what the scientific causes are, because there are many. You know, an ecosystem is complicated. And all these things are interrelated, and we understand that. And it's great to get the science, but we need to know what, if anything, can we do about it? What can we manage? People are asking, what can we do? So that's a critical factor as we go forward today. So welcome.
Thank you. This is wonderful. Okay, so we're going to move on, and Doug, you're gonna be up next. You can present from wherever you're comfortable. You can sit, you can stand, you can point at the screen. Dale, you got Doug's presentation ready to come up? All right. And the first part of your presentation is trying to go through the trend analysis, right?
Right.
Okay, thank you.
Okay, you hear me? I'm Doug Endicott. I work for Great Lakes Environmental Center, and I was asked to come today and talk about trends in water quality in Torch Lake. So let's get right into it. I think I'm holding it upside down, aren't I? Okay, so here's what I'm going to talk about. We're going to review monitoring data for following water quality parameters, Secchi depth, the optical measure of clarity in the lake, total phosphorus, the concentration of the limiting nutrient in most lakes, chlorophyll A, measure of phytoplankton concentration, and chloride and ion associated with anthropogenic activity, including road de-icing. So these parameters have been monitored annually since before 2000. There was sampling prior to that, tended to be a little more sporadic, going back to the 1960s and 70s. The presentation I give here will include data results through 2021, and I'm also including a 2017 National Lake Assessment condition estimate for the health of Torch Lake. So we'll start with Secchi depths. This is volunteer Torch Lake data, years 2000 to 2021. This is from the North Torch Lake Deep Station, and you can see Secchi depth for each of the years pretty much. Like I said, that's North Torch Lake. They also measured Secchi depths in South Torch Lake. That's what the data looks like. In most years, there are 10 to 20 Secchi depth measurements. A few years, there are fewer measurements, and that maybe creates a little problem interpreting this data, but I think we can get past that. It's a little hard to see what's going on within any particular year with this kind of a display. So what I did is I took all the data and I plotted Secchi depth as a function of the day of the year. So I don't know where 1st of July is, probably somewhere around day 200. Can't remember everything, but this is all the data. And what you see is that as the season progresses, Secchi depth declines in Torch Lake, and it does so in a pretty consistent manner. There's obviously variation from year to year. That's the North Station. The same thing going on if you look at the south station. And so what do we say about this? The Secchi depths decline over the summer, and we pretty much know that's due to calcite or solid calcium carbonate precipitation. So calcite is bad for water clarity, but it's, as we'll see, it's good for phosphorus removal. So once we look at the seasonal variation, now we can look at some annual things going on. So I just plotted here the annual average Secchi depths, and the bars are the minimum and maximum value for each year. That's North Torch Lake. You can see it looks like a trend of improving water clarity from as you move through time. That's the North Lake. Here's the South Lake. Same thing's going on. Seems to be some indication that the lake is becoming clearer. That's Secchi depth. So now we move on to total phosphorus. This is CLMP data. It's measured in the spring and fall. This is going from 2000 to 2004, 2021. I got data through 21, although this data was most recently published in 2019. So there's a couple of years I just have spreadsheets of data. Regardless of that, this is a presentation of that data that my boss at GLEC made a few years ago. I've extended it here, and here's where I get to make my soliloquy. As some of you know, I was involved in the mass balance study that Three Lakes conducted in 2004 and 2005, and among other duties, I was responsible for QA of the data for the surface water, and that taught me a number of things. It taught me that water quality at the North Torch Lake Station couldn't be statistically distinguished from the South Lake. Same thing regarding nearshore water quality versus water quality at the surface once you went out into the middle of the lake. But the most important lesson I got was to distrust total phosphorus concentrations higher than 5 parts per billion. And, you know, despite some heroic efforts by Three Lakes associations, we continued to have sporadic total phosphorus measurements that would be higher than 5. These had statistical properties of outliers. I judged them to be non-representative of well-mixed conditions in a lake like Torch. And in a couple specific instances, we were able to relate the high concentrations to sample contamination events. So hence Endicott's rule, treat total phosphorus greater than 5 as an outlier. And so I take it out of the trend analysis, and here's what's left. And you see much lower total phosphorus concentrations as you march through the 2000s. In fact, these are really low concentrations compared to Michigan and other places, even nationally. And in fact, they are so low, they were too low for the laboratory to measure quite often. So this red area, these are all samples that are actually below the limit of detection. So what does that mean? So I'll just reiterate, the total phosphorus data is based on laboratory analysis using a 3 part per billion detection limit most of the time. Since the early 2000s, most of the data from sampling has been less than that value. And it's common to replace the below detect data with half of the detection limit for presentations. But in reality, all we know is that the total phosphorus concentration in those years is somewhere below the detection limit that's being reported. So here's a way to look at this. I've taken all the below detection limit data, which is this series of data here, and I've replaced the data symbol with just a bar going from 0 up to the detection limit. So we know the concentration's somewhere in that range. We don't really know anything more than that. So there's your interval of where I think total phosphorus concentration has been going back to 2004. And it makes it look more consistent with some of the more recent data, which, you know, they are very low numbers, but it's maybe consistent with what's going on. So it's difficult to, what I'm trying to say is it's very difficult to see what the trend is over this period of time because of the detection limit problem. Tip of the Mitt also has a monitoring program. They go out in the spring. This is their data. They sample surface, middle, and bottom in the lake, and their data shows what looks like declining total phosphorus concentrations starting in the mid-90s, going down through the early teens, and then maybe thereafter there's an increasing trend, possibly. And again, I applied my rule to it, so the above 5 values, but there are a few in this data set. I've censored them out. And in 2020, I don't remember who sponsored this work, but the consulting firm was ET&C. They did nearshore monitoring of water quality in Torch Lake. And so the blue line there is their summary of nearshore phosphorus concentrations. What you can see is that they're similar to the seasonal variation. In fact, it's similar to what we saw in 2005 in the mass balance data. And the spring concentrations are also similar to tip of the mitts monitoring data. I think the last thing I've got to show about phosphorus is that we also deployed sediment traps in Torch Lake 2 times, 2005 and 2021, to measure the loss of phosphorus by settling. Calcite, I mentioned, it absorbs phosphorus and removes the nutrient from the water column as it settles. This removes most of the phosphorus input to the lake each year. So if we measure total phosphorus settling flux, it provides a good estimate of the total loading to the lake. It's our rationale. This is what the data looks like. This is the concentration of phosphorus that we collect in the trap samples. Different seasons where we had traps deployed. From that data, we estimate settling flux of phosphorus. And to interpret that, we tend to focus on the stratified summer period because they avoid resuspended sediment. So that's those samples. The timing of the removal appeared to vary. In 2005, essentially all the settling took place in the late summer. In 2021, it was sort of divided between the early and the late season. This timing may be related to when the surface water warms, but I don't know that for sure. The sediment trap data, however, does not suggest significant changes in total phosphorus loadings or concentrations between 2004 and 2021. All right, chlorophyll A, moving right along. This is Volunteer Lake data and CLMP, somehow combined. Summer data from 96 through 2021. Again, so this is North Torch Lake. This is South Torch Lake. And again, as was the case with phosphorus, these are extremely low concentrations. And again, they're too low for the laboratory to measure in many years. So between 2004 and 2017, the chlorophyll concentrations were reported as less than 1 part per billion. So again, all we really know is that chlorophyll concentration in those years was somewhere between 0 and 1. We also see these very low chlorophyll concentrations being reported for 2020 and 2021. I decided I'd just present some seasonal data for chlorophyll to focus in on that. This is data from the mass balance study in 2005. Chlorophyll has these nice early and late season peaks and sort of lower values in the middle. Now I'm adding the 2020 nearshore data. Something different's going on in the nearshore region, but again, the concentrations in generally the same range. Now I'm putting CLMP data from 2020 in, which is the green line and the golden line, and you can see they've got some similarity to the shape we saw in 2005, but the values are significantly lower. And then in 2021, the bottom seems to fall out. The North Torch Lake sample, which is the red, 4 of the 5 values that were reported were less than 0.003 parts per trillion, which I'm assuming is a detection limit, but I don't know from the data I have. But, you know, that's almost nothing. So something's going on for chlorophyll. I guess that's maybe part of what this group will deliberate on. Anyway, I'm also going to show chloride data because I think Jan was interested in the possibility of chloride having a stressor impact on the aquatic community. Anyway, shows that chloride concentrations since the early 90s have at least doubled, maybe more than doubled. And again, this is, I don't know, I tend to hear a lot of talks at scientific conferences about road de-icing being the culprit here. We use a lot of salt to keep our roads clean. So anyway, the last thing, I don't know, I'm doing okay on time, I guess. The last thing I want to present is the National Lake Assessment evaluation. Pardon me? Oh, I'll just keep going. It's a measure of the lake's national level assessment of lake health. These are the sites that were visited in 2012 by EPA crews. There were some northern Lake Michigan northern Michigan lakes sampled. Torch Lake was not sampled. I think Crooked Lake was sampled, among others. I'm not sure exactly, but anyway, in 2017, Gleck was hired to apply the evaluation to Torch Lake. And so this is a presentation of the right. The bars, you can see their description over on the left-hand side. These are the different conditions that were ranked. The ones at the top are anthropogenic influences, land disturbance, lakeshore habitat complexity, and so on. The lower bars are all water quality parameters. And the way to read this is all data that comes from the 2012 Michigan subpopulation of lakes. And so you can see lakeshore habitat complexity of all Michigan's lakes, about 50% of them are red or poor, and then there's yellow, which is fair, and then the remainder is green or good. And so this just shows how the distribution of lakes in Michigan fall out in terms of these criteria. And then the white bars, those are where Torch Lake falls in terms of each of these factors. And so you can see for lakeshore habitat complexity, riparian vegetation, shallow water habitat, lakeshore disturbance, Torch Lake falls in the poor category of lakes. But for all the water quality parameters, which are the lower half of the bars, Torch Lake's way off in the very good quality. And so just to think about that a little bit more, forgot I put the arrows there. So the top one's mostly land disturbance and near shore, and the bottom one's water quality. But you know, you look over on the left-hand side is where they're evaluating the conditions where Torch Lake tends to fall out in the poor category. And it's worth pointing out that what this condition index considers to be really good is this. So it's possible that Torch Lake would never look like that, even in the complete absence of, you know, anthropogenic influences, to put not too fine a point on it. So I think that's all I wanted to say on that subject. So thanks for your attention.
Hey Doug,
Yes, sir.
try to hold your microphone a little closer when you're talking, please.
Okay. And for some reason, I've been asked to give another presentation here, so you're going to have to endure listening to me again. So I'm going to talk about modeling of the water quality in Torch Lake. And is this better? Is this clear? Okay. I'm sorry. I don't usually work with a microphone. So again, we'll start with an outline. There's two models that I'm aware of that have been previously applied to model water quality in Torch Lake. The first is the development of predictive nutrient-based water quality model. It was the work I did in 2005 for 3 lakes with DEQ funding. We used the Lake 2K model. It was calibrated to data from 2005. The other modeling effort was modeling impacts of calcite precipitation on an epilimnion of an ultra-oligotrophic hard water lake. That was Betsy Homan, Steve Chaffra's work from Tufts University. They worked with the data set from a year after that, 2006, and they used some combination of the Lake 2K light model and something they called the hard water lake calcite precipitation model. I'll talk a little bit more about that in a minute. And I'm also going to address some questions in the work group packet about modeling Torch Lake if we get to that. So anyway, the predictive nutrient-based water quality model, the work plan said we'd develop it as a tool for relating changes in nutrient loadings to expected short and long-term changes in water quality in Torch Lake. And those questions included, what's the baseline water quality of Torch Lake? Of course, realizing this is 2005. What are the current loadings of phosphorus? And finally, how will water quality in Torch Lake be affected by changes in nutrient loading? So the model we applied is Lake 2K. It segments the lake into 3 layers vertically. So these are all the inputs and outputs of water, and there is phosphorus and other water quality constituents following each of those pathways. And in each of these 3 segments, mass balances are constructed to compute changes to water quality over time. This is a schematic, which I have nowhere near enough time to go into in any detail, so you can all relax. But just to point out, over on the upper right, there are the phosphorus state variables. Phosphorus interacts with calcite and settles, which is a big loss mechanism. In the upper left is phytoplankton, photosynthesis and respiration produces CO2 gas, which becomes part of the dissolved inorganic carbon cycle, which is in the lower left. So I've left everything else out of here: hydrology and thermal balances, temperature, light model. It had a fairly elaborate light model in it. So just moving on, this was the calibration process. Again, I'm not intending you're going to look at this in any detail. The model was calibrated to data from 2003 through 2005, temperature, phosphorus concentrations, chlorophyll, dissolved oxygen, and Secchi depth. And again, I'll reiterate, this model was calibrated to 2005 data. A lot of things have apparently changed in Torch Lake, and so that should be borne in mind. One of the things we did with the model was to construct a total phosphorus loading estimate. And so you see this thing, it's almost like a peace sign, but it's actually the three slices of the pie. We found out that tributary loading, precipitation, and shallow groundwater all were major contributors of phosphorus to the lake, which surprised me a little bit. I thought at least one of them would kind of fall out as being insignificant. So this was the mass balance in numbers. Again, loadings, outflow through Torch River, particulate phosphorus settling, which we determined at that time to be about 90% of the loading, which was enormous. The mass balance indicated a small decline in phosphorus, more loss than entering each year, but by a relatively small margin. So the lake was fairly near steady state. The model demonstrated that Torch Lake was near steady state with respect to phosphorus, and that significant changes in total phosphorus loadings would probably be observable in a couple of year time frame. So this is the other model. It got published a few years ago, 2011, I guess more than a few years ago. Modeling impacts of calcite precipitation. And they, Betsy and Steve came up with some intriguing findings, and I encourage you all to read this. It's freely available. I think it's in the modeling packet.
It's in the packet.
Okay, so the findings were that the combined effect of raising temperature in the water and carbon dioxide gas transfer, they lead to calcite generation, and that's what explains the decrease in water clarity each year. And they parsed apart the issue of whether temperature change or primary production, in other words, algal activity, were driving the calcite precipitation, and they determined it was really temperature that was responsible. And finally, this is the one that really made me pay attention to the results. The presence or absence of primary production had a negligible effect on what's happening to clarity in Torch Lake. That pretty much contradicts conventional understanding of the relationship between primary productivity, algal abundance, and light clarity. So it's pretty interesting. So some work group questions. How will backmixing, lack thereof, in Torch Lake be accounted for in the model? We found that it wasn't really a factor. So here's my answer. We had a peer review in Torch Lake. Remember this, Dean? 2004, it was a freezing cold day at the yacht club. I think Ray Canale was there. He was wearing a sleeping bag because we were sitting outside for some reason. Anyway, at this peer review, 3 modeling experts, which I don't count myself among, but Ray Canale, Tom Cole, and Barry Lesh, they all agreed it was safe to assume that Torch Lake was longitudinally and laterally well mixed. And so I took their word for it. And the assumption was confirmed by total phosphorus data collected during the mass balance project, as well as other data that I think has been collected subsequently. Okay, moving on. Prior modeling of Torch Lake in 2006 did not predict the increase in golden brown algae. Why, and what's the implication of this gap? You know, it wasn't mentioned as a water quality objective when we started out the study. Somebody said, well, we ought to look at marl incrustation. And I'm not sure that's really the same thing. So, you know, we didn't identify it as an objective. Even if it had been an objective, golden brown algae couldn't have been modeled given the state of the science at that time. You know, what is it? Why is it appearing? How do you measure its abundance? What's the factor that limits its growth? They were all basically unknowns. In other words, you can't really model what you don't know, and these were all things we didn't know. A single phytoplankton state variable was used for chlorophyll A, which is, you know, which as I've shown, has been monitored since at least the 1990s. So we had data for that, so that's what we modeled. How will golden brown algae be modeled? I don't really know. It's, but I would say I would look at nuisance benthic algae modeling that's been going on in Lake Erie, you know, since I don't know, the early teens. It's been fairly intensively studied, and there was a conference that NOAA put on a few years back, State of Knowledge of Cladophora in the Great Lakes Workshop. They were dealing with benthic algae there, and I would point to that. Finally, what are the limitations of the Lake 2K model? For example, can the model separate nearshore and deep basin? So what's a limitation? It doesn't have a fixed benthic algae species in it. In its present form, the model can't distinguish nearshore from deep basins. It only has the 3 vertical layers. Let's see, what was the last question? Oh, how were the macros developed by Elizabeth Homa embedded in the model? I reached out to both Becky and Steve Chapra, and Becky said, I'm not sure why the calcite modeling work I did is being referred to as a macro, and which model they may be referring to, since Chapra may have added my modeling work to various Lake 2K packages. And Steve Chapra didn't respond to my question. In all fairness, I didn't send it out till Wednesday, so maybe he's retired. I mean, what do you expect? But anyway, so it's kind of a garbled message on that. I don't really have an answer for that. It may be that the work Becky did had nothing to do with a version of a Lake 2K model. Although Lake 2K does have a fairly well-developed light submodel in it. So I can't answer that question. And again, I think I'm done. So thanks for your attention.
So at this point, I'd like to take just a little bit of time, because the topic of modeling is very important to us going forward. And Dale, Jeff, and the USGS team have had a lot of discussions with us on how we're going to benefit from the existing models that have been prepared, and what can we do to optimize the model for Torch Lake. So if there's anyone on the panel that has a question or a follow-up question for Doug, this would be a good time to have some dialogue on that topic. Anything from anybody?
I have a question, but it doesn't relate to modeling.
Okay, fine.
You presented some chlorophyll data. Were they surface chlorophyll or vertically integrated? Or?
I think they're, well, there's other people in this room that probably know the answer, but I think it's photic zone, because they're always accompanied by a Secchi depth measurement.
So they're measured over the whole, they're integrated over the depth to the Secchi range.
Two
Or do we go
Secchi
beyond?
depths,
Well, Carol can answer that question.
Yeah, so the state CLMP.
Here you go.
Oh, sorry, the state CLMP.
You got to push it on.
Okay,
There
can
you
you
go,
guys
sorry.
hear me? So the Watershed Council's QAP says to collect the chlorophyll A sample at twice the depth of the Secchi disk. And I would think that the state's QAP is the same, since our programs were kind of created at the same time. So
Okay.
yeah, more than just surface, yep.
Any other questions?
I can elaborate on that with Jeff's question. The study that Doug was speaking of, the 2020 TLPA ECT, was surface samples. That was just a half meter below the surface, that data.
That separate chart that he had,
Yeah.
right?
Mhm. So that was a different data set.
I think you're next, right? Now that we got you up here,
I just had one quick question.
Chris.
How did you determine or how did you verify that the components or sources of phosphorus, the 3 different ones you looked at in the model, kind of match sample results? It looked like you were saying they're roughly equal contributors, the atmospheric, groundwater, and the tributary loads.
They're all, the loading estimates are all based on measurements, and they're inputs to the model. So they're not modeled by the lake model. They're fed to it as input data. Does that explain it?
I think so. I just wasn't sure how, like, you're simulating it in the model. So I just wanted to see how your concentrations and loads compared to what you were getting out of your model results.
No,
The-
what I
yeah,
was
again,
trying
the
to,
model's not going to do that.
what I was trying to say is we have to input those to the model. The model wants to know what is the tributary loading in March? Okay, you give it that number. That number is based upon an analysis of monitoring data.
Okay.
So it's less magic than you might think.
Okay, very good. Any more questions? All right, so we're gonna
Yeah.
ask.
Oh, sorry, Tom.
That's okay.
I got a question. Just so I understand the calcite phosphorus cycle. So it sounds like phosphorus binds to the calcite settles down to the bottom, but then the calcite dissolves and then releases that phosphorus on the bottom.
Actually, from what I've looked at and studied, it actually ends up in the sediment, bound to the calcite. You know, if you look at deep water sediment data from Torch Lake, it's 3/4 calcium carbonate. I mean, if anybody's dropped an anchor down deep in Torch Lake and pulled it up, it comes up looking like chalk. And so I believe that carries the phosphorus right into the sediment bed. The phosphorus concentrations we measure in our trap solids looks very similar to surface sediment phosphorus concentrations.
Okay. Well, so I guess, I mean, do you think any of that phosphorus that's settled down to the bottom would be biologically available? I mean, could any?
No.
No? Okay.
I don't
so
believe
it's-
so.
There was some core, you're familiar with the core study that was done in association with the 2005, right? We did some sediment cores for Torch Lake and compared them to 2 other lakes in Michigan. So you remove the cores and then you put them in a anoxic environment. And at least for that limited study, neither Torch Lake or Clam Lake or Lake Bel Air really had significant phosphorus release. So, but the other lakes did. Now, one other observation from that was that those cores had higher iron content. But in this particular study, they didn't do a real analysis of what was in those cores in that respect, but they could see that the cores were rusting when they were sitting around in the lab. So, whereas the Torch Lake cores did not exhibit any oxides of iron being formed and also didn't have a release. So that's an interesting area for future exploration or investigations is it true that there is such a low level of phosphorus release? And could phosphorus be released? There was, probably within the experimental error, there was some indication that there was actually oxic release. And that's also a question mark that would really warrant some further study.
Thank you. Thank you very much. Didn't want to put you on a spot, but you did a good job. Okay, if there's no more questions, Trish, would you like to come up, please, and give us a brief overview of the phytoplankton?
I'm going to go sit over here. I'm not comfortable. So, yeah, you're in trouble. All right. Can you hear me if I do this one, Mike? Does that one work?
You got to be a little closer, probably.
All right. Well, I was asked to present results on Torch Lake, for Torch Lake on phytoplankton in the absence of Dr. Ann St. John. So I'm very sad she's not here on top of many other reasons. But I'm hoping that with some positive thoughts and great medical care, that she will be here. She will, what? Here. Okay, I don't think it's working.
I don't think it's on.
Did you push the button?
Now? No. Now?
Sorry.
Huge.
Yeah.
There.
Test that.
Hello? All right, so I'm going to start again. I was asked to present results for Torch Lake on phytoplankton in the absence of Dr. Ann St. Amand. She's unable to join us today, but I hope she will be able to join us at this gathering next year because she will be able to provide a lot more information from her years of experience in studying phytoplankton. What I'm going to go over is a study that Doug alluded to, the 2020, what I call the TLPA ECT report. The TLPA also contracted with Dr. Saint Amand to analyze samples collected by ECT. Up on the screen, thank you, Dale, you'll see the map of Torch Lake, and you'll see that there were 3 areas that were sampled for phytoplankton. One is the green dot on the lower section in the middle. Thank you. That's what Doug was alluding to, is the Torch Lake South Basin CLMP site. Then you'll also look, you'll see some yellow dots, one down at the sandbar, and then one over on the west side, and one up on the, on the east side, and then one way up in the northwest. These are considered high-density sites, and that's just more than 3 houses in a 500-foot shoreline section. And then you'll also see some red dots, which are considered low-density sites. The one is probably up at the top, that TLL1 is probably Hayawenta, if you've been out there. So anyway, so those were the sample locations. There were only 3 sample dates or collection dates. One was in the late spring, one date was in late summer, and one date was in the fall. So if you don't mind going to the next slide, Dale, for me. The samples were mounted on slides and identified and counted to the genus level by Phycotech, which is Dr. Ann's laboratory. She used the imaging flow cytobot, which is the IFCB, and it was used to assess the phytoplankton assemblage. This IFCB is a semi-quantitative technology that processes samples quickly and at a lower cost. Unfortunately, the fall sample sprouted legs and was not analyzed for the deep basin. What I want you to look at is basically just 3 colors on this chart. And those are the dark blue at the top, which is cyanobacteria, which is non-harmful. The yellow section, which is the golden algae and diatoms. And then the green, the true green, is the green algae. And if we can go to the next slide. Thank you. This is the findings for the deep basin. The deep basin samples for spring and summer, they contained overall a low concentration of phytoplankton and demonstrated a seasonal change in the algal species. This slide is showing the May sample, which was considered the spring sample, and you can see the length of the golden algae and diatoms in the yellow. And if you go to the next slide, you can see here by the summer sample, we had a lot more cyanobacteria. Dominated by cyanobacteria and a small concentration of golden and green algae. The cyanobacteria present, as in the title of the coloration, do not present or contribute substantially to harmful algal blooms. If we go to the next sample, the nearshore samples contained extremely low concentrations of phytoplankton also. Cyanobacteria dominated the phytoplankton community regardless of the sample date or location. Again, the cyanobacteria was non-HAB species. Many of it was picoplankton, very small plankton. Here the chart will show you the yellow again is golden algae, and this lighter shade or true blue shade of blue is cyanobacteria. The gray is the diatoms, and this is from a high density nearshore site. And this is the only place where there was some interesting findings or significantly different findings, in that golden algae were significantly higher in concentration at the high density sites compared to the low density sites during the May sampling only. So if you go to the next slide, you can see this is the low density site sampling, and we do not have that golden algae there. So to summarize the results from this 2020, the algae assemblage in surface waters of Torch Lake were low in concentrations and were mostly comprised of cyanobacteria, approximately 1 to 15% being golden brown algae. However, despite cyanobacteria being present, the relative biomass of the algae present was extremely low and predominantly small, the Phytoplankton. And the question that would come up is, well, what about comparing this 2020 data to 2019 or 2018? Or what about comparing it to 5 years or 10 years ago? And we don't have anything to compare it to. I was able to go back to some old records in 1970. These were old student reports, and these were students of Dr. Laverne Curry, who collected phytoplankton samples and identified dominant species in their samples. And we could probably do a crude qualitative comparison of these samples from the 70s with the 2020s, because in addition to these types of feed readouts on the samples, Dr. Aman also analyzed the samples to the genus level. But basically, if I were to sum it up, Torch Lake, we have low concentration of phytoplankton, and we have a small amount of information about the low concentration of phytoplankton. So with that, I will stop, and we'll let that go, because I think Doug's handled that about the chlorophyll. No questions. No questions.
It's not a question, but in 2005, we submitted phytoplankton samples to Phycotech for analysis. So
I didn't see it written up. I did, if you go to the next slide, Doug, I did put you in here with your North and South Basin results.
yeah, I think those
chlorophyll A.
samples were collected subsequent to the work. You know, we were working in Clam and Bel Air, and there was increasing concern about cyanobacteria and, possible
We also
toxic.
had that bloom in Lake Bel Air.
Yeah,
right?
so that may have been from 2005 or 2006, but the data definitely exists. You may just not have it.
Okay, it's
so-
not on the Three Lakes website then. I didn't find it
We
in
could
any of the appendices.
certainly share it with you, because I've still got, I remember
Okay.
looking at those graphs. They're real pretty, so.
Okay. Okay, great.
We would very much appreciate if you in fact could share that with us. One of the things that I was going to ask all the presenters for permission is the packet that we developed prior to this meeting, we want to supplement with any additional information, including the presentations today, if we have the permission of all the presenters. Okay. As I said in the beginning, today is not a destination, today's a start point. So Jan Stevenson, Dr. Stevenson, are you ready?
Yeah.
Okay. So, Bob?
Could you real quick introduce our panel, so everybody knows who we are?
We were supposed to do that in the beginning. I'm sorry. Fred kind of, that's okay, Fred. Could have gave you a little warning. It's probably a good idea and probably get things relaxed a little bit. Would you mind, Teresa, if I started with you? Okay, and then we'll go through the panel and have everyone introduce themselves.
Good morning, everyone. I'm Teresa Sherwood. I work for the Michigan Department of Environment, Great Lakes and Energy, otherwise known as EGLE, in our Water Resources Division. I work out of our Gaylord office, primarily in our non-point source program, covering the 22 counties that our Cadillac and Gaylord district covers.
Thank you. Chris Horde, I'm with the U.S. Geological Survey in Lansing, Michigan. I'm the network operations chief there, overseeing our gauging and groundwater monitoring program.
Good morning. I'm Jeff Schlotto. I'm from the University of California at Davis. Thank you. Thank you. It's a good start. Anybody else? Department of Civil and Environmental Engineering. I also direct the Tahoe Environmental Research Center. So pleasure to be here, especially to you.
Hi, I'm Dale Robertson. I'm a research hydrologist with the USGS out of Madison, Wisconsin. And I guess I'm the one to blame for this meeting. They're asking the USGS to come in and see where we can help out. And I knew so little about the lake that I really wanted to bring all the scientists together to tell me specifically, but everybody else, to hear everything at the same time. And so instead of having to read a paper, having to do this, I really wanted the experts to come in and say what they knew. And from there, hopefully, we can start to look at some of the problems with problems in Torch Lake. So.
Well, I'm Doug Endicott. I work for Great Lakes Environmental Center down the road in Traverse City, and I am not responsible for this meeting. So.
Okay, we'll go to Amanda, and then we'll end up with Steve.
Hi, everyone. I'm Amanda Chambers. I also work for Eagle. I'm out of the Lansing office. I'm in the Lake Michigan unit of the Surface Water Assessment section. I work as an aquatic biologist, and part of my coverage area includes the Northwest Lower Peninsula watershed, so including Torch Lake. A lot of what I do is benthic macroinvertebrate sampling and water chemistry sampling to determine if water bodies are meeting water quality standards.
Hi, Aaron Parker. I'm also with the Michigan Department of Environment, Great Lakes and Energy. From talking to a lot of you, it sounds like a lot of you saw my presentation earlier with tip of the mitts, so maybe I don't need to introduce myself. But my focus is on harmful cyanobacteria blooms that have occurred throughout the state of Michigan. And so while I'm here, I'm going to be thinking about some of the oligotrophic lakes up here in northern Michigan that have been experiencing harmful cyanobacteria blooms. And then just one other add-on. So prior to coming up here, I spent 4.5 years working at Florida International University in an aquatic ecology lab. However, I worked very closely with Evelyn Geiser's paraphyton lab. So I was very interested to read Jan's report and hear about talking about the parallels between what's going on in Torch Lake and then Everglades paraphyton. So, yeah.
Hi, I'm Sherry Martin. I'm with the USGS down in the Lansing office. I'm in the groundwater and watershed modeling team. My training is as a limnologist. I've done a lot of work up on Higgins Lake. I'm really happy to be getting out on Torch Lake and seeing some of the parallels that are there.
Hi, everybody. My name is Caroline Keeson. I'm the Monitoring Programs Coordinator at Tip of the Mitt Watershed Council. We're a nonprofit that's been in this area since 1979. And we're focused on the tip of the mitt, as it says in our name. My role includes overseeing our volunteer monitoring programs for stream and lakes, and then all of our data collection activities in the field.
Hi, my name is Heather Smith. I work for the Watershed Center, Grand Traverse Bay. We're a nonprofit that advocates for clean water in Grand Traverse Bay. We haven't been very involved in the conversations around GBA, so I'm here today to learn and see what we can chime in on. So thanks for having me.
Wonderful, thank you. Thank you very much, and I apologize for not doing that sooner. That's okay. It's all yours. Introduce yourself.
All right.
Tell us all about you.
I'm Jan Stevenson. I'm from Michigan State University. Yeah, so I'm going to talk about work on golden brown algae studies, and this is the title that I was kind of assigned on Torch Lake and hypotheses for enhancing algal growth. And I want to emphasize you can get a lot of accumulation without fast growth rates if you don't have losses. So that's going to come into actually the idea about how do you model algae, and that's one of the things you think about is how fast do they grow and how fast you lose them. So it's possible we get this accumulation on the bottom with very, very slow growth rates. So I'm going to talk about that a bit. All right, so first though, I want to acknowledge all the help. And I can't take a lot of time. I've got 30 minutes to go through 6 years of studies. So Becky, I'm sorry she's not here. She's been my primary communication person over the last 6, 7 years, maybe 8, 9, who knows. And then Dean was the person that called me and had me come up and start talking with Rex and Pat and Trish and the group about what's going on here. And then I know Trish and Rick and Fred have been out helping a whole lot with sampling. And then Art Hoadley has produced these amazing informative aerial photographs that are are great. So Three Lakes has funded some of this, and then Tim Bravika did all the chemistries, almost all the chemistries. Rex Lau and Pakaselic at U of M were involved with this, and then both of them really good friends, algal ecologists that were involved in the first year, published a paper on the unique diatom flora of Torch Lake. Sheng Pan Lin helped me out in the first year, as did Sherry. Sherry Martin helped with some of the water chemistries during the first year. So with that, acknowledgements, lots of help, and I'm just kind of bringing together stuff from over that time. So the problem is we have gone from a nice clear sandy bottom to one with, this is the last time I think I'll use this phrase, these beautiful patterns of golden brown algae on the bottom. I'm an algal ecologist, I completely understand the issue, and I wish I could tell all the anecdotal stories. So this is a crust of a lot of diatoms and cyanobacteria that are on the bottom of the lake. They're deep as well as shallow, and they occur around the lake, not just Torch Lake, but many others. So over the years, we've done lots of stuff, and I'll talk about that kind of in a chronological order. Several reports have come out, public presentations, YouTube videos, scientific presentations. So this is out there. I've been talking to people about it a lot. And again, I'm trying to move on so I can get to what I was going to talk about, which is kind of chronologically go through and discuss how we developed the hypotheses, tested the hypotheses, or evaluated is a much better term, because none of them are off the table. Evaluated them, prioritized, tried to figure out what we're going to do next. And then I want to spend some time to clarify this low phosphorus hypothesis, which I've just written up in the newsletter, or an article for the newsletter, and emphasize its plausibility. Not that I think it's the best hypothesis, but to not have it on the table is a mistake. So with that, the first thing we did is we looked at the algae on the bottom of the lake and tried to come up with ideas on what's causing it. Most of it's diatoms where it was sampled. And it seems like the algae, there's a huge density of algae in clean sand that are on the sand grains and in the interstitial spaces. And what's different here is now the algae are on top of the sand. That's the difference. In Lake Michigan, any place you go and look at algae, look for algae in sands, there's millions of cells per centimeter squared. Now they're on top of the sand for some reason. All right, so we started generating this idea on how this system worked. Oops, probably should keep this close to my mouth. So what's causing this GBA, kind of working back up a causal model? It's probably related to water chemistry, either what's going on in the surface water or the groundwater, or particularly that interface, that sand surface water interface where the algae are growing. It could be related to chemistry. It could be related to other things like herbivores and bioturbation, bioorganisms moving around and knocking it off. And then those things could be related to both natural factors and human factors, a whole diversity of them. Most of what we've done is on this area because we've not invested in relating the natural factors and the human factors to chemistries and to the biota. So with that, we generated a list of hypotheses, and I'll go through those as I go through the evaluation of each of them to save some time. And this picture is one of the great pictures of what the crust of GBA looks like. These are the crusts that we find at the bottom of deeper parts of the lake too, where I've sampled and scraped them off the bottom and looked at them, but also in some shallow water habitats, but not always. Sometimes it's loose and flocky, sometimes it's a little gray, but these are the crusts that are really causing most of the golden on the bottom of the lake. One of the key questions that we've had from the beginning is, what's changed that could cause this? So I'm going to get to that, or get to some of those ideas. In all of this hypothesis evaluation, we're kind of using these frameworks of causal analysis. You want to make sure that, for example, you want to make sure that the cause preceded the effect. You want to make sure there's a relationship between GBA on the bottom and any environmental factor that you see either changing through time or changing spatially, which was what we had hoped we'd find, spatial relationships between high and low density, high and low groundwater, high and low surface water, closeness to a stream, looking for that relationship. And we want experimental evidence to prove cause-effect relationships. So we're, plus a host of other things. So we're keeping that in mind as we go. So first, we're going to knock off the idea that the changing light conditions cause this, because if I can see the bottom, the algae have enough light. Algae are really good at harvesting light and growing slowly. So I just didn't think the changing in Secchi depth and anything like that was a significant factor. We're not in a range of light conditions that affect the benthic algal growth. We have no evidence that we've lost grazers or bioturbators, that things that disturb the bottom that would affect it. That means we have no evidence. That doesn't mean it didn't happen. We just don't have that evidence. In addition, there aren't many examples of invasive algal species. Now, people talk about Didymo being invasive, and if you know Didymo, there's a lot of question about whether that's true or not. So, I don't think that's going to cause GBA. So the next question, the next thing was to address the hypothesis of whether algae in the news, whether people were just seeing this problem because algae and biofuels, algae and harmful algal blooms, all those kinds of. I was questioning this possibility that people were just excited about algae, say, oh, we got algae on the bottom of the lake. Well, these photographs that Art took really show that between 2010 and 2020, the algae developed. And actually between 2010 and 2015, there are good, Art's got pictures of 2015 that show it. So we got involved around 2014. So that pushes the date of when GBA first really probably happened around 2012, 2013, that it started to develop. And then we can use Art's photographs actually to kind of document, did it pop all of a sudden and then just stay the same, or has it changed over time? So I'm trying to move along so I can get done in 30 minutes. So excuse the flip. So there's stories. I'm telling the story. So we just knocked off that hypothesis. Next one, climate change in a warming lake. I've heard about the lake getting warmer. It has to precede, remember, cause needs to precede the effect. So we use satellite imagery, Shang-Pan Lin, for reasons we obviously, did some great remote sensing stuff. He was a graduate student working in my lab that had that ability. He did it for fun. That was Shang-Pan. So Shang-Pan did this assessment and looking at near shore, deep, and the whole lake basin itself, or the whole lake itself, because he was able to do that. Nice annual variability from about 2000 to 2015. No increase in temperature. So that's probably not it. And it's probably not in the range that's going to affect the algae either. So that leaves us with these big three. Zebra mussels, enriching the sediments. So I want to distinguish that from reducing phosphorus in the water column. Runoff from big storms, enriching the surface water, which would probably cause plankton blooms before it would cause benthic algal blooms. And then groundwater contamination. And we are particularly concerned about groundwater contamination and the benthic algae being the canary in the coal mine. We know that groundwater contamination is possible. Groundwater flows through both this higher through flow kind of groundwater, maybe more from riparian sources, and then the deep groundwater moving into the lake. They're pushed by different things into the lake, and they're susceptible to different kinds of contamination. So we wanted to really focus on that early. Just last year, a paper was published in Bioscience, kind of indicating the novelty of people looking at this in lakes. We've looked at this for years in streams, the importance of groundwater coming out, enriching the surface water and algal blooms being associated with that groundwater discharge into the surface waters. So as part of this, piezometers were stuck down into and pounded down into the rocks and sand. Samples were taken. We also used this, and so we sampled from probably 1 to 2 feet down into the lake floor groundwater. And then we also used these kind of surface water peepers made out of dialysis tubing so we could figure out what was going on right at that surface of the sand-water interface. And what we found is basically phosphorus is higher in the groundwater than the surface water, much higher. But also right at the sand-water interface, there's an enrichment of there is an enrichment. It's not higher than the groundwater, which is what I would expect if it was zebra mussels enriching the surface with pseudofeces. We don't see that a pop like it's higher at the sediment water interface than in the groundwater. So that groundwater information was really important. The groundwater concentrations that we measured from 2015 to 2020 are indistinguishable from 2016. Or, I'm sorry, 2006. The study that Gluck was involved with and written up in a report, they're indistinguishable. So we have no change in groundwater that we can measure. So we have no cause preceding the effect. In addition, when we look around the lake at areas that we'd expect high and low groundwater concentrations or high and low surface water inputs, we don't see a pattern. So part of this was measured with a GoPro camera while I was sampling the bottom and just looking at how much algae is there, because I've done this for 40 years. I'm pretty good at telling algae from dirt, and we do this all the time, doing what we call a visual assessment, like a Secchi depth, but we do it based on color and texture. And almost every place on the bottom, we were above what I would call, there's GBA present. There's GBA there. But there are some places that it's really high, and one of those places is near the Clam River, where the Clam River comes in. But there are other places that are really high too. I mean, these are really high levels of GBA. Hoadley's pictures also show the same thing. Just GBA is just everywhere. It's not just close to the streams. It's not close to where you'd expect groundwater sources. There are a couple places, curiously, north of Tahoe Point, and also near some hills over on the west side of the lake. The GBA tends to be a little bit lower consistently. So when I go back and do some work on low GBA sites, I'm going to go to that point, that spot just north of Tahoe Point. So zebra mussels we kind of crossed off for enriching the sediments because we don't see a pulse of nutrients there. Really, the zebra mussel densities in most of the lake they're non-existent. The zebra mussels don't colonize sand, and there are really no records of high zebra mussel densities that I can find that are enough to filter out a whole lake and put the feces on the bottom and distribute those across the bottom. Now, maybe they help clear the lake. That's a different issue, and reduce phosphorus. Runoff from big storms. We don't have phytoplankton blooms, which is what we'd expect in those areas. Most of the streams don't have that high of nutrients. Clam River is not even that much higher. It's just a little bit higher, and probably not. And groundwater contamination, we're just not finding a shot at groundwater contamination. We're not finding the evidence that groundwater contamination is doing it. That doesn't mean that groundwater is not affecting these patterns in benthic algae, but we're not finding a human source of groundwater that's enriching the groundwater, especially at the spatial extent around the lake where we're finding GBA. We're not finding a groundwater nutrient concentration GBA relationship. So that leaves us, so again, to emphasize, I think these patterns, these lobes of GBA are related to groundwater. I'm not convinced that it's related to changes in phosphorus concentration in the groundwater caused by humans. It's more like faster and slower fluxes of groundwater that might be natural. So that leaves us with this question, what's changed? And one of the things that's changed is phosphorus has decreased in the water. We've seen it here in a lot of different data. Doug would argue that it hasn't changed. That's something to debate. If we look at tip of the watershed and CLMP data, over the years, 15 out of 18 lakes, most of them the big lakes, have seen a decrease in phosphorus concentration. If we look at historic data, this doesn't just go back 20 to 30 years. Historic data based on diatom species composition and paleoecology in the bottom of the lakes indicates this goes back to before the 1950s that we've seen a decrease in phosphorus in the lakes. So is it possible that a decrease in phosphorus could be causing an accumulation of algae in the bottom of the lakes? There are two places where this is really dramatic. One is the Everglades, where these floating calcareous algal mats, very much like those that we find here, except they're not floating usually, same species even, this Encyanema everglanianum, occur only in low phosphorus. And the same thing with Didymosphenia. Didymosphenia, which is now blooming more in the streams of Michigan, also only occurs in extremely low phosphorus. So just to give you an idea of what goes on in the Everglades, where I used to work a lot in the 1990s, and I'm actually now still working with the National Park Service, in fact, writing a paper on that right now. In the Everglades, what happened is this floating calcareous algal mat disappeared. First, we weren't sure. Is that possible? They thought phosphorus is causing the loss of this floating calcareous algal mat. And what we found when traveling along this gradient of phosphorus from a point source down south is that along this gradient, you would not see calcareous algae close to the point source, these canal waters, the canal waters that go into the marshes. And then all of a sudden you get to a certain distance, and then you almost always see high abundances of floating calcareous algae in these wetlands. So we figured this out by actually traveling back and forth in an airboat. I did this work with Kurt Richardson from Duke University. There's a really fun story about Bob Johnson, who used to be on Mutual of Omaha's Wild Kingdom. He was the guy in the alligator suit because Jim couldn't swim. So many of you probably can relate to that, that story. And he was the guy that kept us safe out in the Everglades. Amazing guy. He also built these mesocosm systems where they experimentally manipulated phosphorus concentrations. And if you did not add phosphorus at really low doses in the Everglades, you get calcareous algal mats. If you add phosphorus, you lose them. You lose the algae. And along this gradient. So we checked that out with the helicopter, flew this gradient. We lose from the natural condition as you go up phosphorus concentrations, we lose the mats at 7.76 kilometers at about 10 micrograms per liter. That is the number that the state of Florida established to protect the Everglades based on that data. The mats that we see from the deeper water, from the 10-foot area, are very much like the mats that we see in the Everglades, just kind of turned upside down for reasons that we could go into, but not enough time. Key thing, Encyanema everglanianum is there. Encyanema everglanianum is this yellow bar that increases in abundance during the summer. And Cyanema everglanianum is this cute little thing forming rafts in the samples. And Cyanema everglanianum is the thing that is most abundant in control conditions in these experiments that I ran versus when I enriched the experiment. So experimentally, it's responding. It's actually more abundant in low phosphorus than high. So the possibility that decreasing phosphorus is causing this problem is biologically plausible. There are analogous situations, and there are tipping points. Kind of like phosphorus decreasing. 2012, it hit that tipping point, and now you've got golden brown algae, like on the phosphorus grade in the Everglades. The next question is, what's causing the phosphorus decrease? Zebra mussels, we'll cover the last 20 to 30 years. Atmospheric deposition of nitrogen actually goes back to the 1950s. So it goes back way back. The sources are there for a long time. Agriculture, automobiles. The same kind of thing has been studied extensively in Scandinavia to the point that they think that lakes were naturally nitrogen limited in general, and that nitrogen deposition has shifted lakes to across Scandinavia from being nitrogen limited to phosphorus limited. There's a body of literature back around the early 2000s that kind of discusses this. So as I'm going down this rabbit hole of, so how this possibly work, these are interesting things to follow along. So the potential cause of the phosphorus decrease is plankton filtration by zebra mussels and atmospheric deposition. So we've got a bunch of hypotheses. We've tested them. When we came up, I mean, the writing was kind of on the wall. We're not seeing these strong relationships between, if you go out and survey the lake, you just don't see any relationship with the patchiness of GBA and where you think you would see it and where you wouldn't, except for Halewinton North. So we started thinking about these alternatives, but we're still thinking about them. I mean, we need more information, and that's what, you know, that's what today's all about. That's what sampling more and actually sampling more places than just Torch is really important to understand where Torch fits into the situation of Michigan lakes, of lakes across the country, of lakes around the world, and test these hypotheses. And a big part of it is that broader assessment, linking the groundwater surface water chemistry and to natural and human sources, and understanding how have things changed and whether or not phosphorus has actually changed in the lakes. Okay. I apologize. I did not know about the questions online, so you guys can ask me that at a coffee break, at beer, or sometime.
Okay, we actually have a 5-minute break at this point. And I want to offer that to all of you, take advantage of it. But I also want to take at this moment, after that terrific presentation, is anyone have questions for Dr. Stevenson? Please, Dale.
Hey, Jan, I got one question for you. Are we looking at totally the wrong thing? And I'm going to come up with a hypothesis at the end. And it's something you said early on. And what you said is that lakes have this golden brown algae commonly mixed in with the sand. And that's really common in most lakes. So is it that the golden brown algae is wrong? Or is it this gray matter that's underneath it is really the question we should be looking at, and what caused the gray matter? Because if the golden brown algae is in most of these lakes, it's just that we're seeing it now. It used to be hidden in the sand. Is it the gray matter that we should be discussing? But this is leading into my hypothesis that I'll present a little bit later. But I'm just wondering if we're looking at the wrong thing.
Please.
Okay. That's possible. So the gray matter and it depends which gray matter you're talking about. But I assume you're talking about the calcium carbonate in the mats. So the calcium carbonate in the mats is, of course, caused by the photosynthesis and alkalization of that mat matrix, and that's what forms the mat in the Everglades. Is this the photosynthesis and the precipitation of calcium carbonate, and that armors that mat. So it's really, winds don't, hurricanes, well, maybe hurricanes do, but winds and these big thunderstorms don't break up this floating mat on the surface. So I think that's, I don't know how that fits into what specifically you're going to get into, but the cycling of phosphorus within these mats maintains this biomass potentially at really high levels. We were working in Weeki Wachee Springs, and we had 6 foot deep mats of cyanobacteria, and we went inside them, and they were just bubbles. They were like these organisms gathering phosphorus and just growing into these massively large organisms. And there was more phosphorus inside these things because they were consuming it. It was like a gut kind of thing. So the same kind of thing might be happening in these calcium, would fit the model of calcium deposition, helping hold the mats together, and then building this thicker matrix in which cycling goes on. That would be the Everglades model.
Dr. Stevenson, two of the things that we've seen in the lake, one are columns of algae this big around, this high, growing up from the bottom.
Yep.
The other thing we've seen are like sheets of algae that are maybe yay high, again, growing up from the bottom, like ribbons. Are those two phenomena the same as golden brown algae, or are those different types of algae communities? Or have you studied those?
Yeah, so
Here, use your mic.
if I haven't studied them here, I've probably seen them other places. And so I think what you're talking about, I've looked at samples of what I think you're referring to as the columns. And there are a number of algae, diatoms, that produce extensive amounts of mucilage and can bubble up and form these kind of columns. They're typically a genus that does this. We get a lot of floating flock in our Lake Michigan shore that people get all upset about, and my legs get white with this mucilage drying out on my legs from these cymbellas mostly, diatoms that produce that. This happens at really low nutrient concentrations. In streams, this is really abundant. And again, they're producing these mucilages that help them harvest nutrients and grow and persist. Not necessarily grow fast. They just need to persist. The success in evolution is not necessarily growing fast. It's persisting. So as Dean would say, the diatoms have been around a lot longer, right? So the ribbons, I'm not sure quite if those are benthic mats that are lifting up. And
They're vertical.
oh, vertical ribbons. Yeah, I'd have to seem to know more, but those are certainly, those are growth forms. They're, a key point that what I was trying to show with some of the slides is there's a lot of different kinds of algae. So I was really interested in your point that it wasn't there before, which is it's an important observation. If indeed it wasn't, but yeah.
Jan, in your talk, you talked about low phosphorus that could change the species composition to more low phosphorus tolerant diatoms. And is it true that some of those are also developing these mucilages or branches or whatever to help harvest whatever phosphorus is out there, and that those branches or those mucilages would make them more visually appearing on the bottom? Is that a true statement, or do we have any evidence for that?
Yeah, so the idea so thanks for the question, Rick, and thanks for having an opportunity to talk to you all. There wasn't time in 30 minutes to say thank you. And Rick brings up a really important point that I didn't bring up. about algal ecology. And typically the low nutrient species their growth is limited by nitrogen and phosphorus to build tissues, to build membranes and DNA, the nucleic acids and things like that require nitrogen and phosphorus, the proteins and cell components. But they can photosynthesize like crazy, flipping that over and then extrude the excess photosynthate because they can't reproduce cells. They extrude that excess photosynthate as mucilages. And those mucilages are helpful for holding the mats together and actually entraining exoenzymes that they secrete to help digest more stuff. So they secrete phosphatases that help them cleave phosphorus off of organic molecules that, you know, they're making they're basically making their own garden down there and slowly accumulating, slowly harvesting more water maybe, or more surface water or plankton that settles down into them, and they eat them, you know, indirectly by helping out the bacteria to help them process all of that. So these are the kind of fine scale mat dynamics that explain how you can get large accumulations in really low phosphorus conditions.
Dale, way over here in block by view. So this is a quick, easy one for you, I think. But you mentioned, you know, what's changed is phosphorus has decreases, and you gave two reasons for that decrease. One was filtration, but then you said deposition. And so what exactly are you saying about deposition that's leading to a phosphorus decrease? Is it the nitrogen deposition that's gone up?
Yeah, so the idea is that nitrogen deposition shifts the ecology of the lakes, and there's a negative relationship between phosphorus concentrations and nitrogen deposition in some studies, and they had I mean, a lot of this is still up in the air on how that could occur, but imagine that you just shift the species assemblage to organisms that now utilize nitrogen, require higher nitrogen concentrations, but they can use phosphorus to lower levels. So they're taking up phosphorus now to lower levels than could be taken up before, and they're settling out of the water column, and they're just harvesting phosphorus now to lower levels than they were able to harvest before. Oh, no, yeah, no, I don't think any of these things are proven. Or, well, some of them are proven in certain places, but then applying that information here is a different issue.
Okay, I'm going to try to get us back on time track. So please take advantage of the paper and pens in front of you if you have further questions and write them down. We do have a Q&A near the end of the agenda, but I want to get us started back on track. The next item on the agenda is our roundtable discussion, and we're going to ask Dale Robertson to head that up. And this is intended to be our interactive session with the panelists, and hopefully it will lead to more additional information that may not come out in the formal presentations. That's our intent, at least. So Dale, if you would, please.
Okay, I got my own agendas here. Anyhow, what I really looked at this part was to try to put Torch Lake into a bigger context. And what I'd really like to see is, are the trends that we're seeing in Torch Lake the same thing we're seeing other places? And so what I thought I would do is to try to get some of the people who are doing bigger regional studies to try to look at the area and say, how does Torch Lake fit into that? So each one of the people that represent the regional areas have a different area of expertise. And some of this is going to be, well, I don't want to put words in anybody's mouth, but I just wanted to see, how does Torch Lake, do we see this golden brown algae? Do we see this gray matter? Do we see some of these things occurring in more lakes than just Torch Lake? And so what I would like to go around and the one thing that hasn't been talked about are some of the other changes, like in the fishery. There are some changes. Jan was sort of mentioning it, but I think there are some changes that could have changed the grazers, like gobies, changes in the fishery. So different people have that kind of expertise. But as I go around, I'd like to try to get each of you to describe what you think is the water quality of Torch Lake with respect to other ones, and also the trends that you're seeing in the other lakes Does Torch Lake fit into that? I think Torch Lake is involved with most of the monitoring programs. And so hopefully you can sort of describe, do you see trends? Is Torch Lake fit into that, or is it an outlier? Okay. So with that, if this is totally new to what you're expecting to say, that's fine. You can describe some of the things more like Tom was saying. But try to put Torch Lake into a regional perspective. So I'll just go around. Carolyn, would you like to introduce yourself a little bit again, and then sort of try to describe what you know about Torch Lake and put it into a regional perspective?
Sure. Again, my name is Caroline Keeson. I work for Tip of the Mitt Watershed Council. So we monitor about 60 lakes every spring, and we've done that, or not every spring, every 3 years. And I think we've done that since, it's probably been about 25 years. So we have a lot of consistent data on lakes, and it's well spread around the Tip of the Mitt region. And I would say that we definitely see the total phosphorus decrease in many of the larger lakes, similar to what Jan was presenting. So that's, Torch Lake definitely falls in line with a lot of those other lakes. I can say that we did a vegetation survey on Torch Lake last year. We haven't talked too much about vegetation, but I'd have to look at the exact numbers, but I think of, let's say, 20 other lakes in the region, I think Torch Lake had some of the fewest number of species were found, and just one of the lowest, what would you call it? The lowest acreage of the lake was covered with plants compared to other lakes in the region. And then one thing that I don't know right offhand is we have done quite a few shoreline surveys on lakes in the area. So we look for impacts happening right at the shoreline that might impact water quality. And so we look at things like, is there turf grass there? Are there native plants? Are there alterations, seawall, riprap? We try to relate that to the water quality. So I don't know how Torch Lake compares to other lakes in the area, but I can definitely get that information to this group, because I'm sure we could easily compare the lake to probably 20 or more other surveys that we've done. That's it.
Oh, thank you. Next, Aaron. Aaron, I actually have heard your talk, but can you zoom it into a little bit into more the area around here instead of comparing Torch Lake with way down in southern Michigan?
Was skipping you?
Oh, she's actually not on this group, so sorry.
Okay. Yeah. So basically at Eagle, we started really keeping track of cyanobacteria blooms throughout the state in earnest starting in 2016. So I just wrote kind of a back of the envelope list of lakes up here at the tip of the mitt that have had confirmed cyanobacteria blooms since 2016. So like Beals Lake, Antrim County, Black Lake on the Sheboygan Presqu'île County border. They've had them for the last 3 years now. Crooked Lake in Emmett County last year. Intermediate Lake here in Antrim. Lake Charlevoix last year. Paradise Lake, Emmett County, and 6 Mile Lake on the Antrim Charlevoix County border. And so all of these lakes, I think all of them, maybe except one, were part of a large study that USGS did in the early to mid 2000s. It was called the Lake Water Quality Assessment, large study where basically every publicly accessible lake in the state was assessed for a myriad of different parameters. But then trophic status indices, either oligotrophic, mesotrophic, eutrophic, were assigned to these different lakes. And so I just went through these lakes here and looked at what they were assigned back then in the early 2000s. And so Black Lake was assigned as being mesotrophic back in 2005. When I went to Black Lake in 2019 and took the trophic parameters, it shook out as being oligotrophic. All the other lakes, so like Crooked was mesotrophic, Intermediate was mesotrophic, Charlevoix was oligotrophic back then, 6 Mile was mesotrophic. But then going back and looking at CLMP data and Tip of the Mitt's data, just looking at phosphorus, it looks like oligotrophic. So I would say that yes, I agree with the hypothesis that there has been a region-wide decrease in phosphorus. Yeah, and then so we're also getting blooms in the Upper Peninsula too. Last year, Isle Royale had 3 lakes with cyanobacteria blooms. And I don't know if maybe these were in areas with similar geology that we have here. But I was wondering, while I have the mic, if I could pick Jan's brain on a couple. Okay, so sorry, Jan, put you on the spot. It looked like there was some paleolimnological data that was done for 100 years looking at diatoms. Was torch one of those?
No, but Bel Air was.
Okay.
And so Sherry Fritz and John Kingston and I think Engstrom, Bel Air was one. They only did cores, I think, on 4. A couple of them were local, but they did surface water sediments to develop a calibration set, if you're familiar with that concept, for the diatoms to infer phosphorus.
Mm-hmm.
And Sherry, when Sherry knew I was working on this project, she sent me all those samples. So, we've got all that to work with as part of the project.
Okay, because I was just wondering, so could some paleo work be done to look for ensynonema to see if that, yeah.
Yeah, so Doug has given me the sediment trap data, or the sediment trap samples from around the year. And I'm looking at those to see if we see benthic diatoms as well as phytoplankton in them. And we are seeing at certain times of the year benthic diatoms in them. So that indicates that we've got the potential for doing cores. It's one of the 3 things that I've recommended. And if I can get my short core fixed, I'm going to take one this summer. I think that would be really valuable to do.
Okay, and then my next question. So I remember from working in the Everglades with the paraphyte, and there seemed to be the floating mat paraphyton, but then also the benthic mat paraphyton. And it seemed like when there was phosphorus enrichment, the floating mats disappeared, but then the benthics tended to stick around longer. So was the ensynonema, would that be in the floating mats or in the benthic?
So I didn't study the benthic mat.
Okay.
We only worked with the epiphytes that came up to form the calcareous floating mat.
Uh-huh.
We do have we have got cores from the Everglades, and it shows the ensynonema increasing and shifting with. But I don't think the ensynonema is going to be in the higher nutrient concentrations. In fact, I'm positive, because right now I'm working with Big Cypress, and I just analyzed the data Saturday, and Ensynonema evergladianum was the lowest phosphorus indicator. Now, other data doesn't show it being quite that low, but on that gradient and in my data, that's what it showed. So Ensynonema evergladianum is restricted to pretty low phosphorus concentrations. There's some.
Okay.
Not to the seminar.
All right.
Sorry, Dale.
That's okay, let's move on. Heather, can I jump back to you? Sorry, I skipped you to begin with.
Hi. Well, the Watershed Center doesn't do any lake monitoring or coordinate any sort of monitoring. And Sarah Uren probably could speak. Sarah Uren from our office could probably speak a little bit more about conditions of torch. She helped with the Urkel Watershed Protection Plan most recently. But I guess I'd like to echo what Caroline said. We know that phosphorus is decreasing in a lot of our lakes. We know that torch is deep. It has not a lot of macrophytes compared to some of our other inland lakes. Thinking about other lakes in the Grand Traverse Bay watershed, we have several small lakes in the Boardman subwatershed. That's probably the Urkel subwatershed and the Boardman subwatershed, where we have most of our smaller lakes in the greater Grand Traverse Bay watershed. Most of those lakes are much smaller than Torch. They're shallower. They have more macrophytes. I don't know of any GBA issues in any of those lakes. Those lakes are like Arbutus, Spider, Indian. There are a number of different lakes in that area. I guess that's what I have to add. Thank you.
Amanda, do you want to introduce yourself and go on with that?
I'm Amanda Chambers. I am an aquatic biologist at Eagle. I'm going to kind of, I guess, defer this a little bit because much of what I do is focused on rivers and streams. We actually have a staff person that does more lentic lake work. And I'm not totally sure how she distributes her sampling sites at this point. But I think a lot of the work that she's doing is primarily associated with like assessing trophic state and taking nutrient samples and things like that. So that's part of some of the lake work. I don't know if you want to add anything to that. I mean, I'm in the area, but a lot of what I do is more lodic in nature.
Yeah, well, so this year just so happens to be the next cycle for the National Lakes Assessment that's going to be occurring. So there's 50 lakes in Michigan that are going to be part of that. I think Doug presented a map showing the 1,000 lakes throughout the country. So Michigan's going to have 50 there. And then there's another study being done by the Forest Service where there's 250 lakes throughout northern Minnesota, northern Wisconsin, and northern Michigan, so Upper Peninsula, and up here. That's also going to be done as well. So we should get some good data from that, including nutrient data to
Aaron,
add
do
to.
you think we have any chance at changing their protocols? In that Torch Lake is so low in chlorophyll, so low in phosphorus. If we follow the protocols that the national group does, all we get is less thans.
Yep.
Can we
Yeah.
get them to drop the things, filter 10 times as much water to get the chlorophyll numbers down? Send it, either have the EPA start thinking about a lab. Jan and I were involved a lot with the EPA. But finding a lab that you can send these, you know your phosphorus concentrations are going to be really low.
Mhm.
You send it to a lab, all you're going to do is get less than 5 or less than 10 maybe. That's fine for them to say that Torch Lake is an outlier. It's an ultra oligotrophic lake, but it doesn't help the lake.
Yeah. I highly doubt it because I think the thing with the NLA is they're applying the same standards nationwide. And so I think EPA would be super resistant to.
Well, I
making-
might have a little pull. Let's see if I can
Yeah.
do something. But I think that they're making a huge mistake by sticking with the same
Yeah.
approach. I understand it for a lot of things, but to go down to lower levels, to filter more water, I think that they've got to at least listen.
Yeah, well, so that does remind me of something I was thinking about. Right? So in the Everglades, I mean, the way they look at nutrients there is they collect periphyton samples and then they look at the nutrients within the periphyton because it's the same thing. If you were just to collect water samples out in the Everglades, everything would just be less than detection. So I mean, basically the only way you can look at nutrients in the Everglades is by grabbing the periphyton and then looking at it within there. So I was wondering if, maybe that was a possibility, going around the lake and just grabbing GBA samples and then seeing if maybe there's a spatial pattern there. So something to think about.
Okay, thank you.
Can I actually add
Sure.
one thing?
Actually, I was going to come back to you and
Oh, were you?
not let you off the hook. Since you're talking about streams, can you say anything about the streams around Torch Lake and compare that to other areas? I don't know if you do any phosphorus concentrations in the streams.
Most of
or-
it's benthic macroinvertebrate, and we're
Okay.
seeing really good benthic community structure in general in the area. But the thing I was going to add is that we have some folks in our department doing Didymo monitoring, and they're sort of pairing up with Dr. Ashley Murky at Lake Superior State University, who's doing more of the research side of things. And she actually presented a webinar very recently, and I'm just seeing a lot of parallels, and I don't know if that's something we might want to think about, just chatting it up with her and just kind of, it just seemed some of the similarities were striking, so.
No, I think that's good. We'll have to get Jan to move out of the lake into the stream.
Been there.
Teresa?
Been there, done that.
do you want to introduce yourself and add anything?
Sure. Again, I'm Teresa Sherwood. I work out at Eagle's Gaylord office. You know, I'm not directly involved in water quality monitoring, and I'm kind of new to this position within Eagle, but I just have a few thoughts to share. And first of all, I will say that I actually grew up in Rapid City, which is the town that borders the south end of Torch Lake. So as a kid, I took my swimming lessons in Torch Lake and have seen, like many of you, the changes over the years. So although all water bodies in my coverage area are important to me, Torch Lake is, you know, I'm very familiar with it. So, you know, overall, I would say, you know, to me, a decrease in phosphorus causing problems is very strange. You know, you tend to think that decreasing nutrients in water bodies is a good thing. So I don't know about the rest of you, but it seems a little bit odd. But I know in a lot of the meetings I've been in with Aaron and Caroline and others that, you know, we are seeing that. Overall, our district has been responding to a lot more algal bloom complaints. So that's just a general trend in our area. You know, I'm not sure necessarily about golden brown algae. A lot of it's the harmful algal blooms that we're responding to. You know, one of the potential causes for the decreased phosphorus is potentially atmospheric deposition of nitrogen, and atmospheric depositions that cause water quality issues are very hard to address. So I'm interested in learning and talking a little bit more about that. We see that being a cause of toxins in fish and causing fish consumption advisories is from atmospheric deposition sources. So that's just one thing I want to kind of learn a little bit more about and talk a little bit more about. But overall, you know, we are right now, you know, I work in our non-point source program, so we work to help identify and address non-point source pollution issues that are related to water quality and affecting our water bodies. I am currently reviewing the Elk River Chain of Lakes Watershed Management Plan, and we hope to, you know, get through that and approve it soon. And, you know, I think that as we work through this issue, if there are some non-point source pollution sources or causes that are contributing to the golden brown algae accumulation or growth, we can certainly look at addressing that through watershed management plans and funding that we can offer through that program. So that's really all I have.
All right, thanks. Yeah, as we go further, I think we'll probably be contacting a few of you. The one thing that hasn't been brought up in a lot of this is, you know, we're talking about sources of pollution. Septic systems haven't come in, and that could be bringing up some of those numbers a little bit higher than what the natural groundwater is, but that's something that we'll have to discuss. I think what I'll do right now is pass this on to Tom, and we'll get into a little bit more of this ranking.
So actually, we're right on target. We're going to take a little lunch break, but there's no free lunches with us. Sorry. Okay. Lunch break means you can get your sandwich or your drink or whatever. However, during the lunch break time, we are going to be having some discussions and some interactions on the survey results that we've asked you all to participate in. And Dan, you're going to take the team through this.
We're going to get you off to lunch very, very quickly. But like Tom said, we're going to make you, especially some of you work. Yeah, if you could help me flip that, Tom. I think Peg has put some results underneath here. So I know you can't capture this on screen, so I'm going to tell you what it is. And I'm going to tell you what we're going to do with it. And I'm going to tell you what work we need from all of you, and most definitely some of you as well, and I'll be brief with all that. So this was the survey results, and not everybody took the survey, and that's fine. It's meant to create a conversation, because whether you want that conversation to happen or not, it's happening hugely outside of the room, if not inside the room. We have a GEM meeting tomorrow. And the turnout at that, this is a Torch Lake Protection Association meeting. I'm told, being new to TLPA, that the GEM attendance is larger. I think Norm is not quite here. Bob could maybe attest to that. But the attendance at that meeting is larger than it's ever been. And the subject is, the subject of this meeting. And we're going to take the results from that meeting because we owe our membership an opinion. They're paying dues. They need to know where we stand, whether you want to stand somewhere or not. They need to know, just like shareholders in a company want to know whether they're going to get a return on their investment. So it's that sterile, it's that crisp, it's not debatable. If you want to debate it, you can debate it on your own. Nonetheless, this is the ranking that people had. It's kind of where people stand, and these numbers are hard to read, so you can look at them later. I'll just verbalize it for you for the moment. We broke it up into the work group, meaning the folks that have been rightfully doing a lot of the interaction and talk and speeches or talks as well, and then the folks outside of the work group. And so that's everybody else. And we sorted this, and what kind of emerged out of what people read, what they've thought, what they've divined from other conversations, and rightfully had their own opinion on. For students even, I know many of you are here. When I was once a student, I was told to go read things and develop your own opinion on it. And I think that still works, especially in the sciences, which you all aspire to. So nonetheless, what emerged as kind of in the top group was non-native mussels, groundwater, decreasing phosphorus, and climate change was kind of in there amongst the work group. And then, as we went to the local advisors, you started to see grazers. And then there were a number of folks in the other category. We don't have time for that now. You can go to the survey yourself and read what people by name put in and some of their comments. It's excellent, just too rich for the meeting. So last thing is what I want you to do, and through the miracles of Peg, what she did was to create some charts, and you can take markers. You have 3 votes. It's each one of those same hypotheses, which were also addressed by Jan in his talk, are split up into work group and local advisors. We'd greatly appreciate, as input to future conversations, what your thoughts are now that you've heard some more material. We're not going to make this any more scientific than, we're not going to delta between the 2 data sets. We're not going to try to do statistics on it. We just want to kind of know what you're hearing. So go ahead, Rick. I'm sorry. Yep. Oops.
And is the latest hypothesis low phosphorus on that list now?
I think phosphorus has always been on there, decreasing phosphorus.
It's on that list?
Yes. And if you feel, if you in particular, or anybody else feels that maybe we need to rethink things. And by the way, we're going to come back and talk about this at lunch, after lunch. So we're going to have a big session on this. And I know Dale has introduced the possibility of another hypothesis, and there may be more. I mean, we could go through the history of science. Those that have done a lot of work in science don't always have the hypothesis that wins out in the long run. Lots of books on that as well. So there could be more. We want to allow for that. There's a richness to this. So if you feel there's something else, there's a box for other. We're good with that. We're good with everything. We just care about the result. Okay, are we good? So you got to work during lunch. Eat fast, think. It's going to be hard to do both at the same time, but try to, and also put your 3 check marks where you see they fit. Well, do we have stickers, Peg, or do we have markers? We have dots. Okay, we'll give you dots. Yep. Okay, are we clear on that? Yeah. Okay, so we're good? Everybody's got it? All right, thank you.
Okay. Time for lunch. All right. I think Dan's getting hungry. Anything more? You got that button? Go up? Okay.
Okay, just a quick. If you could just listen up for a minute. Thank you, Jane. We're going to have 3 tables set up here. We have turkey and Colby sandwiches. We have ham and Swiss, and we have a few veggie sandwiches, apples and chips. And at the back table, we'll have iced tea and lemonade, as well as different flavored LaCroix, Diet Coke, and regular Coke, as well as our tea and coffee at the back. So enjoy.
Okay, let's we'll let the people get things organized. I think the drinks are available to start with, right? We had to completely reconfigure this room to make all this work today, by the way.
How are you doing? Good to see you.
Hi. I'm glad you made it.
Yeah.
I knew you were gonna be late, but I was wondering.
I had a meeting this morning,
Yeah.
so I told Tail, I'm like, I'm gonna be there. I will
Okay.
be there.
Wonderful, wonderful. What about tomorrow? Yeah, oh yeah. Were you gonna be available tomorrow too?
I wasn't planning on tomorrow.
Okay.
It sounds like, is that the?
That's the
forever
GEM meeting. No, that was yesterday.
Oh, okay.
Tomorrow's the GEM meeting where we present to our members what's going on.
Tail.
Dale thought it might be over for tomorrow. He wasn't sure about that.
People on their toes.
Yeah. Isn't it amazing? It's been almost a year. It's 11 months since we very first talked. And it's come a long way.
And you know, it's funny, I used to work with
Yeah, I didn't know that.
way back.
So it was her work on Silver Lake. When she did the Silver Lake study, we picked up that report. We were trying to find out where do we go from here, I think 2 years ago. And that's what triggered the Silver Lake study. I still think that's very, very well done. I really do. Yep. It was just
Similar questions, similar methods.
yeah, but it was just systemic approach to the whole problem and the modeling and everything just clicked with us. And
They didn't have as much data as you guys have, so much data.
well, I don't know. I don't think many people appreciate how much data they had. We went through this whole package.
I was looking at that package.
It was a huge effort.
I believe I could He's a twin.
I swear, she didn't sleep for a month. I swear, she was just.
I was really impressed by that package.
We were meeting almost every week. With the agenda was always 1 hour. We always went to 2 hours.
Yeah.
And then we got a million assignments, and then we were emailing each other day and night, morning. It was quite a.
It's an amazing accomplishment.
Well,
That material.
The most amazing thing is that never before have the three lake associations ever successfully collaborated on anything.
That is awesome.
Yeah, it is.
Right, like at the end of all of it, right? Good things are going to come out of it. I mean, like look at the turnout of people.
Yeah. It's we're actually at limit. I mean, there are available seats.
Oh, good. Okay.
We'll get
Well,
you
it's really nice meeting in person.
I
so
know.
I'll be here for the second
So many
half of the day.
people that the only way I knew was over a screen from a
I
computer.
have employees, like people I've worked with for 2 years, I've never met. Never met him in person.
And it means so much more to be able to be in person.
Hey, Dale, how are you? Good to see you.
He's wondering what we got him into now.
Well, but
All good things.
I think it's cool is that I have all these. I have my dad's stuff. I got a whole new theory of the whole place. And you got me just where I was saying it's not the gold one out there. It's not so much. But it's the green that's working. And so what Jan was saying that all these things got all this guy that's stuck in the sand mine. They can't get to the sand. They're still they're showing themselves.
Well, it's
Is that up there? They should put it up there in the other category.
it'll be in the other.
The other thing is that I have the mic here, so I can.
Oh, no.
I gotta
That's
watch
dangerous.
it.
It's
He's got it
dangerous
turned on
now.
right here. So we just told her.
And then, we're doing the nucleus. It'll just underneath.
And then I'll let it, a few days and then we go.
He's got camera set up over there in the corner. So we have we did a lot of pre-planning.
Where do we put nitrogen on that? Nitrogen in the air? Is that
Well,
under climate
it's
change
nitrogen
or is it under
deposition, but the only place that nitrogen deposition comes in is in decreasing the phosphorus. When it's inside the body.
Okay, so nitrogen in the air has nothing.
Nitrogen deposition changes the ratio of nitrogen to phosphorus and makes it looks like phosphorus decreases.
Okay, got it. All right, sounds good.
That's a challenge.
Okay. All right, if you haven't already grabbed your sandwich, it's probably a good time to get your sandwich and your drink, because we do want to get those dots up on our survey voting.
Oh, I just saw that.
You did talk?
Yes, well, I'm just about to pick it out.
Okay, so you guys together. I would have loved to do it. No, I'm the one that signed it. We'll find out.
That sounds good.
So, and they're doing more of this now. Okay.
What's in lawn fertilizer? Phosphorus and nitrogen, right?
Mostly nitrogen is taking the,
So nitrogen eats the phosphorus. So nitrogen, it really doesn't seem to be a problem because it's an alkaline. So really the fertilizer in the yard, the way I'm reading what you're saying, is not a big issue. I'm reading something into it that's
And I think the reason why
plants grow more, they stimulate, and they actually use more on all kinds of dynamic. I don't have. So if you have no phosphorus in the soil, you could still be generating phosphorus out of the soil.
But I just don't want
I can give you a solution. This is Well, that's, yeah, that's the conclusion I would draw. Not a scientist, obviously. Was that yellow, I guess,
I don't think we can take, I don't think we can take.
I was
the low
trying to make it
phosphorus
easy.
hypothesis. I know, yeah, no, I mean, that would be wonderful.
You're making it complicated.
Actually, sewers would have been easy, but
Yeah. Well, I'm
expensive.
hoping that we spend some more time on the septic systems through the course of the conversation.
But of course, they release nutrients no matter
Yes,
what, right?
right. But that wasn't
That's
on
their
that
job.
chart there.
Well, it's on.
Well, I didn't see it.
Oh.
specifically,
Actually, it's groundwater pollution.
Okay.
So in my hypothesis, this is groundwater contamination.
All right, I put my mark there.
From
Yeah.
septic system.
Okay. I see you just mentioned my personal opinion is where the most of it's coming from.
Some of your fishing questions.
The solution to the problem is to add more phosphorus.
Yeah, you're going, good luck with that.
Oh, I know. Well, so I'll tell you a story about the NLA data, the 2007 NLA data. There's a paper out there by Roger Bachman who looked at the tops bottoms comparisons, and something we never really got time to write up and analyze. And it showed that lakes had higher phosphorus in the past than they do now in certain regions of the country. So I've got to go back and actually look at that paper and then map out where that is. But the EPA went nuts. And that's I think one of the big reasons they dropped diatoms, sedimentary diatoms out of the program.
Huh.
They usually, the taxonomy is an excuse, but
Didn't
they
like the
didn't like
answer.
the controversy. Yeah, they didn't like the answer.
Jen, you said you were looking through some of Doug's data from the last sediment collection for so you were
I
looking
was
at,
sending them the samples after we.
well, just, okay, well, just to understand your interest. So if you find benthic diatoms in there, then you're optimistic that if we did cores, we could track benthic diatom change over time. Is that, okay. Well, how do you think the benthic diatoms got into the trap sediments?
Oh, they just get suspended in the nearshore zone from wave action, and they just float up off the bottom. And
Well, then my question for both of you is, wouldn't particulate phosphorus potentially then also be in the trap sample? And if so, are we overestimating the acetylene loss?
Particulate phosphorus is the only kind of phosphorus that's in the trap sample.
Okay, but if it's coming from turbation of the shallows, then our estimate of true settling is overstated, isn't it?
well, except that phosphorus had to get into the system somehow anyway, right? So if you take one step farther out, does it matter whether it's coming from the phytoplankton?
Well, it kind of, because when you see the experimental setup of the trap, there's care taken in their placement to be in the epilemium, right? So
Right underneath the hole. When we avoid the winter stuff, because we know there's a lot of resuspension well distributed through the lake during that period. The stratified period, you don't get any resuspension of stuff that's down below the thermocline. There's still resuspension in shallow. That's why you need really ideally a trap to get a bigger lake. And that's why it came out in Lake Michigan. You need to get far enough away from the shoreline that you don't see that influence of wiggle zone resuspension. The Torch Lake probably does not completely get you far. You can't get far enough away from the shoreline.
I mean, otherwise the benthic diatom shouldn't be there in general. They shouldn't be
In
getting
general, you wouldn't see them in the summer samples.
Sure.
But you can see very small numbers, very small abundances of those. Sea of calcite.
That's the idea.
Just, I know you guys are big into science. I'm an MD doctor, so I have a little bit of a science brain, but not like you guys do. I just what Trish had said earlier about what's going on in Torch Lake, and I live on Torch Lake, and what I have noticed as well is that there are two different things happening, and they might be the same thing. One is that the pictures that we're seeing in this presentation, that covering of the stones with that mat. But then there is this, to me, and I haven't physically touched it myself, but when I've been in a boat, it looks like they're almost plant growth coming up that she's describing. Tends to be out further. I've noticed it. It's probably out at least maybe up to your head level if you're out walking in the water. There's more of a plant growth and you see these long, when you're in a boat, long trails going up the lake, and it's a certain distance
So it's
offshore.
floating on the surface? Or?
Well, it looks like it's really, really dark. And I've seen other people holding plant life. They're pulling this plant muck up. And I'm like, that doesn't look the same as the cows, the hard.
Send me a picture and a sample.
Well, that's what I'm wondering, if this Dr. Saint Armand did she see that when she was doing her testing of the water?
Not that that's a map. She's just on water.
It's
You
not
want to
a
avoid
mat.
that.
Usually it's not coming to the surface where I live, which is on the southeast side. It's not coming to the surface, but it's a dark, looks like plant growth that's maybe, like she said, up this high, and it's under the water.
Is it a magnified of some kind?
I haven't touched it. I don't know what it is, but I'm just thinking we need to get a sample of what that is. Yeah, and maybe I can try and get out there this year on the paddleboard. Okay. Yeah, but this is new. This was never there before. What she was talking about, Trish said, there's something else. And that's when you guys got into this discussion of it, the columns. And I'm thinking,
There's some here short columns that I'm used to.
Okay.
Used to have those kind of columns. I wouldn't say that.
So it's not really a plant then. It's
so those tend to be lightly colored because there's not any algae. There aren't any cells in it.
Okay. Okay, well, maybe we can get a sample of that, but keep it in your mind, because I don't know, I'm not going to be around for all your scientific stuff.
You folks gonna vote? You can register right now.
Yes, sir.
Not yet. Can we wait till the end, or does it have to be now? Okay, we have to do, all right, all right. Oh, really? All right. All right, I guess I never did.
Please pay attention to Dan.
In about 5 minutes, we're going to take the food away. So it's last call on food. When do you have to take it back? Well, 5 minutes. At about 12:30.
So we went to airport. I remember Chicago. Yeah, no, it's been a while, for sure. And actually sitting down and talking about that stuff. So your perspective from your original well, there's that whole side of that conceptual model. We have Sherry. Sherry used to work with a team of hydrologists at MSU that I worked with. And we approach problems like that. It's so small that I do most of the work volunteer. I think it's an interesting problem. And they need to help. Well, it serves them then, right? Next steps. And I'd like to get a couple of pictures, but that's going to have to do solve problems, or understand the problem like that you can't solve it. Or it's too dangerous to solve, or whatever it is. Because I would They're already collecting samples. I mean, it's probably a little trickier, but not as tricky as some of the nitrogen species are. It's also jam. It's like a band. It's like a band. Jam ideas. No, so people are, Anthony shared that Dave Hyman was their advisor, and Dave and I are really close friends. So they have a nice process basement. I don't know whether advertising. Oh yeah. He might just be interested. I've never met him. But we had a wonderful conversation on, what's going on. It's not something else that really got me involved in. I remember years ago. He just wanted to talk. I couldn't keep up with you. So you might want to just. I retire next year. This is just fun. I've got a place north of here, but it's still that's not important. It's body of knowledge.
Can you hear me okay? We're going to start in 5 minutes. 5-minute warning. Minutes to go, no? My stopwatch. I'm gonna go see if anybody's in the bath. I'm gonna see if-
I think so, yeah.
Okay, can people take their seats? Time to get going, Amanda. If everybody could take their seats, we're gonna go. There was somebody sitting there. Bob. So Bob and somebody else, right? Okay, Bob's okay, we're gonna get going, folks. We're gonna get going. Good, Norm? All right, we're gonna start in like literally seconds, so if folks could find a seat and quickly swallow whatever you eat. So I think we're missing Jan's there and Doug. Is Doug okay? I thought I saw him a moment ago. I want to make sure everybody's geared up and ready to go because I know this is the lull after lunch, and so we need to manage the lull after lunch. So I'm not a biologist, but I can pretend I am with the best of them. So the way the meeting's evolved, Dale and I have had a discussion, and we're going to adapt something we'll talk about later to the agenda to be something slightly different based on the way we're kind of the conversation's going, which is really good. So we're going to switch on the agenda, the two talks that it's in order, Dale, followed by Jeff. And then we're going to do those two talks, and then the session about, that will help set up, the discussion will then have about golden brown benthic algae and the voting, which you folks were all kind enough to do. And it'll add more content to that discussion is what we're betting the farm on here. And then we're going to roll into the monitoring. So we're good with that? Oh, and one request, one of you, Kathy, was very kind enough to mention this. If whoever's speaking at the tables could make a deliberate point of speaking close into the microphone and projecting, right, Kathy? That would be helpful to the room. Okay.
Okay, thanks, Dan. What he didn't say was, I was really trying to get a little more time, and I might need a little bit. Jan was talking about trying to get 6 years of talks into 25 minutes or 30 minutes that he had. I'm going to try to get 30 years of slides into a short period of time. I'm a little bit of an outlier to this group. I haven't been dealing with Torch Lake very long, and I don't know how much connections you really have had to the U.S. Geological Survey. We do a lot more than just putting in stream gauges and monitoring stream flow. And what I'd like to do really quickly is talk about some of our capabilities, especially in lakes and reservoirs. Now, when we start looking at lakes and reservoirs, there's a number of factors that are affecting water quality. And they really vary by which lake you're involved with. But we have ones like natural factors where you got natural runoff coming in from forests, atmosphere, whether that's natural or influenced by anthropogenic things. We also have other factors like urban areas, maybe for torch that's suburban areas. And there is some agriculture coming in from not directly into the lake, but upstream. So we have all these factors that are affecting lakes. And in addition to that, there wasn't really much talk about this, but the fisheries in the lakes have been changing. Each one of these can have an effect on water quality. And what I'd like to do is to try to get into some of those areas. Now, one of the main factors that's driving water quality, and maybe not so much on torch, because there's been talk about maybe this whole thing going in the opposite direction, but it really deals with eutrophication. And with eutrophication, what we have is back about 10,000 years ago, most of these lakes formed, and those were lakes that have low nutrient concentrations, low algae concentrations, clear water, and there are still a few of these that exist. We're just lucky enough to be by Torch Lake, which is very similar to this. However, with time, all these lakes start to get more nutrients, they get sedimentation, they fill in, they become mesotrophic, moderate nutrient concentrations, moderate algae, moderate clarity. And luckily enough, we don't have this on the agenda today, but there are a lot of lakes that are eutrophic. Higher nutrient concentrations, high algae, and poor clarity. Now, if you look at this, I've really been talking about 3 indicators. And if we talk about the monitoring that Doug was talking about, we really concentrate on these 3. Phosphorus concentrations, the source of the problem, chlorophyll A, and water clarity. Now, one of the things that we should worry about with Torch Lake is that logical progression, going in from a oligotrophic system into a mesotrophic system. So as we go forward with this, that may not be on the top issue on everybody's mind right now, but it is something to worry about. If we get many more nutrients, it is going to have more algae out there. All the models say it. We've seen it across the state. It is something to be concerned about. And then we're going to start to worry about it flipping back and what we can do to try to change that whole process. Now, if we look at Torch Lake, we've got all those problems with there. They're just to what level? So if we look at the sources of nutrients, we've got things like what's coming in from forests. We've got things coming from urban areas. It may be pretty low right now, but we want to keep it there. We want to make sure that we can measure it and make sure it doesn't increase. So even though we're not monitoring what's coming in the streams, we might want to add that so we can see these kind of changes taking place. Okay, so now I'm hoping you can see this. It's pretty small, but what we've done within the USGS, we've been working on a lot of different studies. And what I've done is try to summarize those. And from the minutes from this study, you'll be able to take a look at those. But I broke them down to hydrologic, water quality, habitat studies, and finally modeling, which tries to put it all together. And we've done this.
Excuse me just a minute. Does anybody have any medical training? We've got a situation out in a car here. Anybody with medical training? Okay, please. Thank you. Okay, continue. Doctor, excuse me.
Wow, that's a first. You sure? Okay. All right. So anyhow, there's really 4 different categories of types of work that we can be looking at. So, and on top of that, usually we look at those kind of studies. We're usually looking at local areas, specific lakes. Each one has their topics. And so we look at those in detail. We also have regional studies. I know this is pretty small, but I'll try to get into all these areas as I go on with this. Now, when we start talking about local studies, this looks like the USGS has only been involved with a few of those. Really, it's more like this. And Jan was talking about 6 years, 30 years. I've been involved with almost all of these lakes. So I've got a lot of different lakes and looking at a lot of different topics. And what I thought I'd do is just go through it in a series of examples really quickly and describe some of these. Okay. So when we look at that hydrologic kind of things, one of the things that a lot of lakes have to deal with is changes in water level. Torch Lake, it may be 6 inches. Some of the lakes I looked at, this is basically a pristine lake in northern Wisconsin. Over a very short period of time, this lake drops 6 feet. Now you'd say, well, it must be due to somebody changing the streams. Not this lake. It was a seepage lake, and it dropped 6 feet very dramatically. And one of the questions they wanted to know was, is that natural? So by looking at historical records, pulling them all together, we were able to see that there actually was a lot of fluctuations in that lake. It goes up, mostly changes in meteorology. And somebody could, if you look at that, there's actually short breaks in that time series, because that was a series of different water level measurements. But by taking pictures, finding historical pictures, and finding those levels, we were able to verify that that really was the case. But we went into a severe drought. And for those people involved with climate change, bingo, it's got to be climate change, and that's what's driving it. But is that really climate change? Maybe part of it. But right after that, it bounced back. And so all these papers, for people involved with Wisconsin, they have something called the Wisconsin Initiative on Climate Change. They're writing all these things that climate in the northern Wisconsin, it's going to get drier. And that's because this was happening. And then right after they wrote the paper, it shot up. So anyhow, you have to be worried about some of those kind of things. Another thing in this category, we're looking at what it would do to try, are there ways to get rid of the nutrients? And so we're looking at short circuiting. And so as part of this project, the river comes in here and it went out right next to it. As part of the project, they dug a channel across it. And we were looking at that, trying to figure out how much of the nutrients can be passed right through the lake, or right around the lake without ever coming into it. And because of the other things, they keep the water level the same. But we wanted to evaluate how these changes in short circuiting would affect water quality. Back to the climate change. One of the things that I've been involved with for a long time is climate change and looking at how that might affect things like ice cover. And the reason why this is sort of important is that's going to get to one of my hypotheses that could be driving some of the things, even in Torch Lake. But anyhow, as part of this study, I've been looking at ice cover for a long time and looked at to see how it doesn't look that dramatic, but that's about a month change in ice cover from the 1890s all the way to the present. And some of those changes can occur pretty abruptly, like right here and right here. And as part of that, I was looking at one specific lake, and that was Lake Mendota in Madison. But as working with some other people at the university, we took that and looked at lakes around the world, mostly in the Northern Hemisphere, and published that in Science. And so what we're seeing is all these lakes were decreasing in ice cover because of climate change, but it really changed on whether you got step changes or this gradual change through time. So that's more the hydrology side. So we're involved with a lot of things that are mostly deal with the volume of the water, the change in climate. We also have a number of studies that we've been doing with water quality. And part of that, and that's going to come into a little bit later in this talk, or this discussion, is trying to set up a monitoring program for a specific lake. Not for regional studies, but for a specific lake. And as part of the lake, I was dealing with Geneva Lake, which is the second deepest lake in the state of Wisconsin. This goes back a number of years. They wanted to set up a monitoring program. And so as part of that, what we did is, if you can see these 5 black dots, we went out there and we knew we wanted to oversample so we could undersample in the future. So we went out there, sampled all 5 of these locations for a couple of years. And from that, we realized that we didn't have to do all that. It was the same water quality at those spots wherever you looked. And so we cut that down to 2 spots. And as a result of that study, we cut that back down to 2. A little bit later, we even went further than that. And with this, it really was a one-dimensional system. We could get almost all the information we needed from one location. The next thing we want to do is to look at how often you have to sample. So we started off by oversampling all 5 of those locations and then cut back to try to see, can we still get all that information without really putting that much money into it? And so we started off doing it every 2 weeks. We end up, now we sample that every month. So we came up with a strategy. We also looked at what constituents to sample. Another thing a lot of times I get the question, why are you doing this monitoring? And there's states, other places have certain criteria to classify a lake as impaired or not. And as part of this detailed study, now this is on the deepest lake in the state of Wisconsin, which is Green Lake, deepest natural lake. We have a very rigorous program going on there. And with that, we're looking at it and found that the lake was actually impaired for total phosphorus. They have a criteria for that, which is 15 for that particular lake. But we also, because of the other things, we found that the lake should be listed for dissolved oxygen, which is extremely rare. And what happens is that this bottom decrease near the bottom, that's not unusual. Almost every lake you look at. I'm not sure if Torch Lake, if we could increase the sampling, we might see this in Torch also later in the year, but it'd be interesting. But we also saw in this particular lake, right below the thermocline, the oxygen ran out. And so the lake was actually listed as impaired for dissolved oxygen and for total phosphorus. One of the things is when you get this detailed information, not only are you collecting at the surface in the bottom, sometimes it's really helpful to get it throughout the water column, and you can start to look to see these changes that are occurring through time. And so very often we'll be sampling, and we'll not just sample top and bottom or in the middle, we'll have very discrete depths, and we can make time series out of that. And through this, we can see the changes that are occurring in a lake. Now, with one of the things, by looking at those changes in different layers, we did this again for Green Lake, and as part of our study, we only sampled the last few years, but we were able to go back into the earlier records, and this particular lake was sampled by Burgeon Judet way back in the 1900s with really fairly sophisticated equipment. Well, actually, it's taking samples and analyzing them in the lab, but they could figure out how that dissolved oxygen was changing at these different depths, because we had these all the way through time. And as a result of that, we could show that the dissolved oxygen had actually stayed really steady about to the mid-1970s. And then all of a sudden, they started to have that problem. As part of this project, we were trying to find out what led to that and what they can do with it. And one of the things that, as part of this study, I'll talk about a little later, we did a lot of modeling to try to explain what caused that problem. Now everything I've talked about is really taking temperature or profiles at one specific location. And one of the questions that often comes up is that site representative of other places in the water column or throughout the lake. And so what we have right now is, we told this, looks like a black suitcase, but really what that is it's a series of different sensors on that. And what we'll do is go through the lake and we'll tow it up and down. You see this? And so we go up and down like a yo-yo through the water column. And from that, what we can do then is this little yellow line on top shows where that we pull that sled. So we're bringing it up and down over 100 feet. Actually, it's about 70 meters that we're bringing it up and down. And from that, we can look at it and see how representative our sampling location is. We can see if things are variable across. And so the one that I was interested in this study really was dissolved oxygen. And we can see that band really across the entire lake. So we knew it wasn't just on the east side of the lake, it wasn't on the west. We really could get that and get a good description of the entire water column. Okay. One of the things that's really helpful, and we've been trying to do that, Doug was talking about this, which was trying to put a time series together. Once you get that monitoring program through it, you can start to get a really good time series. And this is actually all, well, up to 1984, that's all USGS data. And then I was able to get some university studies to add it and go back in time. But we're able to get this long time series together, and we can link that to management actions. So in this particular study, this was what I would argue is one of the most extensive rehabilitation efforts ever done. And that was on Delavan Lake in southern Wisconsin. I've been working on this lake since about 1990. But as part of this long time series, we can look at it and see what was driving some of those things. Yes, it's correlation, but you can see things like right here. If you could read it, there's this huge drop from about 200 micrograms per liter down to about 15, a massive drop. Well, they applied elm to it. They sucked all the water, the phosphorus out of the water column, sunk it to the bottom. But you can also see some of these other things when they did work on wetlands. Basically, they eradicated the fish in the lake. As part of this project, they killed every fish in the lake, which was, it had its reasons, and I'll show you that in a second. But the problem was that they didn't take the fish out. So they had 1 million pounds of dead fish out there rotting. Well, that's what caused that phosphorus concentration to rise. So, but anyhow, this time series can be really useful, and you can start to see the effects of what some of these actions are going to do. And once we get a time series together, once we get this time series together for Torch Lake, we can start to look at management actions in the future and how they're affecting things. So that's mostly the water chemistry. We also do some things, we don't really get into the biology really deeply, but we do get into some of the plankton studies. And so instead of just taking our water samples, we'll be towing nets through that, whether we take it from discrete depths or all the way through the water column. And with that, we can look at that and see how other things are changing through time. It's nice to have that on a short snapshot, get it what it's today. But when you get these long time series together and they're consistent, you can start to see changes in this. For a few of the people, the last number of years of data, I think since about 2012, those are all done by Phytotech. And St. Amand did a lot of these analysis for us. But we've been able to generate a really good time series. And what we can see then is, if you look up there at the very top, it was large zooplankton originally. By the time we get to the bottom, that's changed into all copepods. And that can have an effect on water quality also. It might not change the phosphorus concentrations, but it can indirectly change other things. In addition to the water column, some of our studies are looking at the bottom sediment itself. A few people were talking about, Aaron, I think you brought this up, about phosphorus release from the bottom. We've got a number of studies, and if we do the monitoring right, we can pick up a lot of that. Because what happens is that after it stratifies, bottom sometimes goes anoxic, sometimes other things change. And right now, most of the sampling I think is pretty early. This lake probably stays stratified until mid-November, just being as deep as it is. It's going to go really long into the year. And that gives a lot of things to cook a little bit further. But if it does go anoxic, not saying it will do it here because there has been some studies, but very often phosphorus will come into the water column and be able to get it up to the top. And so what you end up with phosphorus, high concentrations in the springtime. Now there's various ways to measure that. And a good monitoring program, we could just have a lot of phosphorus and see how much builds up in the bottom. So that's the way to indirectly measure how much is coming out from the bottom. The other thing we can do with these kind of things is, like I was saying, instead of taking measurements in the water column, you can take cores. And I've done this with a number of different groups. We take cores from the lake and we can bring them back into the lab. And I've worked with a couple different groups. The Army Corps of Engineers, Bill James, this is actually his lab, where I send him the cores, incubates them under different temperatures, different pHs, different DO conditions, and sees how much comes off of it. Bill recently retired, and I've managed to get the Wisconsin State Lab of Hygiene to be able to do that for me now. Now, as part of those cores, one of the things we were looking at, this was upstream of Delavan Lake, when I was saying they got rid of they killed all the fish. Well, they killed all the carp. This particular lake was completely turbid, not a weed on it. After that, the weeds grew up like crazy, and I wanted to see what the effects of that were. And I thought what was happening was that the weeds grew up on the top, and I thought the bottom would go anoxic and would release all this phosphorus. Well, that was the theory going in. But after going through this, what we did is we put a little platform out there. This is the same lake early in the spring. All that the weeds grew up as the year went on. But we were out there, we measured temperature, oxygen, pH, and got all those conditions, took the sediment cores, and then we could calculate how much phosphorus was coming off. And as a result of that, we found some really interesting things. And some of this is some of the first early times when they started to look at the effects of pH on sediment release. But what we found is that they had all these carp originally. They got rid of the carp, and it got really clear. The macrophytes grew up, and based on some numbers, we could estimate how much phosphorus was coming up into the weeds that later die. So we estimated how much that was. That increased the photosynthesis, which resulted in an increase in pH. So all of a sudden, we're getting up to 9, 10 for pH. And it turned out that there was an oxic release, and this was one of the first times that was showing that the pH was really affecting how much was coming in. And my theory originally about anoxia was really pretty wrong. But anyhow, by going through all this, we can get an idea where the phosphorus is coming from. We also have various studies where we've gone out and taken sediment, brought them back to the lab, looked to see how much stuff is really into the bottom sediments. Are there PCBs in there? Is there DDT? Is there mercury? Those kind of things. And we did that as part of a study on Geneva Lake. Up to now, what I've been talking about is water quality at certain lakes. As a part of a big study that was done here in Michigan a few years ago, they tried to link water clarity in with things that we can get from satellites. We can get to the satellite to get an idea of clarity and correlate that with Secchi depth, and we put that all together. And so as part of that, we're able to describe the water clarity in lakes throughout the entire state of Michigan. Now, because water clarity is often, I was talking about those 3 indicators, they're often correlated with chlorophyll, they're correlated with phosphorus, and it's sort of noisy, but it still correlates pretty well. By that, we were able to describe the water quality of lakes throughout the state using satellite information. Our sampling isn't just in the summertime. We also go out in the winter. I have to throw this in because I don't get out on the lakes very often, but that is me sampling the lake in the earlier days, I guess. But anyhow, we go out there and we collect that because it is important to sample in the wintertime on a lot of lakes because there's different things that occur. One of the things we want to do is you'll never see this on Torch Lake, but on some of these small lakes in other places of the state, country, you can also have this problem with winter kills. All the oxygen runs out. This is a little Saint Germain, and as part of this early part of the study, we went out and red is bad. It's basically less than 0.5 milligrams per liter of oxygen, so it was winter killing. We knew that, especially, and this was able to quantify it. As a result of this study, we convinced them that they should stick aerators in it. The study continued on, so we could see, actually, there should be a hole in the ice up above it, because it actually put a hole through there. And so what it did is it wasn't really pumping in oxygen per se, it was really putting a hole in the ice so that the water could circulate and get oxygen mostly from the atmosphere. But as a result of this, we could come up with a strategy to improve water quality and evaluate it. Now this is all the lake itself. One of the things that we also do is really want to look at the effects of the watershed. And to do that, usually we want to quantify how much phosphorus comes in, but to do that, we often have to quantify how much water comes in first, because the phosphorus generally comes in with the water. And so as part of that, a number of our studies, we go out and we do a complete water mass balance. To do that, we look at the change in storage, hopefully over a year that's close to 0, if we do it long enough. Then we want to measure precip, stream flow, groundwater in, and then measure all the things that are going out, like evaporation, surface water flows, groundwater. Some of those we can measure, some we have to get from models. Evaporation, generally we're getting from some type of a model, whether it's a simple average, depending on the scale we're looking at, or using a more sophisticated model. But we want to get all of those. Now this is where usually what people think of the USGS. We are very good at measuring stream flow. We can go out there and get an instantaneous measurement, whether it's using more standard techniques or using dragging acoustic velocity meters of some sort, some kind of a Doppler meter across the the river, and get estimates of flow. That'll give us spot measurements, and those are good. And if you look at Torch Lake, that's what it's relied on in the past. There's a few of these, and I think even then, I think they called it the USGS measurement, and they threw an orange in it and measured it. But anyhow, we have a little bit more sophisticated measurements than taking the old orange technique. But what we can do, and that's what's really important, is if you really want to measure how much is coming in through time, we can make those things and make them automatic. And so what we do is we've got some more traditional ones, just measuring water elevation. And from that, we can estimate and get a relationship and get flow through time. Or we also have other ones where we put acoustic velocity meters out. And from that, we're measuring how fast the water is moving. Using the shape of the river, we can estimate how much is actually going down that. With that, we can get a continuous trace. So if you go out here and just take a few measurements, it's really tough to get an estimate of how much water is coming in. And to date, I think that other than higher up in the watershed, this is really, that's the only place this kind of approach has been used. And so most of the water budgets and everything else on Torch have been used on just a few spots on here. And you can see how you could get answers that might not be quite right. For groundwater, we've also got ways of coming up with how much groundwater is coming into it. We can put in some, Jan was talking about piezometers. Usually we stay out of the water. You can't tell because of the snow, but this is just up, so we don't have to worry about water coming in from the sides. We usually go up onto the land, put a piezometer in there, get a water level. And then from that, if we can get some kind of a model, we got a number of groundwater modelers that have done this. I've worked with several of them. This is for a lake in northern Wisconsin, Mercer Lake. But from that, we get those few measurements. By putting a full groundwater model, we can not only tell how much water is coming in, but we can tell where it's coming from. And that, a lot of times, you can trace it back to sources of where those, not only the water, but also the nutrients are coming from. But the really reason why we do most of these water budgets is to tie it all together in terms of phosphorus. That's what we want to do, is to know where all that phosphorus is coming from, how much is coming in from precip, inflow, outflow, groundwater. And then we got these little weird ones like geese, septic systems, point sources. But you really have to get all those together on a lake, even if there's zeros, to get to understand where it's all coming from. Somebody was asking about atmospheric deposition. We have had studies where we put some of these out, put up our own little weather station, collected the rainfall, and then at other times we basically just take samples after a week, water it down, and figure out how much is coming in from dry fall. And we've done this in a couple places already. Uh-oh, there it goes. So in addition to that, now if we go back to that stream flow, in addition to measuring flow, we also have ways that we go out there and collect uniform samples, a sample that really represents the cross section. So in just taking a grab in the middle, usually we'll go out there and take samples throughout and get a really good concentration to put together with the flow. Now that'll give us points. And again, that's what's been done on Torch, but it doesn't really give us all the points in between. And so what we do also have is we have something that sucks the water out whenever we tell it to, whether it's changes with flow, changes in elevation, changes in time. We take the water, suck it up through a tube into our ISCO samplers. Inside there, we have a series of different bottles, and we can fill them up and tell the instrument to collect them whenever we want to. So instead of just having somebody, whoever can get out there, we can automate all that and really get a good time series of that. And from that, I know it's hard to see, but here we got continuous flow, a whole series of phosphorus measurements. We can put that together to get a really good estimate of the total load coming in from the stream. And we've done that in a number of different studies. Groundwater, in addition to measuring the water level, we can suck the water out of that and estimate the concentration of the water. Now, if we put that together with our groundwater model, we can go through there, and what we're seeing in this particular one is that it's got higher flow, but it also had higher concentrations, and we can estimate how much phosphorus is coming in from the groundwater. And there's ways to actually separate this from how much is coming in from septic systems. How much is coming in from septic systems? Usually that is not trying to measure plumes. That's going to be a hit or miss. So usually what it comes down to is theory. And so we count how many people are directly around the lake, multiply that by a number, and then assume a certain amount being delivered into it. And by that, you can get the low number, high number, and get a range for that. Okay. For geese, that can actually be a fairly big number on some place, or just waterfowl in general, because a lot of times the last lake that's opened up, a lot of times starts to get all the geese accumulating in one spot. And in some of these, it's been amazing how important waterfowl is to a total nutrient budget. So anyhow, after you're all done with all of those kind of things, really what you can do is to construct a phosphorus budget. This is something that I think that Doug was mentioning. He was saying that 30-30-30 idea. Well, by doing this, you get a little bit more confidence in each one of those. And we can figure out where it's coming from, how much is coming from this stream versus that stream, how much is coming in from waterfall, how much is coming in from septic systems, and get all that information. And so you can see where should you go out there and and, you know, to put your management. And we've done this on a number of lakes. Most recently, I've done this on Green Lake. And again, that's the deepest lake, so it's not that all that different than Torch Lake. But once you get this down, it really tells you where to go after things. I had mentioned a little bit about the biology. Well, biology can actually have a lot bigger effect than a lot of times you think. And one way to look at that is looking at a trophic state index. A few people mentioned that. And very often you get the same trophic state, whether you measure Secchi, chlorophyll, or phosphorus. You use the equations, they all come back together to the same number. And that's what happened here. If you look at before this rehabilitation I was talking about, they're all saying the same rough thing. They're saying it's basically a hypereutrophic system. Then this management came in, and all of a sudden things really started to say something different. Phosphorus concentration said it should be a eutrophic system, but the clarity said it should be almost an oligotrophic system. And what I'll show you is I think that was driven by the biology. Now in Delavan Lake, this lake was loaded with trash fish, carp, bigmouth buffalo. And so what they did is they came through here, they had a lot of carp, very few, smaller fish, and that resulted in small zooplankton. And when you have small zooplankton, that's like having a really lot of small vacuum cleaners that don't work very well, and you end up with a lot of phytoplankton. But what they wanted to do was to go through here, kill all the fish in the lake, replace those with walleyes. Those walleyes are going to eat all these fish that are really good at zooplankton, eating zooplankton. You end up with big zooplankton now, and those guys are good vacuum cleaners to eat all the algae. And so you get a very different kind of thing. And what we're looking at was trying to see how the biology could affect things like water clarity. So if you look at this now, before everything was telling the same thing, the reason why we had high phosphorus but still really good water quality, it's because those fish were eating all the small those plankton, or they weren't eating the big ones. So the big ones are around to bring it down. And so that was all done in that study. So that's a good way to look at all of this, but that's piece by piece. Those are all the tools that you can look at and try to look at those in details. The best way to put all this together is using models. And all these models have the same general traits to them. You want to find the source of the problem. Sometimes that requires other models. Once you have the source of the problem, you want to quantify it. So if it's phosphorus, you want to go out there and measure all the phosphorus getting into it. Then you'd want to take that model and somehow determine how the water quality responds to changes in that source. And then finally, once you have that response curve, you can go through here and figure out exactly what kind of actions you want to take. So if we go back to Torch Lake, and let's say the problem is in the future, it might be nutrients. So we want to go out there and measure all the nutrients into the lake. I just want to go through a quick example for Green Lake. Again, that's the deepest lake that we have in Wisconsin. We wanted to go out there and figure out how much was, what was really causing the phosphorus problems in that lake. So the first thing we wanted to do is to measure the problem, really quantify it. We got the total phosphorus budget. Then we go through and try to put it into, there's a whole series of models you can use. The one we use is a very simple model, which just said that phosphorus is a function of the loading to the lake. It's a very, very, very simple model. But what we can do with that model is to take that and show that it works for the lake. So on this one, it says calculated phosphorus versus measured. So we went out there. Our lake sort of fit right down that curve. So we felt that the model worked. We then ran the model at a whole series of different concentrations. In this case, we increased the phosphorus by 50%, decreased it by 50, and we get this response curve. And that's something that you would want to do for like Torch Lake, or maybe you don't want to go down, you want to go higher. But from that, we could figure out what phosphorus reduction you would need to try to get that lake to be no longer listed as impaired. Okay. You can also use these kind of lakes to go further. And what I did in this study was to go out and look at a calcareous lake, a lake very much like Torch Lake, and try to, can we use that same basic approach to figure out how the lake should respond to changes in loading. And we did that by first of all computing a phosphorus budget. Don't want to look at the numbers, but we figured out how much was coming to the lake, put it into those models I just showed you, generated the response curves. However, when we first did this, we found out those response curves didn't work right. They constantly over-predicted to that. And that's because a lot of that phosphorus was coming in, and precipitating just like in Torch Lake. And so had to modify those response curves. But from there, we could use that to see how changes in loading should affect that lake. Have another study fairly close by on Silver Lake. We wanted to take that same general approach and try to look at not only, in this case, we wanted to try to see how would reducing septic input affect the lake. So again, we followed very similar to what I was just talking about. However, in this case, again, we sort of start with that basic equation, but now instead of just using the equation, we actually use bathtub, which has those equations within it. And so by going through there, we can not only look at phosphorus, it has equations similar to this for chlorophyll and Secchi. And by doing that, we could go through, get a phosphorus budget for the lake, and then start to run scenarios to try to find out what would change the septic systems and other kinds of things due to the water quality of the lake. And so in this case, we had a base condition, and then we can see how would changing the amount of loading to it would really affect things. Running these type of general models for other kinds of lakes. In this case, now it's not the deepest lake in the state of Wisconsin, but this is the largest one. What we wanted to do is to try to figure out what it would take to get this lake no longer listed as impaired. So again, now what we wanted to do is to first of all go out there and quantify how much is coming into that lake. So we wanted to get the phosphorus budget basically. But now instead of just having one lake, what bathtub is really good is putting these lakes together. So one lake feeds to another, to another, to another. And what you can do is do everything we did for Silver Lake, but now do it for a series of lakes. And what we end up then is a whole series of response curves, the same thing, where we have certain phosphorus reductions, come up with one, or increases or decreases. And by doing that, what we could do then is run specific scenarios. We could say, what was that lake back if all the streams had 20 micrograms per liter or 75 micrograms per liter? We can drop off the point sources. We can run all kinds of scenarios and figure out how that lake should respond. Now, the one thing we did in this lake, it wanted to say how quickly would it respond. I think Doug said if you change the loading, it would affect things in 2 years. This is actually, we developed the model. It's called the Jensen model, where we looked at that, added internal loading to it, and tried to figure out how long it would take for that lake to change through time. In that lake, it was actually, it was loaded with sediment, and they knew it was going to take a while. So yeah, you're looking at it, even if you drop the loading, it was going to be about 50 to 75 years before you're going to get that lake to be no longer listed as impaired. Now, those are really basic, simple models. Sometimes you need more complicated models. If we go back to Anvil Lake. Yep. Yep, 2 minutes. Yep, 2 minutes. Sometimes you need more complicated models. Now, in this particular model, we wanted to figure out what these changes in water level would do. So in this kind of thing, what we did is we had to go into a full-blown hydrodynamic model where we're simulating more than just the top or the bottom. This one, you had to simulate things all the way through time and space. So it's going into the actual processes. With that, we could look at those changes in water level. Once we calibrated the model, we could run it under shallow lake conditions, deep lake conditions, changes climate. So if we had cold climate, warm climate, we could figure out exactly how all those are changing. My last really detailed model here is on Green Lake. That's the deepest lake in the state. What we wanted to do is to figure out how to change those dissolved oxygen concentrations. To do that, now we don't just take the process hydrodynamic, we had to add all the biology to that and chemistry to that. And so by doing that, we're able to actually simulate the dissolved oxygen. The top is the observed, the bottom is the measured, and they look really similar to each other. And then what we wanted to do is to take and see what is the lowest concentration on any given day and see how that changes through time. And so this is what the base condition was. Now once we have that model and we trust it, we can go through there and run scenarios. And so what we were doing in this case was going through and running a whole series of changes in phosphorus concentrations and trying to see what we could get at the end result. So now, what I've really done is shown that we have a lot of different capabilities, and I see a lot of those kind of capabilities in here. It's all these tools that we have, and hopefully we can get the tools together and then put those into the right types of models. I guess that's, so really what it comes down to is which of these tools do you want to try to use and try to put those all together so that we can try to answer questions. So.
Let's be quick, and then we'll go to Jeff. So before you step off, Dale, maybe like time for two of the best questions you can think of.
Did you ask
Fred.
a question? I'm sorry.
No, I was going to say, if you have a question, go ahead.
Well, I do. Little detailed. Dale, I thought I saw in the water balance that you have to start with before you get to nutrients, that residential septic systems were in that equation. Is that correct for the water balance? Okay.
Those are only in the, assume that there's really no flow of water coming in from them.
Okay. Because of course they take groundwater, because it's usually a well and septic, they take groundwater and they return it to groundwater before it enters the lake. So there wouldn't be a net addition. We do have 3 municipal systems on this lake. And take, for example, Bel Air, hundreds of millions of gallons every season are extracted from groundwater. That then is released directly to surface water. Would that be enough of a volume to be in the water balance?
Oh, definitely, except that we have that in the Clam River. So usually when we do that, I'm only talking about the periphery, and we try to take whatever we can and put those back to where, where we can measure it. And so in that particular case, you have to subtract off of the Clam River. But yes, if it's a point source like that, yeah, you need it. It's just how you actually measure it.
Okay. Quickest of final questions for Dale on this topic. Quickest of final questions for Dale. If none, we're good. So Jeff? Thanks, Dale. I think we're good on introductions. Jeff knows who he is, and he knows he's from UC Davis, and that's all Trish cared about. So.
As long as Trish is happy. Yeah, so once again, it's great to be here. Yeah, thank you for the invitation. I should say I've known Dale a long time, and he always eats into my time. So today is no exception. So I guess Dale said his talk was going to be long because he has 30 years of data to summarize. I have 50 years of data to summarize, but I'll try to keep it brief. Okay, so some of you have been to Lake Tahoe. If you haven't, this is what it looks like. Like Torch Lake, it's got its pluses. So beautiful lake, blue water, we have mountains, and this is what it looks like in many parts of the lake under the water. So quickly, the outline I'll be following today, just a little bit of an introduction to Tahoe. I'm going to talk a little bit about data collection. We have the blessing and the curse of a long-term data set. Blessing because you can say a lot about change with data. Curse because you don't want to be the person who stops it. So you got to keep bringing in the money, and it's expensive. It really is. There's no way around that. Talk about periphyton at Tahoe, which are different than the periphyton here. It's going to be one of the points, is that if you've read all the papers from Tahoe, yeah, they don't deal with periphyton. They don't deal with the same kind of periphyton you're dealing with here. We're talking about metaphyton, which may be a little bit closer to what you have here, and then just finish off with the role of modeling. So introduction, okay. All right, very quickly, 1,650 feet deep, 11th deepest lake in the world, ultra-oligotrophic, 190 square mile surface area, 310 square mile watershed area, largely forested, never glaciated, never freezes, residence time, a long time, 500-600 years. 65,000 permanent residents according to census data, 15 million visitors per year, exceeding the combined Yellowstone, Yosemite, and Glacier National Parks. So that's a stress on the system. A lot of people have asked, oh, do you have this shallow area around the lake? Well, yes and no. The highest contours you see there are 50-meter contours, 150, 160 feet deep. So there are shelves at the, oh yeah, so there's a shelf there, there are shelves there, but generally we have very, very steep walls to see the lake. This is actually USGS data, side scan sonar data. It's a hole in the ground, pretty much formed by earthquake faults over a 2 million year period. This is our center, an incline village in Tahoe. Some of you have been to Tahoe, many of you haven't. If you do go out, please look me up. We have this incredible set of labs, offices, and a fantastic visitor center and museum. Okay, long-term data collection. So I think Tom mentioned early on, he wants to have a state of the lake report. Be careful what you wish for. I wanted to have one about 15 years ago, and if I wasn't here today, I'd be writing the 2022 State of the Lake report. It's incredibly useful, mainly because it forces us to look at our own data every year. So I'm just going to show you a couple of things from here just to put it in the context of what you can do if you have a lot of data and what a lot of data means. So this is the annual surface water temperature of the lake going back to 1968. So each of those blue bars represents about 20 or 30 individual measurements averaged over the year. So yeah, we have warming, but we have lots of interannual variability. So if you don't have a long-term data set or you have just sparse readings in different years, you're on very thin ice if you start joining points and saying it's warming, it's cooling, it hasn't changed. You can only do that when you have the data. And all of those things could be true in different places. This is the phosphorus input, the phosphorus load from the upper Truckee River. This is the largest inflow to Tahoe. We have 63 streams coming in. Upper Truckee River is about 25% of the inflow. And again, the main thing to stress here is the interannual variability. We have a very different climate, a lot flashier. We have droughts and we have wet years. Excuse me. The dark part of the bars is total phosphorus. Actually, the whole thing is total phosphorus, but the light green is orthophosphate. So the part that really makes a difference for algal growth. Looking within the lake, this is different kind of curve or different kind of graph. That's time across the horizontal axis. That's depth down to 1,400 feet. We're showing nitrate distribution here. Again, just showing there's variability. If you just measure it at the top or somewhere, a few times a year, you lack this understanding. Basically, here the nitrogen is largely depleted in the upper 200 feet, 300 feet, and we have this accumulation of nitrate that when the lake turns over, fully mixes in March of each year, you get a lot of that coming up. It's interesting if we look, I guess this is the period I was talking about, March, you see a little bit of lightening there. Essentially, you have phosphorus that came up at that point, but this was a year of very shallow mixing at Tahoe, possibly only down to about 500 feet, so we didn't have much coming up. If it had been a deep mixing year, which we get every 5 or 6 years, we would have this surge of nitrate throughout the lake. Chlorophyll. This is the same sort of data, so each of the vertical lines are days we went out and sampled. Each of the dots are depths at which water was withdrawn and chlorophyll was measured. We have what's known as a deep chlorophyll maximum. I would be shocked, but of course, I'm not a biologist, so I'm easily shocked by these things. I would be shocked if there was not a deep chlorophyll maximum here. So relying on surface chlorophyll measurements, which even at Lake Tahoe are pretty consistently low throughout the summer, may be providing some degree of misinterpretation of what's happening. We have a high chlorophyll or higher chlorophyll at the surface in the winter, but that's only because we've had deep mixing that's bringing up this deep chlorophyll maximum to the surface. Okay, Secchi depth, clarity, that's sort of the big thing that Tahoe has historically been concerned about. So going back to 68, we could see down 30 meters, about 100 feet into the lake. That has gone down. This is sort of measured from the surface down. To now it's around 60 to 70 feet. This is an issue of concern. Again, a pitch for data. So some of you are students and you're going to do projects that last a few years. You think, I got 3 years of data. I can really show a trend. Look at this period. We have improving clarity. That's 7 or 8 years. That's not what the long-term trend is. It seems like a long time, 7 years. But there are large forces at play. You get improving clarity, you get declining, you get improving. Lots of factors, physical, biological, chemical, all coming into action. Again, clarity is highly variable. And a lot of that variability is due to the one thing I know a little bit about, and that's lake physics. This is showing individual Secchi depths for 3 different years. And let's just play around with this one. This is from 2019. So we see at the beginning of the year, it's about 70 feet. After about a month and a half, it's measured at over 110 feet. Within a few weeks of that, it's 60 feet, 50 feet, and it changes. And it's not changing because we have an algal bloom, we don't, we do, we don't. It's changing in large part because of the physics. And we'll see an example of that at the end of what's producing that. Okay, so finally, he gets on to what he was brought here to talk about. So this is sort of from our 2021 State of the Lake report. This is showing distribution of periphyton around the lake. So it's all color-coded. And it's got units from 0 to greater than 1.5. What are these units? These are actually, we measure periphyton in many ways. This one is actually works pretty well for the kind of periphyton we have. We have mainly filamentous periphyton, little stalks. And so what we're measuring here is something we call the periphyton biomass index. So it's essentially the fraction of the substrate covered by periphyton multiplied by the average length in centimeters of these filaments. It's a relative measure, but it shows some areas, say here on the west shore, are red, it's high. Some areas on the east shore are very low, and they're often very low. A large part of that is because our prevailing winds are from the southwest, and the east shore gets pounded by waves. So whatever grows gets sloughed off at pretty regular intervals. Okay, so, well, that was just the figure from that State of the Lake report. Let's look a little bit closer at the periphyton. Okay, so here we've only been collecting periphyton for 30 years, not 50 years. And that's actually a very important fact that I'll come back to. So here are the routine sites that we've used on the left. There are about 9 of them over time. The color, so the little blue dots represent the actual sites. The color coding in these patches represents level of development, green being low development, yellow medium, and the red high development. The original plan, which I had nothing to do with, was to try to capture some of the things that have been raised here. What's the effect of development? And then once a year, well, when I say once a year, sometimes it takes a week or 2, depending on the weather, we do a synoptic, meaning we go out to about 50 sites around the lake around the time of maximum growth to just try to get a snapshot, a more dense spatial distribution. Okay, so here's the first difference between our periphyton and your periphyton. Ours start growing in about February. They peak in about March or April. By summer, they're gone. Oh, sorry, they're not gone. They're there, but it's way past its peak biomass. So this is a different creature. Not a creature. You know what I mean. Well, we have ways of dealing with them too. Sadly, they say. So let's look at our periphyton. Essentially, they vary with depth. So this is the lake surface. We have a dam, a reservoir, that's 62.29 feet. The natural rim of the lake is 6 feet lower, 62.23. So this is sort of typically, the lake level varies about 2 feet over the year in a normal year, and no year is normal. So sometimes we have years where we're below the natural rim, sometimes we're full. In the upper part, this littoral zone, we have these stalk diatoms and some filamentous green algae. And then below that, we have blue-green algae. And this is, sorry, big point, I forgot. This is growing on rocks, boulders, not on the sand. Very different than yours. On the sand, we have very little growing. I mean, Jan is undoubtedly right. There are millions and millions within the pore water, but our sands get moved around and abraded, and we have very little periphyton obvious on the sands. We measure, at least the program was set up, just to measure at 0.5 meter depth, 0.5 meter below whatever the surface is. And I said the surface could be up here at the dam height, or it could be below the natural rim. So we could be sampling in this stuff, or we could be sampling in that stuff. It's just one of the challenges when you have a fixed budget that doesn't grow and only shrinks. So we sample at one depth. Oops. Okay, what do these things look like? The top layer, the stalk diatoms, they sort of create this carpet on the rocks. Again, they're just little stalks. They can be green or brown, generally more brown, and often they slough off in these mats that float around and accumulate in marinas and look ugly. They're literally just hairy rocks. Usually mixed in with that, but a bit deeper, we get green filamentous algae. I think Cladophora and Zygnema are common ones. And then lower down, we get the cyanobacteria. Okay, so here's these are hard to decipher. So I'm just going to tell you what you're seeing. So at top plot, you're seeing the ash-free dry mass. So this is just a measure of the biomass. So each of those points represents literally the average from all our, or the measurements from all our sites in a particular year. And so this is the range within a year. They ran out of money for about 15 years, and then it picked up again. And you see the variation again. No matter what kind of statistics you run through it, there is no increase in the ash-free dry mass. Do the same thing with chlorophyll A. The middle plot, there is no statistical increase. And I guess the bottom one is just the number of samples, how many times things were measured. So it was pretty consistently measured during the years when it was measured. Again, an even messier plot. This is just showing time on the horizontal axis, and then the vertical axis. Oh, I reversed the titles on the top. The left one actually is ash-free dry mass. The right one is chlorophyll. The point of both of these is this is when it's peaking. It's April. I mean, different creature, different system to yours. And this is just showing again, from these synoptic sites, just what it looks like around the lake. Basically, we do get more in this on the west and northwest, especially, much less on the east, whichever metric you use, but it doesn't correlate with development and not development. Again, don't take that as that's what must be the case here. We have different soils. We have fractured bedrock. We don't have sands and silts and glacial remnants that you have. Okay. So basically, That second point is really important. Periphyton biomass has not increased despite widespread public belief that it has. Everybody at Lake Tahoe says it's way worse than it ever was. It used to be clear as gin. Maybe gin has something with the remembrance, but I don't know. But they say that. There may be some truth in it because our measurement program didn't start till the 70s. Development, big changes at Tahoe happened in the 60s. So it may have been that when they were dealing with gin, it was in the 60s, when it was clear, there wasn't periphyton, and we came along, and since that time, it has stayed constant. So it's hard, even with a lot of data, sometimes you don't get all the answers you want. Periphyton peaks in March, April. High biomass occurred at sites regardless of urban development levels. All right. Let's talk metaphyton. And again, I want to reiterate, I'm not a biologist. I'm not an algal phycologist. I'm really good at repeating things that my staff tell me. And what we've found is in the last few years, last 10, 15 years, we've had a lot of metaphyton, which I refer to, or which I consider to be non-attached algae. I mean, not free-floating algae, but these are, think of it like tumbleweed, just stuff that's, you see it on the bottom in shallow water, and you swim up to it and move it, and it kind of disperses. You can't grab it. Sort of like that. Okay, so here you're seeing pictures of it. I think this is probably Zygnema. And you see interspersed between those little patches, shells. And they're shells from Asian clams, Corbicula fluminea. And they were introduced to Tahoe about 20 years ago. And this is what the bottom of the lake looked like in the past. This is what parts of the lake look like now. I mean, there's parts that don't look like this. But wherever we have Asian clam, we're getting Zygnema. How much, can you sort of, this should be an animation. Can you mess with that and start the animation, Dale? Oh, there you are. Goes. Brilliant. Okay. So here we are. You're starting to see a little bit more of that tiny little patches of, let's call it Zygnema. But then as we get closer and as we start to encounter more of the, these are now dead clam shells on the surface, you're getting more and more of it. Totally different than what, you know, the photos I've seen from Jan's work and what people showed me from the boat yesterday. Different. Okay. Okay, we've done something very similar to what's being done here. I guess Art, who's Art? I've heard Art the pilot. Is Art the pilot? That's not here. Well, give him my best because it's great. We don't have Art, we have Mike. Mike's our helicopter pilot. And he's been flying up to Tahoe for many years, and he just got really annoyed that every year it's worse. It's worse, it's worse. Why aren't you guys doing anything about it? And the answer was, what? And he was talking about the metaphyton that he was starting to see. We would go out to our regular sites, and they didn't often coincide with where the metaphyton was. So anyway, done a lot of work in the last few years using his helicopter, using drones, using ground truthing. All right, so essentially, aside from just taking the pictures, we're actually trying to quantify them. We're using, I guess, ArcGIS products and literally creating georeferenced maps of the entire shoreline so we can track where the metaphyton is. And there's large amounts of the shore that don't have it. And it's a totally new way for us anyway of looking at this data in a quantitative way. Often it looks like one thing. We go out, we do ground truthing, we sample, ash-free dry mass, all of that. And it isn't as bad as it appears. Sometimes it's worse, but we're starting to build up a measure of how these things are changing. We experimented with lots of different cameras, some of them very expensive, some not so. Best ones we found were GoPro cameras. So this is relatively easy to do. So these are some worst of Tahoe images. This is from earlier this year. So you may say, oh, that looks golden brown. You have the same thing we have. It is not the same thing you have. This is that green metaphyton that's just dried out and been washed up. This is a part of the lake, the South Shore, where we do have those shallow banks, and we're having a drought, and lake level is falling, and this is what we see. It's also very good. Again, this is just an image from the helicopter. I mean, there you see a beautiful white sand beach, but we're seeing have a couple of streams here, and we see this is primarily dissolved organic matter coming out of the stream. Oh, one of my really ugly photos didn't, I lost it. Anyway, Tahoe is now looking in parts like it never has before, which is very disturbing for us, and it's sort of, it's disturbing for everybody, and they're all asking the same questions that are being asked here. Okay, so just the final part about modeling. And I guess I'm picking up where Dale left off, and I'm talking about the other end of models where you're actually looking at putting it where you have the lake motions and you have the biology, you have the chemistry, the food web, everything interacting. All these physical things like inputs and circulation, interacting with algae and nutrients and zooplankton and recycling of nutrients, all of these things. And the way one does this, I think, most effectively, depending on your question, but I think your question is rapidly approaching this need is with a full three-dimensional linked model. So I haven't modeled this lake. I hadn't been here 2 days ago. So now I'm here, but I've been working on a lake that's sort of similar in at least its morphometry. This is Lake Massawippi in southern Quebec. So it's a little bit smaller than Torch Lake. It's about 10 miles long, about a mile wide, close enough. And it's bent, not quite the same. All right, so we've had, one of the things we've had at Lake Massawippi is we've had instruments, or thermistor change, literally just temperature measuring instruments at different depths, at different points. And so every 10 minutes, they measure temperature. It gives us a lot of information about the physics of the lake, how things are changing. So we ran this model. I'll show you the result, the animation in a minute. What I'm showing here is temperature measured at different depths on this chain. So this is measured at 3.5 meters below the surface, 4.5, 6.5, 14.5, 18.5 meters. What was measured are the gray lines, and what the model predicted were the green lines. So, of course, I would love you to say, oh, that model is so good, it almost got what was measured. That's not the point. The point is, have you ever considered in this lake, this is June, by the way, this is right now, this month, that your temperatures will drop from, I'm sorry, this is Celsius, 15 degrees Celsius to 6 degrees Celsius in a few hours. It's not because of climate change. It's not because of a huge inflow from the USGS or anything like that. And then a few hours later, a day later, it bounces back. That there are all these oscillations, all these details happening. I suspect many of you don't. You look out at that beautiful lake, it's calm, and that's how you like it. And it's not like that. So let me show you Lake Massawippi. This is an animation we whipped up based on the on this model. What we've done is the surface of the lake represents temperature. This is surface temperature, so you'll see colors ranging from cold, 4 degrees Celsius, like 32 Fahrenheit, up to 23 Celsius, which is about 65, 66 Fahrenheit. This black area in the top, that's just, I've marked that because we put tracers in there, in the model. We didn't do anything in the lake. We put a tracer, little well, sticky particles. We put them in there so you can see where they go over a period of about 3 weeks. So again, I venture to say this lake is very similar to yours. Not quite as deep, not quite as big. And so can you do your magic again, Dale? Okay, so now you're seeing these particles start to move around. At the top, you're seeing time go by. So they're hours. So now it's been running for 2.5 days. And you're seeing these particles are moving back and forth. Can you get ready to pause it at some point there, Dale? You don't want to? Okay, well, not yet, but why don't you practice? Okay, you can do it. Now let it go again. Very good. Reflects well on your agency. Keep going. Okay, that's good. So it's starting to, I mean, it's pretty, no, Dale, I didn't ask you to stop. It's doing well. Okay, now, stop now. No, let's keep going. Keep going. What we're going to see in a minute, at the northern part, is it's suddenly going to go blue. Oh, it's getting very warm there, everywhere. But, come on, Dale, get ready. Get ready. You don't want to miss this. Okay, now, stop. Okay. What the hell happened? So you see, there are jets coming down the west shore. There's 4-degree water. This is a reflection of the data and the model results I showed you before. What happened? What I think happened, what I know happened, is they have northerly winds blowing for a day or two. That blows all that warm surface water to the south. So as that gets moved to the south, bottom water comes up. It's called an upwelling. It brings with it whatever's at the bottom of the lake, high nitrate, high phosphorus, or low phosphorus, whatever is there. And it's moving that south. In a few hours, a few days, the winds are going to drop. The system is going to relax. It's not going to spring back perfectly. There's lots of mixing and stuff happening along the way. Dale, that's your cue to continue. And you see it starts going back, the warm water comes back. So the idea with models is not that they can predict the future. They can't. I mean, they can predict the outcome of management actions or of climate change or of changes in quantifiable inputs, influx of populations, droughts, and the like. But they're a really effective way of testing hypotheses, both physical hypotheses, chemical, biological. You can start to see, does this hypothesis make sense? So we had lots of good hypotheses there. Some of them can be tested by a model if there's enough data to feed the model. I mean, the old modeling paradigm, garbage in, garbage out. You need good data to put into it to get anything you want. Oops, okay, I could have done. Okay, so one more thing you can do with a model. So this is the same model applied to Lake Tahoe. We've just released this app. You can get it, it's not an app, it's just a web page. Tahoe.ucdavis.edu, lake conditions. Essentially, we're using National Weather Service forecast of what the weather will be today, tomorrow, the day after, and showing what the lake temperature, the lake currents, the lake wave heights will be. We have something like 12 drownings a year at Tahoe. A large number of them are preventable. They're just people who go out and don't realize the water is a lot colder a lot colder in this part than in that part, or that there are waves coming in the afternoon. So check it out. Again, it's the same model. It can be used on any lake. And I think with that, that's it. I'll stop. Thank you very much.
We have time for a couple questions. Yeah, Rick. Don't talk yet.
First, quick question. What is the average pH of Lake Tahoe, the range or average, or it's an alkaline lake?
It's around 8.
Around 8?
Yeah, it's actually incredibly hard to measure.
Okay. And then second question is, and maybe you can or can't answer it, but what's about the total budget that is used to monitor the lake on an annual basis?
In dollars.
In dollars, yeah, yeah. Not in Australian dollars, in US dollars.
Well, it's never enough, but it's probably a lot more than what we have.
Yeah. It is. It's yeah, it is more than you have. And it's more than most people have. And that's causes me no end of grief when I'm reminded of that. It's probably something on the order of what we do. We receive maybe $500,000 a year. Well, but it's a way bigger lake. It's a way deeper lake. No, and I said there's a curse of the long-term data set. I don't want to be the one who has to stop something that's been happening for 50 years, although I'm about to. Yeah, it could be a lot less, most easily.
One more question, maybe, unless it's pressing. Otherwise, we're going to talk about next steps. Anybody have a question? Oh, sorry. Yeah.
Just really interested.
Go, Dave, use your microphone, maybe. Get close.
Really interested on your picture of the Asian clams and wondered, speak to the history of that. If anything significant happened biologically related to those communities?
I mean, they probably came in on a boat. This was sort of pre-boat inspections. We now have boat inspections. I mean, they're nothing like zebra mussels and quagga mussels. You don't cut your feet walking on them. So everybody was in a panic about them. Initially, we devised ways of suffocating them with rubber mats, and it worked, but nobody wanted to pay for it. And until we started getting the metaphyton, nobody really cared. Now it's suddenly, okay, maybe this is a problem. I think the solution that we're exploring anyway is, well, we can't get rid of them, we believe. Why not use them? So they're very effective at concentrating nutrients. So the excrement from the clams is about 10 to 100 times richer in nitrogen and phosphorus. So the idea we're playing around with is actually harvesting, sucking up the metaphyton from those areas. And then when it washes up on beaches, organizing citizen pickup things, using it as mulch, things like that. So yeah, as many people in this room know, invasive species are called that because once they're there, you can't get rid of them. But that's, yeah, we're trying to use their concentrating abilities for good.
Thank you very much, Jeff. Thank you. Part of this whole deal was a reciprocal visit to Lake Tahoe, and I volunteered to go to represent the group, okay? So, yeah, the winter, that's fine, we ski. All right, we got a opportunity for a 5-minute break here, and ladies have been gracious enough to supply us with some cookies and some nuts for an afternoon snack to keep your cells awake, because we got some pretty good interaction items coming up on the agenda here. So let's take a little break and a breather.
So I've been interacting with Evelyn a lot lately. Yeah, we because of the work with with the national park, I wanted to kind of link together with her to work on to harmonize our taxonomy for one thing. So I contacted her about that, and then she contacted me about a West Palm case where they're trying to build a road and then discharge into a wetland. So I've ended up being she's the major expert witness in it, but they've asked me to come in as, I think, icing on the cake, to deal with that. And then, apparently she's got some money from the poor to kind of do what I'm doing with the National Park Service, with the National Park Service, and developing a monitoring system that's a little tighter than the one that she's had before, and that's what I'm doing with the National Park Service. We're taking all the data that we've collected over the years and tweak it into metrics and stuff like that. So anyway, I'm just having the pleasure of dealing with that one. I had no idea that you.
Oh, yeah, I know. Yeah. So, did you ever know Joel Trixon?
Okay, I was sure.
Yeah, so I worked for Joel
Oh,
for 4.5 years, and I was the project manager for the SERP project.
So basically,
I would for 2.5 years straight. I would fly out all over from Lake Okeechobee down to the southern tip, and then it was always me and somebody from the Parapipe lab, from Evelyn's lab.
Okay. So, when were you there?
So I was there from 2007 to 2012. Yeah.
Sorry, I thought you were old.
Yeah. That's what everybody
So,
thinks.
no. So I was just thinking potentially you were because of the experience. Because Joel got there mid '90s, late '90s.
He had been there for a while.
So the project that I was involved with was with Kurt Richardson and Duke. And you may have heard the old stories about how heated the discussions were, and Ron Davis, right? So anyway, there was somebody at FIU that was kind of stirring the pot. That actually, I think really knew what he was talking about, but he shot from the hip.
Yeah.
So anyway, I Joel came in, and anyway, it was before Evelyn was down there, and they started the music assistant in the park.
I wondered, you guys had the samples of Higgins?
I only got one sample.
Oh, really?
Oh, the old sample?
Yeah. When we went out
years ago?
Yeah. Somebody was working at those communities, right? I wonder if they have similar compositions.
So I don't remember that stuff. So that was kind of a past project that I didn't confirm the taxonomy. Actually, it was Bo County sent the original samples here, and he missed. Everybody he was lumping it with something else. And there's just that level of taxonomy.
Yeah. You'd be
So
like,
I
why is this guy up
I got
here?
a sample from. I got a sample from somebody out of Higgins.
Justin Bragg. It was Justin and I were.
That's how that. I knew you were in that email somehow.
Yep, I just arranged for you and Justin to meet.
Oh, that's right. And I sent you a little report.
Yep.
So I looked at it just briefly, and it's got some of the same things, but it's not the crusty kind of stuff. But when remember when we were out there in the pontoon boat? No, it's almost died. The wind's coming up. And,
I do. Yes, I do.
Not what I expected.
I remember you said it was like mostly mucilage.
Well, there was a lot of mucilage, but some of the same species were in it.
Okay.
So the key is tipping it over and getting it crust. But you kind of got to go look for them, and even there, I mean, there's just all kinds of forms of stuff. And so I've got Peg's comment, and I've got Becky here.
Becky Norris? Yeah.
I've got her sample, pictures, tons of her pictures, and I've been kind of going through those just to get a broader sense. Now, nobody's talking to me about ribbons and stuff floating up that are chocolate brown.
Yeah.
Give me a picture.
Because I wonder if the same thing is happening in Higgins, except they have such a phosphorus input in the groundwater. They're constantly getting high phosphorus waters there. The whole groundwater, like all the way across, all of the samples except for up in that corner where they have the sewer treatment plant, all of that groundwater is high.
Remember that park? It was an old camp. It was on the north side that we were going by. It had a big shelf. The shelf was really interesting.
Yeah. So if they have a high density of.
That's right. We do have those samples. But hasn't anybody else looked at algae up there?
No, yeah. Well, because they don't care. They don't care yet. They're gonna care.
Yeah, but maybe it's not forming the crusts. Because if there's no phosphorus going in, one hypothesis
it is there. I know, that's what I was thinking.
Can we try to gather back to our seats so we can go on to the next item, please? I don't mean to be so rude, but. All right, we want to get back to our agenda items. If we could get back in our seats, please. Okay, I want to remind everybody we have a social event planned at the Torch Lake Cafe after the meeting. So we don't have to do all our socializing now. We can go back to the meeting. It is directly after this meeting that the room in the back of the cafe has been reserved starting at 4:30 for us. And Bob Cook, you are invited. Yes, we're not picking up your beer tab, but you're invited. Okay, so as you all know, we pressed everyone to take and put their vote up or survey results on the causal factors or hypotheses of GBA. And Dan's going to try to go through the results with us now and see if we can glean anything from those results.
Okay, folks, you got to take your seats. You got to stop talking. Was that clear enough? Yeah, okay. You don't need to think about what did he mean by that? Okay. So we're gonna go, and so we took the screen up. We need you guys to comment and gals to comment. We need your input. This is why you were invited from here on out. The talks were nice. We laughed at your jokes. The dinner was good, but this is where we need your input, big time. Everyone's input as well. This is kind of what we get paid for. So this was the vote. You saw the before. The question is, and I don't want you to do any analysis. I'll just suggest an analysis, and you can apply your own analysis. The analysis is of the results kind of is that there was, within the work group, there was some compression. There's more maybe at the top than there was a tail here. Let's not overanalyze it, but decreasing phosphorus in groundwater was at the top, non-native mussels, and it's climate change is hanging in there among the work group, and it's very interesting. There were 2 votes on non-native fish, and they were named, gobies. So we want to get your feedback on that. Then among the local advisors, and I'm just going to paraphrase this, and then we want to hear from you folks. Very interesting. You could say that they agree with the work group that most of the stuff is up at increasing phosphorus in groundwater. Non-native mussels are still being mentioned, This is after the talks and the conversation. I'm not saying anything's right or wrong. This is observing. And runoff from storms persists amongst that group, despite the fact it was 0 even before and 0 now among the work group. But in general, you have, if you're a risk management person and you look at distributions and you say these are potential hypotheses that may be legitimate causes, you're dealing with this from a mitigating risk, this would be called a fat tail. And if you've read books like Black Swan and stuff like that, usually the fat tail sometimes, I'm not saying it's applicable here, but it's reason to pay attention to stuff like that. Sometimes things in the fat tail of distributions is where a lot of stuff occurs that you don't anticipate. It's just, these are idle comments. So that's just thought. So now we need to hear from you folks. What do you, in the next, say, 20 minutes, what do you folks think is going on with golden brown benthic algae? And if you walked out of the room and talked to a reporter or a neighbor and said, what do you, tell me what you guys talked about all day and what do you think? You've got to think something. So what do we say? Go ahead. Dale, then Rick. I'm sorry, Rick, then Dale.
My view is I've always been very intrigued by Jan's hypotheses and this whole low phosphorus and its effect of, changing what the algae species are and how they visually appear at the bottom of the lake seems very plausible to me personally. So my view is we need to carry on more experiments, generate data to either validate or invalidate that hypothesis. I think that's something we need to do in fairly short order.
To say it differently, but no better, you would keep that hypothesis in the running and just look to continue to understand where it takes us. Got it. Tremendous.
I'm going to add something else out here. And if you looked at that, climate change was only by one person. You probably guess who that was. And the reason why I say that is, if you think about it, if you were out here right now, we were out there on the lake, and everybody said, wait till next month, it's going to get bad, right? Wait till next month, it's going to get bad. If you go back in climate change, there isn't much. I agree with Jan. It hasn't warmed much up in the summers. What it has done is shorten the winters. So right now, when you go out in July this year, that's like what it used to be in I have to think of which way I'm going, June. So what happened is everything has shifted a month. So everybody was complaining, if you came out, instead of coming out in July, if you came out in August, it would look worse, right? That's what's happening out there. That's part of it. So the winters have gotten shorter. That's the one thing you can agree with climate change. You can disagree with what happens in the summer, and I'm one of the strongest advocates because it was warmer in the 30s than it is today in the summer. The winters have gotten shorter. And so that's something to consider is that, and it's actually up there, it's the sort of algae in the news. There is part of this that it's just, even if nothing else had changed, because the summer started a month earlier, and that's how much things have changed now, is that algae has an extra month to grow. That's one. The other one I want to throw out there, and that's not even on here either, is acid rain. Okay. We talked about atmospheric deposition. Acid rain is a possibility in here. One of the things with acid rain is it has gone down. Acid rain is one of the big things they pushed for in the 80s, and they did do a lot of improvement on there. Okay, why would acid come into play? This is what I was talking about, that gray matter. We just changed the pH of what's coming into the lake. I don't know if the pH of the lake changed. If the pH of the lake changed, it could completely change this calcium carbonate that's precipitating. That's why when I was asking Jan about this gray matter, is that really what's causing problems? All that sand, if we had all the diatoms and they're hidden in there, maybe we wouldn't see it like we do in other lakes. But now it's stuck on top. We could do some cores, look to see if that calcium carbonate is precipitating more now than it did in the past. If it is, maybe we're adding a flock to this top and basically covering up the sand and giving a place for this stuff to grow. So it's something else to consider. I think we could do this with core analysis to go out there to see, do we get a lot more calcium carbonate precipitating than we used to? So a couple thoughts.
Norm had a question next that I want to get, and then this is your time to talk, not my time to talk, so.
Yeah, I don't want to embarrass anybody, but there were 2 votes for the Gobi from the experts. What's the, if you don't mind, explaining what the process is that the gobies would be impacting the golden brown algae? What's the mechanism there?
I signed it. Who's the other one? You want it? I can take it or you can take it, but I'll tell you my theory.
Tell us only when you get the microphone.
There's other things out there that eat algae off the bottom, and the gobies have gone through there. And my bet is, like, if it's anything like the Great Lakes, they completely changed the kind of fish and other things that are around the shoreline. And so things that used to probably eat algae, the gobies probably replaced them. And so what happens is that you lost, not so much, it's sort of an invasive, well, it is an invasive species. But what happens is that if you have an aquarium, by any chance? Okay. A lot of times if you have aquariums, you buy algae eaters. And the algae eaters will clean up your, so if you put a bunch of algae eaters out here, they could eat all that algae. You get rid of all your, now I just killed all of my algae eaters and put out something that doesn't eat algae. And so this is where I was thinking that it could be that you just got rid of all your algae eaters.
Can I just say one thing to that, please? One of the things that we are hoping to get today, and Dave is here, and he can clarify a little bit, but the DNR has made fish counts, including Torch Lake, and we've been waiting for a report that is still in work. But anecdotally, we have seen a shift of the smaller species fish that can't compete with gobies. Gobies are voracious eaters. They are very quick, and they have migrated the other small fish in that area to move out. Is that pretty valid, Dave? Would you like to add to that?
Yeah.
And you could use that microphone right in front of you.
Turn it on. Turn it on.
Yeah, I spoke with someone at the break. I'm kind of representing Heather here today, but my job is on Lake Michigan, so I can't speak to the status of that report actually, but we definitely have seen on Lake Michigan things like sculpins. There's no longer a fish that used to be the most abundant fish. It's called Johnny Darter. They're no longer in Lake Michigan at all. So they definitely, gobies can definitely displace some of these fish, and some of them are do eat algae. So I think I wasn't one of the ones who put the sticker up there, but it's a plausible. So
Somebody has said, I've heard comments.
Hold it, don't, unfortunately.
I've heard that when the gobies came in, all of a sudden we started seeing an increase in the algae. Around the 2010 area. I don't know. I mean, I don't have any data to support that.
yeah,
I've been told that the prevalence of gobies has sometime around that time period. So I don't know.
I don't have firsthand knowledge of what the survey on Torch Lake said, but we could put that graph together pretty easily to show what the goby numbers are. So yeah.
Right.
Yeah, I wasn't someone on the listed on fish, but this conversation has inspired me to think of maybe from the DNR standpoint, what would be interesting is they've had a very successful, at least a decade-long now program of Atlantic salmon being introduced. There's also some work being done on Elk Lake where they've stocked brown trout and can't find them. Now, maybe they were eaten, or maybe they're thinking they migrated to Torch for some reason. My question is there a way to assess whether we have more apex predators now than a decade ago? We know the program was successful because we have at least 2 commercial outfits now running charters on Torch.
And I think, again, that goes back to Heather. By the way, the reason why Heather's not here today is she is on Elk Lake today, working on a very important study that was not scheduled to interfere with today until we are missing that little.
But we can get those answers, hopefully, about just total fish population size, especially the apex predators. Have they increased in Torch or?
Well, she didn't share that conclusion with me, and unfortunately, the report should include that information when we do get it. Well, everybody has access to it anyway, but within this group, we will intend to share it with everyone that once we get it. And it's not that far
Well,
away.
No, and I would say too that in Torch and elk and these lakes here, lake trout are probably a lot bigger player than brown trout and Atlantics are. They're the fishery's important for those other species, but in terms of what the total population is, lake trout's probably pushing things mainly. And they do eat in Lake Michigan. They eat 50% of their diet's gobies, so they eat gobies also. But
So we're gonna go another 5 minutes or so, and then we're gonna take a shot at what does this mean. But Jen, like you're listening to this and you've been studying this for a couple more days than most of the folks in the room.
Decades.
What's your what comes to mind? Oops, actually, you can probably use that one. Okay. Figure out what the word, that'll make it better.
First, this whole process of gathering more data is really valuable. When I went down through those hypotheses, remember they all stand until, and more, stand until as we gather more information and test those hypotheses and then figure out what we can do about it, right? So the effect of gobies, for example, on Johnny darters and sculpins, I don't think Johnny darters and sculpins are eating algae too much on the bottom, but they are incredibly valuable bioturbators. I can tell you all kinds of stories about Johnny darters just bouncing around in a deeper part of a pool because they would stay in the deeper part so they wouldn't get eaten by birds. And the entire surrounding the edge of the pool had a nice crust of algae, but there was nothing in the deeper part of the pool because just the Johnny darters moving around on the surface, and they broke up that mat. So you can imagine how that would work. So the Johnny darter did the gobies get rid of enough Johnny darters, and this comes into the modeling and conceptually thinking about how that would work. So getting more data on that, providing evidence that that's possible, and then gathering more information that's the best example that I can think of how I've been going about the process and talking with other people about the process. Is here's a list of hypotheses. What do we know? So
Good, thanks.
I would just continue that process.
Okay, good.
Keeping them all in mind, gathering information.
Good, okay. Couple more comments, and then we're going to talk about what this means.
Thank you. Hi.
Please introduce yourself because
Yeah.
you came in a little late, you need to get introduced, and you're one of our favorites.
Oh, thank you. Thanks. That's very nice. I'm Angela Brennan. I'm with the U.S. Geological Survey. I was an author on the Silver Lake study that you guys have heard a little bit about. I recently took another job within the USGS Water. So I don't work in the Water Science Center anymore. So these guys are doing all the good work now. One thing I just wanted to, maybe it's more of an observation. I'm sorry I missed this morning. But I think as we move forward and we're looking at ranking and developing these hypotheses is understanding the likelihood of it being a combination, right? What is the likelihood that it's one thing? It's most likely going to be a combination. For instance, you see climate change and runoff from storms, and we know that those go hand in hand, right? As we're seeing more frequent heavy rains, right? And so runoff is becoming kind of a more significant part of like the nutrient loading in a lot of these systems. And the same with like the land use change, right? So climate change, land use, runoff from storms, those hypotheses have a lot of similarities among them. So just understanding that, you know, the likelihood of it being a combination, right, of these different things is very possible.
Thanks, Angela.
Yeah.
Maybe, Trish or Peg, with your backgrounds too, is anything you want to add to the discussion on what you see? You don't have to. These are just questions. No? Trish?
I just want to maybe say something about the tributaries, because I still voted for that because I know we haven't ever looked at our tributary, periphyton, and there are tannins that could be discoloring them so that they may not be looking like our golden brown algae. I would like to continue to rule out my feeling on tributaries by looking at some of the periphyton that is at the mouth of these tributaries.
Let's just check the, maybe one more comment from someone, then we're going to talk about what this means. Anybody have anything else oh, Dave, sorry.
Just one, yep, sorry. And this is probably me being a bit naive to the issue, but one thing that we always try to think about with research that we're doing is sort of what's the end point. So for the group on the lake, what's the main problem, I guess? And is it just sort of an aesthetic? Is it worry about the biology of the system? And where does that lead you then for some kind of management action down the road? And then that maybe helps inform a little bit about what you decide to study and.
So, and maybe I'll take a shot at answering that one. Yeah. You know, kind of being trained as a scientist engineer, but then in management, and I'd say none of that matters in this case. And maybe Peg can help me here. And Norm, and others that I've had prior, I think what really matters to a lot of folks is what's happening to the lake colors and the lake bottom, and what it looks like and what it feels like. And I don't mean to belittle, because like the science, believe me, I understand the value of science and spent a lot of time in my life in it, but I also have spent a lot of my life on Torch. And I think that's part of the framing of the issue outside of the room, whether we want to hear that or not. But I'm sorry, Norm, is that fair?
It is the aesthetics.
No, that's fine.
Yeah, okay.
We all know that, you know, this is not lethal. It's just disgusting. And, you know, it's that disgust compared to what we had, 10 years ago that's got our members upset. And they'd like to know how to fix it, obviously. There may not be a fix, but they would like to at least explore it. And, what we're proceeding to do here is lay out a plan on how we're going to do more testing. So if we can determine a cause, there may not be a solution, and we can tell them that. But right now, they want to know what is being done or what can be done to eliminate it. Not because it's making them sick or anything like that, but it just looks disgusting.
Yep.
It's not as bad as a rock snot, in my opinion.
Yeah.
I hope we don't get that.
That's good, Joe. I was not trying to belittle it or anything. I just, like for us, like an example.
It's been identified as our number one member issue.
Yep.
That's why we're spending all this time and effort and money to see what the cause is, if we can.
Maybe last comment, Trish, then we're going to wrap this.
I have a phone-in comment.
Oh, okay.
Becky Norris has asked me to state that perhaps GBA is the canary in the coal mine. I'm concerned about where this is going.
That was basically where I was coming from too. Is the comment do we know that this is not indicative of opening up the door of what's next? And if I could also quickly say that I've also noticed as third generation that as a child and a young man, I could stand in the lake and I would have minnows on my legs all day long. And I can't remember the last time I saw one.
Yeah. So another way of saying it is, we don't know what caused GBA, and we don't know where it's going. And you can restate this in various colorful ways. So let me I'm sorry. Good. Use your microphone. Yep.
I just wonder, moving forward, if I think that some of these issues are not in your control to change. And so ultimately, do you want to find out all of the, like, spend a bunch of resources to find out all of the causes, even though many of them may not be something you can do something about? Or do you want to focus on finding the ones that you can do something about? It's just a thought, so.
Call the strategy question. What's our strategy going to be here? And we're not going to answer it by talking about it longer, but we're going to talk about it a lot longer outside the room, I think. So let me just take a shot at kind of summarizing where this discussion has got to, and you folks see if this fits in some way. But let's just pretend to make the conversation go. Again, we all stepped outside of the room and we met a neighbor, or we walked into a meeting of 100 and some folks, like a lot of us will tomorrow, or we met a press reporter and they said, so what did you decide? So let me take a shot at that. Well, despite the best conversations and the best expertise we could pull together, we're still not sure what's causing it. We think we've studied a lot of things really well. We think we can study some of those things we've chosen to study so far with more depth. But there's some other interesting ideas we need to pursue before we say we're really close to nailing it. And like it or not, that's where we stand. Is that fair or is that off the mark? Would someone else like to propose another elevator speech to the press reporter that asks you what came out of the meeting on GBA? It's not bad or good. It's just what it is, right? We can't change that. Anybody else? Rick, Fred, Trish, Peg, Tom?
I guess the only comment I would make is, all the activity to, do a better job of understanding the nutrient balance and all that is good. And I think is important to, maybe be able to put in more early warning systems of what might be changing or how the lake is changing. I'm still failing to see a little bit of connection with causes of GBA, and that's what people are concerned about. We're also concerned about making sure that we have the effective monitoring in place. So how do we merge those two? How do we merge those two together a little bit better? I think is.
I don't think anybody can. Yeah, it's an open question. Yeah. Okay, got it. Okay, so I think again, it is what it is. If anything, this is one of those conversations that, rather than converging, has more of a tendency to diverge, and rather than wouldn't life be great if we had a meeting of all the experts, and out of the confusion that created the meeting, confusion of causes and that we all wondrously, like a math equation, a differential equation, we solved it. And there was a solution. Unfortunately, that is not the way things work. I'll leave the last word. Dale or Jeff, as outsiders, with that message of that, we've kind of got a lot of causes still on the table, perhaps more than we had when we even entered. Does that fit with you folks too? Or is that, as somebody outside in being more objective than us inside out, what do you think?
I'm not supposed to be an outsider anymore.
You have not gone native yet. Well, then maybe, Jeff, you're the only outsider.
Well, thank you for calling me an outsider. Thank you, that's nice. I guess my only caution would be, as you go through this process, would be not to paint yourself into a corner and invest so heavily in one or two or three hypotheses and the monitoring that goes with them, that if it turns out they're not it, then you don't have the data you may need to look at hypotheses that aren't here or other ones that didn't rise to the top. So I think that's where the wisdom and care needs to be taken in finding a monitoring program that covers enough and that is affordable at the same time. I'm not saying it's easy, but it takes a little bit of thinking outside the box that's being built right now.
Okay. I'm sorry.
Therefore, how do you design the monitoring?
Well, that's exactly the next topic. So we're going to hit that next. Defining the problem of GBA.
I think actually what touched on it is we have a dual purpose. We want to try to get the problem definition is dual. One is we don't know enough about GBA.
So defining the problem, one, we don't know enough about GBA to come to a definite conclusion. All right. And secondly, we don't know enough because we don't have long-term data. So the solution or the answer to that is continue, as Rick suggested, to test and examine and question the hypotheses that we do have. And in the meantime, structure a long-term program such that we can get the data that we need going forward, either to unravel the GBA question or, as Becky Norris's comment was perfect, the canary in the coal mine, okay? Fortunately, if we had all this long-term data, such as they had in Lake Tahoe, which was, to me, was terrific, okay? Perhaps we could catch the next change that comes up or better understand the progression of GBA. So does that capture the problem definition, Dave? That pretty well explained?
Yeah, I think so. My comment, I guess, was more, I don't know, immediate or just, you know, like, let's take plant control as an example.
Mm-hmm.
For me, as a research biologist, I'd be more interested in kind of why plants are there and, you know, what the dynamics are and how they got there. A homeowner might just want them out of there, and let's clear those things out, you know? So it seems like this group is really interested in understanding what's going on. So that's a big part of the goal here, which is good.
I think Norm put it correctly that if you're sitting on a board of any one of the three associations, that's the thing that the membership thinks about. The people that made the investment in the property, the people that are in homes that are generationally going to be passed forward, the people that do see these changes, they want those changes to stop. They want the clarity that we saw once before to return. So that's their perspective. And then as scientists, you could just simply say, well, if it's not hurting anything, it's just ugly. Well, just ugly is bad enough for the people that are on the lake. All right? So that's why we're here today. Did you want to say something? Because we all listen when you talk.
So I was just thinking about but I think you put the problem statement well, that the immediate problem, kind of what we're here today about, is GBA. And that's one reason to set up a monitoring and assessment program. But it was also, as in your line B, which is all the lakes need to be doing more than than Eagle can do. People of the state do not want to pay for that. You guys live around the lake. Unfortunately, you should be ponying up. And actually, I think if you work together as groups of lakes, for example, you know, there is just a lot of value in these lakes up here. And you get more bang for your buck in a co-op than you can run it by yourself. Now, you don't get to control it quite as much, but you get more bang for your buck running as a co-op.
Fred, don't you wish you was on the boat yesterday? We had exactly that conversation yesterday. We've got friends and associates that are in Elk Lake that are very much involved in their things. And you can't separate this chain of lakes. You can't, in a vacuum, study one lake. You have to have the study in the entire system.
Yeah, Dean organized a group from Traverse, from Glen, and then this is Walloon, it's Charlevoix. And I don't know about Douglas and that group up there, but.
Well, and to complicate things.
I haven't seen it that much.
We always have to interface with the governmental agencies, of which we have so many to deal with. Okay, are we ready for the next? Okay, so we're gonna drop the screen down, get it. I want to make sure I wasn't gonna get hit in the head. Dale, are you ready? Okay.
But that conversation went in circles, I'm telling you. I thought I was gonna be talking about something that made absolutely no sense, and then all of a sudden, Rick brought me home back to where it made sense, and then it went back again, so I'm not sure. But anyhow, I think there's two things that we're talking about. One, we're developing a monitoring program for the lake in general, and two, we're trying to do some scientific studies. And I think we need to keep doing both of those. But right now, I think there's a real shortness in this thing, and that really deals with the monitoring program. And when we look at a monitoring program,
Need more light.
When we look at a monitoring, we have some very specific goals. And the first one is to describe the current water quality, describe the changes in water quality, and describe the changes in the factors driving the water quality. And right now, I would argue that none of those are being met. And when I'm just saying describing water quality, we want to describe the spatial variability in water quality. Right now, there's primarily just 2 sites being monitored. And now we're starting to throw a little bit on the near shore, which I think really has to expand. But we really want to know what types of things we should be measuring at those sites, the number of sites. Can we just live with just those surface samples? Do they tell us really anything at all? And we want to describe that temporal variability in water quality, which is going to tell us how often to sample. Right now, there's a little bit going on with Secchi disk. We have a few measurements, but for the most part, we're living off of 2, maybe 3 measurements a year. And is that really sufficient? Jeff was talking about some of that variability that's out there. I don't think we'll ever have a monitoring program that'll pick that kind of stuff up. But I'm not so sure that we'll ever be able to pick up changes in water quality. Doug mentioned some of those things, and he started scratching off the top and scratching off the bottom. You can't live, a monitoring program can't just start throwing numbers out whenever they feel like it. Finally, we want to describe things at a sufficient resolution. And that's specifically what Doug was mentioning. He went through there and he threw everything off higher than 5. Ugh, that's scary. Okay, the goals of the monitoring program, again, describe the changes in water quality. What characteristics do we really want to evaluate? Is phosphorus, chlorophyll, and Secchi enough? I'd argue no. A lot of those things we're talking about, Jan mentioned nitrogen and phosphorus kind of things. There isn't even any nitrogen other than what tip of the mid is doing once every 3 years, twice. That's just not going to cut it to really understand what's going on. Where do we want to quantify those changes? A monitoring program really should be tied together with, if you have scientific studies, that's great, but we can't do the scientific studies everywhere. So what we want to do is to know where we want to quantify those changes, where we want to look for trends, and where we want to do those studies and try to tie those together as best we can. We want to know the magnitude of the changes. One of the things right now, and this is what I was talking with Aaron, is that you go out there and you get part of a national program, and they simply say you're less than a value. To me, that doesn't cut it. What it says is that you have to get a better lab. You have to, if that's the best they can do, we got to find a better lab, get the samples analyzed so we can get down to things where we can pick up those changes. It seemed like the most recent numbers, they were at least hitting the scales. The other ones, if Doug, remember the plots that Doug showed just had those yellow bars? That just means it's too low.
I just want to throw out that just know that lower detection limits often are more expensive.
I
And
know.
so I believe that's why the state lab, they don't go as low, which is why it's more affordable. So just be considering that.
No, one of the things that I should have thrown up front, Stan, said, don't worry about money right now. We'll worry about money next. All right.
Easy for him to say.
Yeah, well, no, it's all part of it. But if you go out there,
Let me just add something. So if we confound what we need with what we can afford, that's 2 separate conversations. There's what we need, and then there's a lens for what we can afford. Because we want to know what does it really take to get torch or any lake in a good state, then can we afford it? If we get those 2 mixed up, we're going to lose on one or both of those. So that time will come. We don't want to have the cost conversation right now.
if we went to Lake Superior, we went to Tahoe, we went to some of these places, and if that's all you have is less thans, yes, you'd pick it up when it comes out of the less than zone, but labs are out there. I would argue one of the things you could do right away with chlorophyll, you should never have a less than value for chlorophyll. It just means you didn't filter enough. You could still use the same lab. Don't filter 500 milliliters of water. Simple, move it up by an order of magnitude. Whatever it takes to try to get those numbers so we can get some values. But money is definitely an issue. I don't agree with that or disagree with that. But Lake Superior has a good monitoring program. Maybe that's where some of these samples have to go, depending on which ones you're looking at. You also want to describe the factors that are driving water quality. And this is where it can be really important. If you want to know the causes of it, can you really, it popped up, and I think Angela mentioned it, some of these big events coming in, the monitoring of the tributaries in the past, like this latest study I saw that had 3 samples during high flow events and 3 samples during low flow events. I don't know if any events were even hit. And so a lot of conclusions are made. We have to do it in a way that makes sense so that we're really getting things out of it. Not only in the lake, but also in the land use in the area. A good monitoring program doesn't just look at what's directly in terms of water quality. You have to look at some of the things that are changing the watershed, whether it's the number of houses around the lake, number of septic systems, land use. And finally, this is where the two sort of come together. Are we measuring anything that's trying to get to golden brown algae? Most of the things I'm going to talk about today, or right now, I shouldn't say today, but right now, are really the basic monitoring program. Once we can try to figure out which one of those things are, we can add some of these hypotheses in. Don't want to throw all your eggs in one basket, but you want to be able to go out there and sample some of those things, at least to be able to test those. And that's going to be where, at least I termed it as adaptive management in terms of this monitoring. We have some ideas. Some of those we can add to a monitoring program. Other ones we'll add in with time. Okay. So right now, we talked about this. Doug mentioned it. The monitoring program really consists of 2 sites out there. Tip of the Mid's going out there twice a year, sampling or twice every 3 years, sampling at 3 depths.
It's
And
actually
there's a number of things.
once every 3 years in the spring.
Once every 3 years. And sampling at 3 depths, which is really good, and adding to the constituent list. It's not just phosphorus, chlorophyll, and sec. Nitrogen pops in here. I think chlorides are in there. But there's a number of other things that could really be useful. And I'll talk about those in a second. So from that, we get some trend analyses going, sort of, kind of. That's just their data. Then you add in the data that's being done by volunteers. But that's just one little spot on the surface, chlorophyll on the surface. Like Jeff mentioned, that may have very little to do with what the chlorophyll really is in a lake like this. There are ways to do profiles and add a chlorophyll sensor to it. Might have to do a little work to try to get the two to correlate right, but there are ways to get that chlorophyll number throughout the water column and other things throughout the water column. So, but anyhow, with the way we are right now, we can put those two together. This is combining those 2 data sets. This is the earliest thing that Doug was talking about, but you have to really be careful with this. So like right here, this is Secchi depth, and it seems to be a trend in the data right now. So it looks like it's increasing. That's the same data. I'm looking at that and I'm going, I don't see a trend in there. Somehow something got messed up. Now, this is the scary part. One of the things is very often what people do is they take all the data and average it together to come up with those numbers. I never like to average all the data together. I'll at least average it by month and then average that together. Because what happens in sampling programs through time is very often you start adding more to different months, and so you start to get more points on a different point. So I never want to take all my data and average together. I always average it by month and then average the things together. So anyhow, things to, just ways that, I've seen this in the state of Wisconsin. They have all their data on something called a SWIMS website. They average all their water quality together. And the first thing I always get is, why doesn't Dale's numbers ever look like our numbers? They always show trends. Dale's never shows trends. It's because what happens is they start adding specific months in, and then they throw all the data together. So you have to be careful when you start to mix all that up. The other thing is this is the chlorophyll numbers that I think we're talking about. To me, that doesn't tell me anything. It will help Aaron. If you want to compare Torch Lake to a lake down in southern Michigan, great. This is it's down there. But for something like chlorophyll, the way they analyze that, it's all dependent on how much water you filter. Filter 10 times as much water and get it down there. You know, so there's ways to try to change your analysis a little bit to help. Okay. This was some data from, it's old data, so I don't want to say too much about it. But, you know, here's an analysis where they had some numbers. You look at those blue numbers, 15, 20, 25. You have to do it in a way that you don't throw numbers out. I'll never take a, you know, you have to be careful with that. You can play stats games and say it's 3 times the standard deviation and throw those numbers out. That's scary. That means you screwed up. That means that something wasn't right when you collected that sample. It could be very often. I was working in some programs. They bent over backwards to clean their equipment. They scrubbed that down every year. I saw those high numbers every spring. They didn't scrub them down right. And what happened was, is at that point, I think people learned from that, but they're using phosphorus soaps. And every year, the concentrations are coming out about 40 in the first one, and they tapered off every time. As soon as we found out what they're doing, cleaned it up. But with a lake like Torch Lake, it's going to push anything. You know, anything when you're down almost at the detection limit, says you have to be really, really careful with that. So, and Doug actually did a very good job with this. I was going to say this trend is garbage. Throw away the first 2 points. You don't have a trend. Doug did that already. So, one of the things that has come out in a few of the reports that I've seen, the first one is a GLEC trends assessment. Here's what they were saying for future monitoring, and this was written in 2018. They wanted standardized data collection. I completely agree with that. Field and laboratory quality control, that's a must in this lake, much more than a lot of other lakes. When you're down at the detection limits, you got to find a lab that can get down that low and with the quality control. Choose the parameters for a long-term trend. Right now, phosphorus, chlorophyll, and SECI. That is the basics. That was the first part of my first talk. That's what most people do, but it's not going to get you a lot of the answers. If you're going to run models like Jeff was talking about, or I was at the end, you need to know the other things that are going into it. And finally, ice cover. I thought that was really interesting because that goes back into my theory was that, now maybe that's not ice cover if there isn't much, but it's still the winter season. And it's when does summer take off? And so if summer takes off a month earlier, you know, and you can't really compare Junes now with Junes back then when everything started earlier. And then some of the stuff from the earlier GLEC modeling study, which comes into here, they're saying consistently monitor tributaries. And right now there's no monitoring on tributaries. Sample lake at least it says every 3 weeks. I would say probably a month, but somewhere in there. And sample for phytoplankton at discrete depths. So this is what's going on right now. What I would suggest, at least to start off, is to add the 2 monitor, you know, have your 2 deep sites, add in a couple near shore sites. Okay, so at least you'll be able to see. Maybe those will come out and say that they're all the same, but I think we should look at that first. This is actually one of my what I give to my field crew. This is a very basic one. This is the way we sample lakes, and it's, you know, I took the name off it. I probably shouldn't have. But what we do is we go out there, and if you look at the months, right now it's April, May, June, July, and actually this was the oxygen one where I sampled a couple times during specific months, August and September. And I would argue for this one, if you get somebody out all the way into November would be important. So you do temperature, oxygen profiles every time. In the springtime, first of all, start off with a detailed spring chemistry. The lake's turned over. This is where you pick up things like your chlorides, but especially like in a lake like this, things like calcium, magnesium, ammonia, or sodium, potassium, and so on. It's a very simple sample. The lake is well mixed. You send it off. It's not that expensive to do to get down to a lot of things that really tell you something. It may not be good for today, but who would have thought that chlorides were going to be important? A lot of my lakes, I can go back 30 years and tell you what the chloride concentrations and how much they increased. These are some real basic kind of things. Do that in the springtime. And then nutrients at selected depths throughout there. And right now, this was sort of top of the epilymnion, bottom of it in the thermocline, top of the metalimnion, and a few other date locations. But now when I say nutrients, I don't mean phosphorus. I'm talking about phosphorus, dissolved phosphorus, and the nitrogen species. And maybe silica also because of what's going on in this lake. But we can get a very detailed one. This is, I'm not saying this is the one that should be done forever, but to start off in the first year, There is a lab cost. I don't disagree with that. But this way you can get a lot of it. Have your master site on top. This could be the north side on the bottom and the southern side on the top. But this is a way to try to get out there and really describe what's happening in that lake. Okay? Now this is what we did in 2017 on this particular lake, and then we cut it back to this. So after we saw that we just didn't pick up much variability, we could cut a lot of that stuff back. The one thing here is, it's interesting, if you look at September, I have phosphorus all the way through the water column. That's because I was trying to see what's coming off the bottom. And in this lake, I'd probably move that off even into October, where you get the phosphorus all the way through there and see if the phosphorus is coming back. Not sure, Aaron, I think you were asking about what comes off the bottom. Right now, unless you go out there really late in the summer, you're probably not knowing what's coming off the bottom, if there is things. But if you do that the first year and see it doesn't come up at all, you see the oxygen going all the way to the bottom, you can adapt really quickly after you have that first year in. So what you can do with outcomes of a detailed monitoring program like this is, one, if you do have things that are coming in, these changes, I was talking about Delavan Lake, they went out there and they did all these things. You can see the impact on the water quality in these different aspects, whether it's phosphorus, nitrogen, and so forth. You can also see what's limiting things. Very often people are arguing Midwest lakes are limited by phosphorus, finding a lot more of them are limited by nitrogen. But the only way you can do that typically is to measure it and see what you get. The other thing is that I really think that monitoring the tributaries, it's one thing, like I said, that one study was looking at three events, low flow and high flow. I don't ever like to sample a stream in events unless I'm worried about the stream and not the lake. So I think what would be really helpful is to have the two, at least the inflow and the outflow, monitor those in a way that what I was talking about a little bit before, maybe add something like Spencer Creek on later, but put out some gauges so we can actually continuously measure flow. And for these other two, the first two sites, inflow and outflow, it's going to be a little bit trickier because they're all affected by backwater, so that would take acoustic velocity meters, a little bit more expensive, but that'd be really the only way. To go out there and chase these kind of things and take a few measurements, it's going to be really tough. Now, the interesting thing here is that I think you might get totally different results than what a lot of these past studies were going on. Past studies said that groundwater, remember the pie, it was 30, 30, 30? Well, I think that surface water was way underestimated. They had those three samples, they're probably all basically low flow samples, totally estimated what's really coming in from the tributaries. This is the one way to really test that. And then this could actually feed back into the models that have been developed in the past. Now, all of a sudden, we can figure out how much is really coming in. And if we put this into surface water, it may take away from groundwater. Talking about groundwater, though, right now it's estimated 30% is coming in. I think in addition to the monitoring, a decent groundwater model would be really helpful for this lake. So that's something to consider. Maybe not right away, but soon, trying to get to this. And if we go back to the lake I showed before, or the one I was showing where you have to know where the groundwater is coming in, put some piezometers in there, sample it to see what the phosphorus or nutrient concentrations are coming in with that. From there, the general pattern, they can zoom into some of the things that Jan was talking about and try to figure out where these things are occurring. So, like in this case, if you know where it's coming in, you can put those piezometers in there, figure out how much nutrients are coming in, and that might start to show why we're seeing the patterns in golden brown algae. I don't think so, but by developing a model like this, you can look to see where not only the amount coming in, or the concentrations, but also the amounts. And so it's maybe a little bit more. So some of these areas that look like they're really impacted, they may have a very short path, whereas some of these other areas that have a lot of development may actually have coming in all the way from agricultural areas and have higher concentrations. But when we're done with that, hopefully between the tributaries and other things, we'll be able to develop a nutrient budget for the lake. I know this was done as part of the earlier studies. This would be a refinement. So if you go back to the modeling study, I think it was 2006 that they're talking about. But this particular study, it was much different, where I had about 85% of the phosphorus coming in from the watershed. But once we go through with this, we can really parse that down. But it's only with a little bit better estimates rather than a simple equation to estimate one thing or another. And it's not necessarily that you have, meaning that you have to put out all these monitors, but we wanted to look at them in a much better way. And at least the ones that we have, do it in a way that makes sense. And it really is, rather than 3 samples and just say multiply that by, what would it be, 100 days. So what can we use the data once we all have it done? Well, we can determine load reductions, something like this. Now, if we had the model, we could set these simple models up really quickly. Looking at those, maybe this isn't the one where we want to look at what it would take to, what it would, reductions in loading would do. It might be the other side and convince people that we can't let the loads go up. Because this is what, we can use these and say, if the loads went up by 10%, 20%, this is what we'd expect our lake to look like. Okay. The other thing, and this is sort of where Jeff was coming in, is that some of these models, the only way we're going to get golden brown algae and understand that is probably to get into a more sophisticated model. And even if we have all these hypotheses, usually these models have all those in there already. Or if they don't, you have to include those in. But you can start off with a hydrodynamic model that Jeff was showing, try to get into the water quality part. Now, if one of the questions before was asked about Lake 2K, Lake 2K, the question was, why didn't they add golden brown algae? The real answer was, that model couldn't take it. You can't just add any kind of component to any kind of model. You have to have the basics of the model in order to add it. You need a model that has all the inputs, all of that, in a completely mixed model like Lake 2K. You can't just throw a piece onto it. You have to have the framework behind it. And so you have to have the right model. I'm not saying this is the right model, but we need to have one. Right now, there is a model like for Lake Ontario that's simulating cladophora and other things. Maybe that's the model we want to start off. But what we want to do to it is come back, get a golden brown algae component into it so that we can have all those basics in it. Somebody said something about you really can't put golden brown algae into it unless you know what's going on. That's true. You can't just throw a component on unless you really understand what's driving it. So we almost need the answer before we can, a model's not going to give us the answer. What we have to do is know the source, know that kind of stuff, then we incorporate it into a model. No model is going to say, oh, golden brown algae appeared if it wasn't in the model to begin with. You have to have it in there and have the drivers for it already known. So it gets a little bit circular in this. So anyhow, what I try to do is to show here is that this is the basic monitoring. It'd be nice to get it down, but that's not going to be the end-all be-all. We want the monitoring program, but we also want to have a research program, I think, on here. And the two should fit hand in hand, where this is the kind of stuff that Jan can hopefully live off of it in some ways. You have this as a background. When you're running these HEC-RAS models and these other models as part of it, it'd be great to have these parts of the watershed monitored and monitored well, so that you're not making a guess. Because a lot of times what happens is that if you say that surface water is down, but you still have to match the flow out of the lake, well, if you don't have as much flow coming in the surface water, you have to give it to groundwater. And so all of a sudden groundwater goes way up, and it may have nothing to do with it, just that you didn't give it enough surface water. So I think we want to have a good monitoring program, but I also think that's just half of what we're talking about here. We also want to try to get in a program where we can gradually test and evaluate some of these hypotheses. And so I see this as really as the monitoring, the overall program is really an adaptive monitoring program that's going to gradually evolve through time, whereas some will stay steady. These things, that's why I was saying you want to know what you want to compare through time, like phosphorus concentration. Some of these you want to measure it well, and don't change through time. But then you want to add pieces to it as you go on. So, but that's it, and I'll open it for any questions.
Yeah, Dale, I think I asked you this before, but if you could just describe a little bit for everyone your thoughts on the length of, sort of maybe a more intense monitoring program, and then, what kind of becomes the baseline? Are we talking like 2 or 3 years of, collecting maybe more intense data or a larger amount of data and then dropping back down to a baseline? What's a minimum time frame, and kind of what's the peaks and valleys of the monitoring activity?
I always like an evaluation over 3 years and then cut back just so you get a little bit of interannual variability in there. But I think even after the first year, you can cut some things back. I was saying that I wanted to measure these things all the way to the bottom. And, if you get down there, you might not want to go quite that detailed. Say you pick up oxygen all the way to the bottom, even in November, and you don't have no phosphorus. Well, I wouldn't want to throw out the bottom sample, but I could see dropping out a lot of the other ones in the water column. So I think you hit it really hard the first year, and you gradually come down into a long-term monitoring program.
In a 3-year period.
No, well,
What's long term?
long-term? How long have you been on the lake?
We need something quicker.
No, you don't. No, you
need something accurate.
That's the problem, is people come in and say, I want it tomorrow. And what you do is you end up jumping all over the place. You know, you hit a high point, you put all your money here.
But isn't it hard to sell if this was a 10 or 20-year program, which I understand really be nice to have those numbers. How can you sell a program like that?
So this is to get a handle enough of it, information to help GBA. So that's line 2A is to establish a monitoring program to understand GBA. Then there's a longer term monitoring plan just to kind of watch things and gather the amount of information so that the next time you get a GBA, you'll have all the data about gobies and zebra mussels and stuff like that that you need to really test hypotheses, which is what we lack. We just,
Exactly.
we don't have it.
Yeah, no, that's very good. That's exactly the way this is. But like this program here, when I started, or they started collecting data in 1984, I don't know, I don't think they ever thought they would be used for so many different kinds of things and looking at it. And one of the things that comes in, into, I look at this as one of the monitoring programs that even the states have all over the place, is that right now they typically measure phosphorus and dissolved phosphorus. And I monitor all this. I'm in charge of estimating how much loads gets into the Great Lakes. And the problem is that 25, 30 years ago, when I went to one of my first scientific conferences, they were saying what we really should measure is bioavailable phosphorus. And they said, oh, shoot, we can't do that. Somebody said it was too expensive, and somebody else said, well, it's too late now. And I'm now 30 years into it, and I'm going, why the heck couldn't they have changed that way back then? Because now 30 years, has passed. It would be great to have it. So really, this monitoring program would have been great to have. And I'm not saying back in 2005, I think, is when a lot of this stuff started. One of the reports I read was by Norris. What's his first name? Who was the president of this thing back in the 90s? Wasn't it Norris?
Jack Norris?
Jack Norris.
Yeah.
That was when I came in, and there was a report that said they were worried, and it came out that they were worried about this gray matter that was appearing.
In the 90s.
And, if you would have had this, so even the 90s was too late for this thing, I think.
I'm not consider, I know what you need long term. I'm trying to think of how we can do something like tomorrow. They're going to say, what do you guys do today? Well, we got to do this long term monitoring. Well, that isn't going to sell.
Yep.
We understand here to prove what's going on and what that means, and I agree a whole lot of part of it. I'm trying to think how we're going to sell it.
Well, I go back to exactly what Jan said, is that's only half of it. You need to have this, but, I agree that if everybody comes in like tomorrow, let's say, everybody is going to say golden brown algae, and I come in and talk about this, it is hard. Because what happens is that you really want to look at not just golden brown algae, that's today. What's the golden brown algae that comes in 5 years from now? You're going to have a different thing every 5 years. It could be chloride, it could be mercury, it could be this, and you're trying to come up with a monitoring program to not only look at today, but also to.
I'm just trying to.
Yep.
It's marketing. It is. It's a market. I'm trying to figure out how we're going to market this thing.
That's a tough one. Yep.
And it's going to start tomorrow, not next month. I know, that's, I guess,
Yeah, we're going to do it. We got it handled.
You asked earlier, how you going to talk to the press? What are you going to say? Well, what are we going to talk to all these?
We're going to tell them.
You got to have a long-term monitoring system, and we can go through all this. That ain't going to sell.
Okay. That's the.
Sorry.
No, I don't disagree with you.
Hi, I'm Kathy Shumrick, and as you probably all saw, I'm a physician,
And I'm listening to all this scientific data collection, monitoring, and I have similar concerns as Dave has. As far as treatment, what have you all done with lakes in the past that have had problems? What kind of treatments have you provided for these lakes and have had good outcomes? Granted, we have no idea what's causing the issue in Torch Lake at this point, but we have to have ways potentially to sell this to our membership that yes, there are treatments out there for lakes. And, you know, if we have this problem, then we treat it this way. If we have this problem, we can treat it this way. If it's going to be like there are no treatments, we don't do anything. We just let them keep going and progressing. I think we have to kind of have an idea, and that's just the doctor in me.
No, that you're the perfect person to ask the question. And I would say, do you get your blood pressure checked? Do you?
Do I?
Yeah.
Yeah.
Do you suggest other people do? Would you suggest that he gets his blood pressure checked?
Yes.
Okay.
No,
but that's what this is. These are the kind of things that we're trying to test and evaluate. We're trying to get the blood pressure of the lake. We're trying to get, it's not now, the blood pressure in a lot of ways is the ones that are routinely monitored. That's phosphorus, chlorophyll, and second. But what happens is that there's the other tests. And when you go to a doctor for so that's your sort of your annual. Every so often you go in for a little bit more in depth. You get your blood tested, right? Not just your blood pressure, but
we have treatments.
Right.
Do you guys have treatments?
If you look
for
on
these
here,
lakes?
this particular lake, they went through and they killed all the fish. They elmed the thing. They had a whole series of things going after each of those. Right now for golden brown algae, we don't have that. We don't have that because we don't understand it yet.
Right.
We got to understand it. So how can we treat cancer if we don't understand, you know, some of these things? And that's where it comes in. So we have to.
I think it's
yeah.
important for the membership to know that you have treated other lakes regardless.
But here's the problem.
Because if you're just data
Yeah.
collectors and don't
No,
do anything.
But the problem with this one is, it is unique. We don't have it.
No, I understand.
You can go in the
that.
literature, and we still don't have it.
I understand that.
So that's the hard
And
part.
it may
Yeah.
come out that we can't do anything about it.
Well,
Get
not
that.
saying we can't. We just can't do it yet.
Right.
But there are other lake problems
Right.
that have succumbed to solutions. So this
Yeah.
is one that
Okay.
hasn't been studied enough. So blue-green algae, for example, have been treated in other algae. Mercury contamination has been treated.
Okay. Good.
So there are other problems. Right now,
I need to hear this.
right now
I
we
think
concentrate.
We all have to realize that it's possible there may be no treatment for GBA. That's a possible outcome. And
Is it
we
a-
just have to have that.
Well, I think there's always
scenario
something,
available
but
so
the
that
question is,
people's
yeah.
expectations.
Well, here's one for you.
Right.
This particular lake, it was rated the second worst lake in the state of Wisconsin. What they decided to do is go out there and kill every fish in it. They wrote on the lake, they killed every fish in it. They restocked it. They applied elm across the surface at a couple $1 million to knock the phosphorus down. They went upstream and they dug wetlands upstream of it to try to trap phosphorus. So there are ways, it's just whether, you know, how much can you step in to do it? And then a lot of times what we're doing is trying to fight Mother Nature. You know, some of this is just natural. We're not even sure if this isn't natural, because one of the questions I asked right away was that, is this just a Torch Lake thing? Or is it a number of lakes? If it's a number of lakes, then it's even harder, because that means that everybody's getting the same kind of thing that's happening, at least in these really clear lakes.
Or easier.
And that makes it even harder to treat, because that means that it is something to do with more natural causes. Yeah, David?
Well, it seems, just following up on the suggestion about treatment, can we grow GBA in an aquarium and figure out what kills it?
I'll pass that on to somebody else to figure it out.
Yeah. Can I?
You're the expert, aren't you?
So there are treatments for all kinds of things. The question is, there's always a cost and a benefit to those treatments. So like let's say there's an invasive species and we want to kill that invasive species, so we're going to use herbicides. Well, those herbicides might be killing native plants too. They might actually be creating a substrate that's going to make it easier for invasive species to grow. So I would caution any thought of just some kind of silver bullet for golden brown algae, especially not knowing what causes it, because if we introduce something new to the system, that's just a whole nother variable that's out there. And I guess I don't really think it's that common. It kind of sounds like the stuff that Dale, you're talking about in Wisconsin. Like, there's not really large scale treatments of lakes in northern Michigan. I don't think that's a super common thing. And so
This isn't common in Wisconsin.
Okay. So I just want to say that, these case studies, that's probably not indicative of what should happen on Torch Lake. That would be very strange for this area.
Hi,
Yeah,
Steve
before you
Zinal.
go to that, one other
And
thing, just to follow up on that, is that, like, an example of what you're talking about is zebra mussels. And people bent over backwards trying to, what they decided to do was to try to prevent them from getting in because they can't stop them. I think they finally figured out a way to stop zebra mussels. It wasn't good because they got quagga mussels. And they sort of took over for the zebra mussels. And so in this case, the only way to solve the problem was to make it worse. And that's what I think you're talking about with some of these things, is that there are solutions to some of these, but you don't like the answer because it's actually something worse. Like, I could get rid of golden brown algae in this lake with no problem at all. All you have to do is get the farmers to put fertilizer on their fields big time, let it come on, and then you'll have the light problem. But that's not the answer you want. And, so there are ways to do it, but you don't want to make things worse.
Steve Zinal, and talking about the aquarium again, I talked to Dr. Stevenson during one of the breaks, and I said, had there been any, can you get an aquarium, put Torch Lake water in it, put the golden brown algae in it, and try to kill it, and maybe change the phosphate and nitrogen levels. And I think that maybe each scenario, if we think it's this ratio, what would we do if we found out that was right? If it was the gobies, what would be a possible solution to that? I mean, and that's what the doctor said about making a cure. I mean, certainly when you monitor blood pressure, I mean, if we couldn't do anything about the blood pressure, there wouldn't be any reason to monitor it. But we monitor it because we can do something. We can give them hypertensives, and there's different classes of hypertensives. So I think it's important really to think about solutions and to try to bring like, some tests or aquariums and change the phosphate nitrogen level, change, if it's chloride, change the chloride just to see.
I agree. And that's what I was saying, is that this is half of it. This is that long-term monitoring. And then we want to run the science experiments associated with it. So I think it's there's two parts to this. And I've never come in here, trying to stop the science. In fact, what I'm trying to do is to provide the background data that can help, help Jan along the way and try to address these things. Like that, if with a groundwater model, maybe that will help. I know you've taken a lot of samples out there, but this way would try to show the patterns and where it's coming from. It'd be really interesting if you came out here and it showed that it didn't quite jive with this until you put the land use upstream of it, where the water came from. It came through weird things and it was picking up. I don't think we'll see that. But that's where you try to put some of these together. So it's really, I'm not trying to stop the science in any way.
Yeah. No, so I would say we've never been separating those things. That's always in our minds, but we need to know the cause, right? I mean, that was why we went for groundwater early, in case we had that strong case that it's groundwater. Then you need to put septic systems in, you need to put sewers in. You need to stop those nutrients from going in. Then you get into Dale's answer that, well, it'd be 50 years, or actually the Heinemann lab used to model the legacy, share part of that, that it's going to take a while for that to happen, but you know that it will. So the two have never been pulled apart, but you can't constrain understanding the problem by, oh, we've only got 3 solutions to this. Engineers solve problems all the time. Once we know what the problem is, then we can start dreaming about ways to solve the problem. And all of this is kind of going on at the same time. So they're not, going on without any mental conversations going on.
I'd hate to see one of the answers is to add phosphorus to the lake.
Yeah.
I just couldn't imagine that, because you were saying it's because the phosphorus decreases. I hate to see that would be a solution.
Yeah.
Well, Lake Erie tried
you
that.
actually said it. I don't think I've ever said that.
But,
but everybody's thinking it, but imagine what would happen. Or you start allowing fertilizations of the lawn. Oh my God. You're just, yeah.
But that is one of the solutions for Lake Erie.
Do it.
They're
Unintended
talking about bringing
consequences.
barges out of
As
phosphorus
Carolyn.
and dumping it in. So they have talked about that. In fact, I think the fisheries people were pushing to barge phosphorus out and drop it into Lake Erie.
Yeah. It's crazy. Although they've got a very valuable fishery there.
It is. They got nice walleyes.
Actually, they use to fertilize lakes in Kentucky all the time for fisheries. Well, I think, Jan, you did touch on one thing here that I was thinking of before you touched on it, but this response to our physicians here. Yeah. It just seemed, I'm not trying to say anything specifically about, I have a great relationship with my doctor, but I noticed the two physicians tend to look at, well, they looked at treatments. They're thinking about drugs. But what about lifestyle? See, and so Jan mentioned the lifestyle changes. So it's not all drugs and treatments. It could be lifestyle. Lifestyle would be what he mentioned, fertilizing your lawn, septic systems versus municipal sewers.
Trees on the shoreline.
Those type of things. So there's that also. On the other point about, I am definitely not into geoengineering. I think that's always more risk than possible gain. But suppose we found out that these golden brown algae are there predominantly due to their enzymology, and that could be disrupted in a benign way. But we wouldn't know that if we didn't do this in-depth research. So the in-depth research is fine. The monitoring side, though, I do have a little difference of opinion because if we get all this nuanced monitoring, Dan said it, he said, we don't want to worry about how we pay for it because that'll constrain us. We want to find out what we really need and then worry about that sequentially. But I do differ that there's another bullet point there about what can be done about it that should at least be dictating our priority of where we put our resources. So if we get a more nuanced monitoring program, a more sensitive monitoring program, we now have to establish the action thresholds, and we have to think about what are the actions. I can think about 3 to 5 impactful regulatory changes. That's 3 to 5 conceivable ones. I can maybe think of 1 or 2 politically viable ones. So if you work a little bit backwards and prioritize those measurements and studies that relate most closely to a politically viable action, management action, I think you've better spent your resources. So if it's septic systems we're concerned about, maybe we should be studying with boron tracers the movement of riparian septic effluent into lakes and understand that better. Because what we find out, we have an action plan that we can follow. So you need a balance between both. But, money I can see, let's first find out and then worry about how we raise the money. But from the standpoint of this comprehensive monitoring plan, I think it would be worth thinking a little bit about what the conceivable action plans and action thresholds would be associated with that monitoring. Okay.
Fred, that was the point I was trying to make in that comment I made before Dale started the presentation about the proposed monitoring, because you do have a membership to answer to, and they want to know what what are you doing about the problem, right? So if you don't know what you're doing about the problem for, however long, that's not going to probably be a very satisfactory answer, right? So it goes to those hypotheses. So there are some of those that you can do something about, and there are some that you cannot. So it seems to me that you would want to focus your resources on the monitoring that is going to lead to is, groundwater an issue? And is it because of failing septics? You can do something about failing septic systems. Is it because of increased stormwater runoff from the tributaries? So I think the tributary and the groundwater monitoring is, in my opinion, very important, because you can also address increased stormwater by implementing buffer strips or whatever it might be. So that was why I made that point before Dan said, don't think about money being an issue. That my mind always goes to resources. And I think that's why I made that suggestion, because I do think the long-term monitoring and research is very important, but it's not going to get you what you need tomorrow or a year from now or 2 years from now.
So
That was the point I was trying to make earlier, before the money is not an issue.
No. So let me help with, I have a comment on the money real quick, then a separate comment. They're comments, they're observations, they're not recommendations or anything. But so the reason for, and I think Fred and I are in violent agreement on that, to separate the conversations on what the lake needs and what we can afford, is so that we don't confuse the two and run the risk of what I'm about to describe happens. So people have studied systems separately. There's a whole systems engineering discipline. And one of the great ways to get breakthroughs is to be interdisciplinary about how you think about things. And so lakes are a system. Limnology, a system. It's a specific study of a lake as a system. Well, people have studied social systems. They've studied safety systems. They've studied business as a system. And when you get into that kind of way of thinking, there's things called vicious cycles and virtuous cycles. So a virtuous cycle is, you could say, the country. When a country develops a good political system, it elects really good people who continue a culture, and the country spirals up into a virtuous cycle. When a lake does really good things, and I think Tahoe may be in that virtuous cycle, in the face of everything making it vicious, it develops a sequence of actions that spiral it up in a good way. So let me describe a vicious cycle. We don't understand the cause of something. We don't understand it because we don't have the data. We can't get the data because we don't have the money. So we don't understand the cause of something. We don't know what to do. We don't have the money to get, that's exactly how the lakes get in trouble. And so you get in this vicious cycle of which we inherited, and regrettably, we can't solve it with marketing problems. I mean, marketing solutions tomorrow, but some of what we're doing is setting things up for our grandchildren. And unless because the lake doesn't operate tomorrow, the next day, and the lake doesn't recognize costs. And so we need to think about that as a bigger system and decide how we want to intervene in that system. That's just an observation. Otherwise, we're subject to this either creating, but probably not living long enough to see, a virtuous cycle, or just throwing our hands up and saying, this is a vicious cycle. We never had the money, we never had the time, we never had the resources, but the lake doesn't care about any of those things. The lake's going to do what it's going to do in its time frame and as it responds. And if our answer to folks is, that's the way it is, then we should just tell them that. We don't have the money and we don't have the resources. That's just a perspective.
Let me add one last thing in here. 90% of the problems that come in lakes usually deal with nutrients one way or the other. And that's, coming in, Jan's trying to track it down. And so, it's almost always, whether it's today or 10 years from now, it's going to probably come back down to this, which is where are the nutrients coming from, and getting a good handle on this. And whether you like the results of the pie or not, it's good to have one. I did one of these in northwestern Wisconsin, and it came down to, like, this one is for in the middle of an agricultural area. 85% is coming in from basically agriculture. And so they had to convince the farmers to change things, which they weren't going to win with anyhow. But that was that particular system. The other one I was dealing with in northwestern Wisconsin, it came down to two things. It came down to precipitation and septic fields. That's all they had. It was a seepage lake, one of the best lakes in Wisconsin. And that's what it came down to. Now you got to turn around and say, okay, what can we do if we really want to fix things? Got to get rid of all the septic fields and put everybody on holding tanks. Nobody liked that answer. But it came down to, do you want the lake to gradually get worse? Or do you want to spend some money? And that's where it gets into it. But what it came down to is having that information first and then making the decision. And it's not easy a lot of times because, and I was trying to convince the woman, which is actually sort of the Tom at that place. And I said, what you really want to do is talk to people and tell them to stop using detergents that have phosphorus in it. And she didn't like the answer because she didn't get enough suds in her hair when she took her showers. And I'm going, well, you can't have everything. And sometimes you got to, once you know where things are coming from, then you can start to make decisions. Whether it's more important to have suds in your hair or have algae in the water. So, but it's when you have this information, then you can deal with it. And I'm not saying nutrients is necessarily the answer. That's only part of the ones that were up there. It could be something that it's acid rain. And that's where the gray stuff came from, and then it's a series of things. But we want to have those science studies to go along with it.
Hey, Dale, I wonder, we talked about salts, and somebody mentioned road salts was way up there in terms of a possible cause for the increase in chloride levels. Have you ever done anything with, most of our people on the lakes have individual septics, but they also have wells, and they have water softeners.
Yeah, I don't
And
think it's a road salt problem. I've seen lake after lake in areas that is a road salt problem. I don't think this is a road salt. There's no road. There's not enough roads there to do it. You can do a mass balance.
But have you ever run into an issue with salts from water softeners? I mean,
Oh, yeah.
people who have them buy usually 1 or 2 of those bags every month. And so that goes into the ground and
No,
it probably gets arrested.
I've seen some studies. I don't know if they've separated the two apart, but whenever they've said road salt, it depends on, there's some lakes, like there's a lake in northern Wisconsin that goes down by a highway and there's very few cottages on it. And there's a major highway. Now that's probably all road salt. But then you get a lake like this, and I just have a hard time imagining.
Where else could it come from? Do you have any other
Besides road salt and septic?
Water softeners? Yeah.
No, I don't have any other sources. Not if it's increasing. The baseline maybe, but not that it would be increasing.
Chloride's going up.
Yeah, oh, it is.
But not enough to cause this kind of change
Right.
in the algae.
Yeah. No, but
that's-
chloride is going up. What's the cause of it? It's one or the other. But you can do a mass balance.
It's really a comment, not to change the subject too much. We flew around Torch and we looked at the golden brown algae and we sort of looked at the inlets and stuff like that. And one of the things I noticed that was, to me, it seemed the worst. And I know your data is great and stuff like that, but we looked over at Wilkinson Creek and it was just terrible over there. I mean, the color, just really went up, really was acute. And so then what we did is we gained some altitude. We went over the beautiful woods, and right when we got to the other side, we saw cows. And we saw cows in the water in the creek. And we saw more cows and more cows. Now, my undergraduate was in zoology, and my graduate was in biology before I went on to medical school. But one thing I remember is one cow equals 26 people. And so I think one of the solutions would be if we try to obtain, we don't have to get 10,000 acres or anything, but one thing we could do is get some acreage or get some land on either side and plant something besides cows.
Yeah, you could do that, or the other thing would be to look at the, sort of the solution or the outcome of it, which have been higher nutrient concentrations. And, what I wonder a little bit with that is it and Jan could probably go in and count, and actually see if it's the algae that's causing it, or is it they stirring up stuff that's settling on top of the gray stuff? So is it really golden brown algae there, or is it other things settling? And that's where it takes somebody to go in and see, to actually measure those and see, is that golden brown algae, or is it stuff sitting on top of the gray stuff? But yeah, because the other thing is we're down at Spencer and you can see this, there's a big browner area there. And I'm not saying that's golden brown algae. It was actually added to the spectrum of colors. It was pretty cool. But I think a lot of that was just the tannic acids coming in, and it was just sustained water at that point. It would look probably from the surface just like golden brown algae because it's a darker area. But I think there's a lot of things that are hidden, and that's where it takes somebody like to go in and say, that's not really golden brown algae. That's sediment sitting on top of it. Over here. Oh, no. You know, my bet is that that's probably your highest phosphorus concentrations coming in. But Jan would like that. He wants higher.
Cows in the stream? Cows in the stream are illegal.
It is illegal there to have them in streams too. So.
You commented about Spencer Creek. There used to be a sawmill there, and there's sawdust on the bottom there. It's been there since late 1800s, and it hasn't gone away.
Yeah, usually you see oxygen problems, but I don't think that's going to lead to anything. Usually you have sawdust still in the bottom that leads to oxygen problems.
Yeah.
I don't think that's leading
It's
to
just,
the
it's just,
water.
it's constantly brown on the bottom there, and it always gets stirred up from the water. I grew up there.
Okay, I wouldn't think that would be causing the tannic acid. I still think that's,
No,
I'm not saying
no,
it's
that,
not there.
you know, there's obviously tannic acid coming down from Spencer Creek, but everything's getting stirred up because the town dock no longer is porous like it was back in the 1800s. And water used to exchange from the both sides, and it doesn't go. So my quick point was to add to something that Jan had pointed out earlier, and I was consistent with some of the feelings I had with some other folks. There are many lakes in northern Michigan that have had this particular issue. So in one aspect, when we were talking about the resources in terms of being able to finance the studies that need to be done, many lakes can benefit from the same research, and there's a potential of collaboration in order to be able to take this on, as well as be able to compare with those other lakes as to what is similar. If all these things happened approximately at the same time, what was the similarities? That's the engineer point of me, is in trying to figure out the analysis of what the problem is, what is the same by all of those lakes, so that the research done at Torch Lake can be compared to the research that's done at Crystal or one of the other lakes that you've mentioned that has the similar problems. So we have a potential of combining financial resources as well as more data that can be compared by all the different lakes that are experiencing the same problem.
That was exactly the reason why I think we have this other group that's outside of Torch Lake. That's right. I saw this meeting as sort of starting off with Torch Lake specific, and then trying to say, is Torch Lake really an outlier, or is it just why we're here? And you got all these other lakes that are really experiencing the same thing. Or, you know, and that's where you can look to see, like if you could almost draw a line around this and say, these are the ones that have something. You know, are these the ones that all happen to have zebra mussels that came in, and these are the ones that didn't for some reason? Or is it these are the ones that have high density septic fields and these don't? But that's where I really think that the regional kind of thing, it's really important to have that view of it. So you're not just thinking that you're the outlier, you're the unusual one.
Okay, got one more?
I'd just like to respond to Kathy's comments about treatment. The monitoring program that Dale is suggesting is focused on water. But as you were asking, what do we have control over? What can we manage? What can we do? And we don't have data about the septic systems in our watershed. We have 4,500 properties in our watershed. We don't have any data on the septic systems. We don't have any data on the lawns being fertilized. We do have data from Tip of the Mitt and the Watershed Center's shoreline survey that we've lost 30% of our buffer, our natural buffer along the lake. We have over 100 operating farms in our watershed, in our little watershed of 75 square miles. So we have no data about what they're growing, what they're applying to their fields. There's a lot of data that we need and monitoring that we need about the things that we can control, that we can make changes about. And so when we're talking about long-term monitoring, I'd like us to consider more than just water quality parameters. The organisms in the lake that we've made reference to that aren't there anymore, that because of the low oxygen levels, because of the high water temperatures in the summer that have changed, you know, the lake is going through changes. GBA is just one symptom. There are biological changes that we have not monitored. The crayfish, for example, which according to the DNR is a keystone species in the lake. You know, we haven't surveyed the minnows. We haven't, you know, the mud puppies. There are a lot of things, biological indicators, besides the loons that are no longer nesting there. But I think we need to look at other parameters, other kinds of monitoring, not just water quality monitoring.
Oh, I completely agree, and that's where a lot of the other agencies come in. Most of the lakes that I work with, you know, the more in-depth ones, I work directly with Wisconsin DNR because they're the ones who monitor the fish, and they do routine fish surveys to try to give me an idea. I don't expect a population, but if you can see the trends in the fishery, very often that can be important, and that's where that whole top-down idea comes into play. So I agree. And the other thing with that fish, if you looked at that monitoring, the last aspect was other factors that I, you know.
You can use your microphone right there.
Yeah, so just quickly, Peg's comment about bioassessment. I missed part of your talk because I had to go up for a call.
That's
But
what I was
did you-
talking about, the biology.
Yeah, so first it's important to just to kind of know what you have in your system. And if you count on the Eagle to do it or the DNR, they do not have the intensity of information that you need. You need to kind of partner up with other groups to get more frequent information and more extensive information. So for the phytoplankton, the benthos, and you don't have to do that all the time. So to keep the expenses down, you go to this kind of targeted system and a lower frequency of sampling. But biological assessment not only tells you what is there, but it also integrates the physical chemical conditions. And that's why the EPA uses biological assessment so extensively in their ecological assessments. So I just wanted to add that other complement and support the idea of physical, chemical, biological assessment.
Okay, I thought that was a very, very insightful discussion, and I really appreciate it. In fact, we've kind of merged into our next topic, which is fine, and that's why I didn't do a time check. What we have left is about 20 minutes. And what I have on the agenda is questions from all attendees and subject matter experts, which I feel we just went through. And then we wanted to try to wrap up our conclusions of the meeting and our action plans, largely so that we all have at least acknowledged agreement on what we've accomplished today. So I'd like to start first by taking a look at our objectives, and I guess they got moved over here somewhere. Oh, here they are. So the first objective was to evaluate the causal hypotheses of GBA formation, select top three, define, to prove or disprove, and validate. We've had a lot of discussions. We had a very interesting summary of it, okay? And I feel that we've accomplished not so much ranking or having definitive actions, but we've accomplished a very sound understanding of our current state. As we've all said, maybe we're not satisfied with that. Maybe it's not where we would like to be, but I think we're a lot closer than we were yesterday. And if nothing else, I think we've had a lot more discussion to drive us to ask more questions and to pursue more answers. which may seem like more work, but I think that we're getting closer to where we want to be. And that's good for me. I feel like we've accomplished that. All right. And I don't know about anybody else, but I'm more energized than I ever have been to take and pursue some experimentation, some more data collection, some more thought process. As Dr. Stevenson has said so many times, we have to establish a relationship. We have to either validate it or invalidate it with experimentation and data. And that's the process. It's that simple. It may take a while, but that is the scientific process. The second objective was to discuss and here it is, work on the formation of a long-term, it's right here. It's right here, okay. Establish long-term monitoring and assessment plan for Torch Lake based on the straw man proposal that was in the packet. I don't know, but I think that we really got a lot of good input today so that the USGS team can take all that input and that from everyone that's been here today and reduce that to a plan. Do you agree? We have a lot more than we had a couple days ago, right? And I think that some of the things that we have talked about are still valid of what we need to present for a long-term monitoring plan. I think we've got some additional ones. I'm pretty excited about that, and I think Dave had a very good point. We may not be able to do an easy salesmanship job tomorrow with the membership because they would like quick answers. But if I listen to Dan, if we wait till next year or wait till the year after that or wait till the year after that, it just goes down one more generation before we get close. So now is the time to act. We're at the point, I think, at least from my perspective, take this information and formulate it into a plan, the plan that will work to get us the data we want, and then go find a way to do it. And anytime that Dale feels like he needs more input to put together that plan, he's going to call me, and I'll go after one of you, and we'll go get it. So I feel real good about meeting that objective. So action plans. Fred, you did a terrific job of identifying some things that we need to take a look at, and that is those regulatory issues and put those on a priority list. So we got science, we got marketing, and then we got the interface with regulatory issues. If we keep those in mind, I think that will help us with our prioritization.
I'd like to say, our strength as an organization may not be science, since we're all just riparians trying to care for the lake, but we do have the strength of volunteer and of volunteer efforts. As we expand the number of organizations involved, we get more volunteers. They're hard to come by, but in aggregate, we have them. And I'll give you an example. 7 years ago, a big push in
better come
Milton
up.
Township.
Come on up.
There was a big push in Milton Township for septic inspection. And that was successful. But that is now a regulation for the point of sale of properties throughout the township to have the septic inspected. But I feel like that group that did that kind of said, wow, we got our job done, and they're gone. Well, one of the justifications during all the arguments, because there was a lot of pushback from the community due to costs, one of the arguments was, we're doing it for education so we can learn. To date, I have not seen or heard from any group that has looked through 7 years of inspection data. There's a report by the sanitarian every year to the township and said, what has this shown us? And it has some important lessons. So I think we should form some subgroups of volunteers within our organizations and task them specifically. We brought up today the fishery question. It seems awfully relevant to me right now. What is the impact of the successful stocking program of Atlantic salmon in Torch Lake? Is it really dominated by lake trout and we've added another 10% of fish volume? Or are the numbers different than that? Is there even a way to get that data? And then what could that mean? Again, Volunteers could do that. We don't need our experts tied up, but we need a small group, 1, 2 people, who over the course of the next year follow that up. And then when we get all together again, we'll have some answers about that. And those are just 2 of the things. I mean, you probably could think
Yeah,
of a dozen.
and, you know, I can envision a corporate structure of committees and subcommittees to collect data, report, analyze everything else. One of our problems, Fred, and I share that with you and a couple other ones, I don't know if what my expiration date is, but some of us are getting older, all right? And that's a difficulty is to bring up the next generation of volunteers. We got to wait till they retire, and then they got to have the interest in it. Because we certainly need the volunteers to have the time available to do it. This is a time-intensive activity. So until we have some of these young students that are back here get involved and have some time, the volunteer ranks are difficult to fill. So we're going to do a balance somehow. We're going to do as much as we can with the professionals, somehow cover the costs, and supplement that with the volunteers. I'm with you. We are constantly, Norma, I think, stepped out, but we discuss every one of our board meetings. How do we go get more help, more volunteers? Okay. We dragged Dan in. He didn't know what was coming to him, but he's been a very valuable asset to our board and been very helpful. So that's an excellent point. And I think that what's important is that we don't give up. We don't push off, because like I said, I got an expiration date. I don't know how long it's going to be. We don't push off. We get as much done as we can that's reasonable, accountable, and productive. So that's my take on this. I'd really like to hear anybody else's take on the day. Peg, got anything else to add?
I'm just glad everybody came, and I hope the conversation continues.
Well, the turnout today has been terrific. I really thank everybody. I want to give you a round of applause, because you sat here all day long. It is terrific. And I want to remind you that, you know, it's going on 4 o'clock. Our room will be started at 4:30 at the Torch Lake Cafe, the back room. And I hope to see all of you there. And some of you are going to, I know, run home and get your spouses or best friends or whatever you're going to bring back. And so we can carry on the casual conversation. We had enough science maybe for one day, but we're open to that too. Oh, is that a take-home package, the apples and the cookies? All right. Bob, Jeannie?
Give us a minute. We probably have something here.
I think you had something, right? Do you have a takeaway? No, you're already done.
No. I think I'm done. I'm hoping that I could hand this to somebody else. So far, no take.
One question.
Certainly.
Sewers, when they put the inspections in, did that help the analysis of the lake?
Not to my knowledge. I don't know that anyone ever followed it up, or there would be a conceivable impact. Yeah. But there was a lot learned about the condition of our septics in Milton Township. No one, I think, even went through there and took the time, because the health department's not going to do it, and looked at which one of those inspections were lakefront or within 1,000 feet of the lake, and which were elsewhere in Milton. Now, Milton has a lot of frontage, so a lot of them will be lakeshore. But it was what they call a mortgage inspection. So there's a lot of check boxes on how the system compares to current code. Mining that data is information. It was one of the reasons the inspections were instituted. But I might be misspeaking here, but I don't believe any of the lake associations have taken the time to go back and mine that data to understand what did we learn about septic systems on our lakes.
Takes a lot of volunteers.
Can I add one thing else? Think about how septic systems work, and think about what they do for nutrients. Very little.
They do nothing for nutrients. The soil that they're on, the inspection is only the physical installed system, and it's designed not to need to be inspected or to have access for inspection. So there's lessons learned there. The soils that they're put on
are supposed to have at least 4 foot of separation. Now, most of the nitrogen and the bacterial action is in the first few inches, the aerobic zone below the headers is where this happens. But I mean, this thing explodes into 1,000 different conversations. You have very expensive homes on Torch Lake, $9 million homes like we saw yesterday. And I'll bet you that home has the minimum code septic system. The homeowner has never even been given the choice of.
What I'm saying though is that
Yeah.
it had the best septic system. Think about what that's for phosphorus.
Well, the best septic system could have an active iron sand filter, and it would do everything. Okay, so, okay, that's a code-related issue. Basically, the model of the septic system, you know, we talk a lot about a failed septic system. And those people that are talking about failure, if you look at the Milton Township data, you will see that there are no condemnable systems that we're running into, almost zero, even if they don't have a permit. That's information. The mode of failure, the only one that's any different than the header of the distribution field is if you had effluent come to the surface and it could run off into the lake. Otherwise, it's all going through the ground anyway. I've looked at some of the stuff in some of the permits in my area where you see mound system. That means that the groundwater is high. I've looked at 2 permits, 70 feet apart, installed at least a decade apart. One says maximum high water table 6 feet from natural ground surface. The other permit says 18 inches. Okay, that makes no physical sense. 70 feet apart on the same topography. Yeah. So that's an issue with the opaqueness of the sanitarian's determination of where the soil modeling is and what is truly high water table. So, I mean, the more you look, it explodes into a million actions. It's not so, it's not just, also I'd like to say, it's not just an issue of we either have septic systems or we don't have them. Or if we have them, we only have to find the failed ones. Because I would posit that the failed systems are so few, 10% maybe at any given time. If there's a loading, it's from the normal, proper operation of all those systems. So you're not just talking about finding just the failed ones. I mean, those may or may not be accurate conclusions, but we have a wealth of data in Milton Township to get started, you know, on getting some insight. So I would suggest a subgroup for that. I'll give you another example. I don't think any of the lake associations know where the municipal water is discharged to our Urkel surface water. And it is Bel Air, Central Lake, and Ellsworth. So they have a point of discharge. They have a discharge permit. Might make sense to at least know what how many millions of gallons and what the phosphorus levels are. Might make sense. So we can weigh it against other things we know. But I don't believe anyone's done that.
I think my takeaway from this discussion, and it's one that goes on forever, is the better job we do from a science standpoint through data collection and modeling, the better job that we have to justify going forward to the regulatories, to the townships, to the counties, to the health department. It says, here's data that says this is what's happening with septic systems influencing our lake. Right now, everything else is just speculation. So it goes back, I think, to our discussion on the long-term monitoring program. It goes back to our discussion on understanding the causal effects in relationships. Because I believe that one of the things you looked at was groundwater, and could it be an influence from septic systems causing GBA, right? Yep. So you can't go forward and try to push the needle because of an assumption septic tanks are contributing to our problems. But it sure would help if we had actual data. And I really think that that's part of the overall monitoring program and the modeling, right? If there is a problem of influence in the lake from septic systems, we should be able to quantify that through modeling.
But that's where you put that big pie together and try to see
Right.
how big is the other pieces.
Yep.
And you make the assumptions on the septic.
But even the lake, your bathtub model, right? So the assumption on, what was that? Not Green Lake.
The Silver Lake. Silver Lake use a bathtub
Silver
model.
Lake, okay, so you've got a very good daily loading number or average yearly loading, whichever it was. and you knew how much of it was coming from groundwater. The assumption then was that I believe the way I read it, the difference between surface water phosphorus concentrations and groundwater concentrations, with the groundwater concentration being higher, half that difference was through the septic system. But, was that an empirical conclusion, or is that just, the best guess? I mean, that's all you're hanging on with modeling. You got to come up with some septic transport model. And I don't know where that data comes from, who does those physical tests. I will tell you one thing on septic systems. Again, what Dale just brought up earlier, the septic system does nothing for nutrients. Basically, a little bit of phosphorus settles in the tank and becomes a sludge. The rest goes through the distribution header and into the soil. And it's the adsorption reactions with minerals in the soil that bind the phosphorus. And according to the health department, with 4 foot of our normal soil here and the proper area of field for the house, no phosphorus forever will get down to the water table and start migrating laterally. That's the assumption. Reality may be a little different. But here's the interesting point. And this is almost counterintuitive. So the larger the house and the more bedrooms, the larger the field area has to be.
Right, so he goes by bedroom
Okay.
size.
But if the soil's all sand and drains well, the field can be smaller, right? If the
That's
soil
counterintuitive.
doesn't drain well, then the field has to be bigger. So you will see in the health code where sand, which has a higher perk rate, will give you a lower per bedroom square footage. That doesn't make a lot of sense. But there's something even bigger. There's nothing in the health code that says the effluent that comes from the septic system has to be evenly distributed over the area they're requiring. So in a standard gravity system, with the big 4-inch laterals that aren't designed for septics, they're designed for perimeter drains on your house or anything. That's what the septic contractors use. If you have a gravity system, you're mostly introducing 1 or 1.5 gallons of water at a time. The distribution system's hundreds of feet for a 3-bedroom home of laterals. Where do you think that 1 to 2 gallons goes? It goes through the first few holes and it keeps burdening that area closest to the distribution header.
Right.
There is a better way, and it's called low pressure distribution, where instead of the flows going into the tank and an equal amount going out to the field, it goes to a pumping station. And that doses the field 100 to 300 gallons each time it runs. And it has to have a different size lateral, smaller holes, further spaced. And what happens then is the health code says for low pressure distribution, the difference in volume delivered between any 2 orifices in the system, in the laterals, has to have less than a 15% volume difference. Now, to me, that makes sense because it's the soil that's arresting the phosphorus. So why wouldn't we take that step? That's probably a 20% increase in the cost of septic system. It is required in Antrim County for mound systems, but not for non-mounded systems. And it's even less of a cost increase for lakefronts because most lakefront systems have pump back systems anyway.
Right.
So why not pump them back under pressure and make sure we're burdening all the field?
Right.
The only argument I can see against it is, well, if we improve them, it'll be harder to get rid of them in the long term. That could be a legitimate argument. But so there's a lot to be learned there, and I don't think I've been on lakes in Michigan since 1982. I've been a riparian, and I've been in my lake association all that time. They've been different ones, but up here for the last 12 years. But septic systems have always been a major point of concern.
Right.
But yet the understanding of them is the same as it was back in 82. Well, the failed systems are bad and everything else is okay. Well, it's not that way.
And again, it starts with the legislation. The state of Michigan is not very strong on septic systems. They're certainly not strong on any maintenance or inspection. Once it's in, okay? We have those things to issue, those issues we have to address. So going back to where we need to go, we need to get solid scientific data so we can carry the argument forward. All right?
I know we're a little bit over
It's
time,
okay.
but I do have a couple of takeaways that I wanted to share. So I'm Jeannie Williams. I'm the executive director of the Three Lakes Association. One of my first takeaways is that I'm so proud to be part of the Three Lakes Association. We have done so much work And seeing it all today really makes me feel honored to be part of it. And also just so grateful that Jan has dedicated so much of his time and intelligence and experience to this project. I mean, it's been a tremendous gift and I'm so grateful. So thank you. I'm also grateful for everybody who's come today. It's amazing to see so many people who really care about this lake. There's something really magical about it. I mean, I think all bodies of water are magical and they draw people in. And I'm so grateful everyone was here today. The other thing I noticed is that we have a lot of decisions to make. So I don't feel like we're closer to knowing what we're going to do. I feel like I have even a broader spectrum of what's possible right now. I'm grateful for that. But I'm wondering, like, who in this room feels like they know what we should do? If you know what you think we should do, would you raise your hand? No, I mean, I don't know. No pressure. I'm just curious. I don't know what we should do right now. I think it's going to be a lot more discussion. I'm not asking for the answer. I'm just curious where you're at right now. It's just a gauge. And then my other question is, how many of you are interested in being part of those discussions about what we're going to do next? I'm kind of curious about that too.
It looks like we need more people in
Hold your hands up for
our
a
Zoom
second, because I
meetings.
think we want to just see this. Keep your hands up. I want us to look around and see, because those of us who are working on this want to remember who your faces are. It's okay if you don't want to or you don't have time, but I'm just curious. Okay, are we seeing this? Okay, thank you. Thank you so much for being here today. Your contributions have been incredible, and thank you for your willingness to continue.
Okay, some of us have volunteered to stay and do our cleanup of this room, but everyone else is free to go. Like I said, Torch Lake Cafe, the back room is reserved for us starting at 4:30, and I hope to see almost everyone, if not everyone there, once we're done with the cleanup. Thank you very much, everyone.